Good morning, everybody, and welcome to the 16th Annual Retina Symposium. It's really my pleasure to put this conference on today and to be with you all, either in person or virtually. We have a great program planned for you today and some outstanding out-of-town speakers, as well as some in-town guest speakers that are guests to UIC. First of all, I'd like to welcome to the podium Dr. Deeba Husain. Dr. Husain is a clinician scientist, director of the Medical Retina Fellowship Program, and the NIH R01 Metabolomics guru. She is probably one of the world's foremost experts in metabolomics, and in particular with regards to age-related macular degeneration. And for those of you who may not know Deeba, who's a good friend of mine, she came to this country after receiving her MD in India with honors and then repeated everything all over again in the US in order to work here. She was a US research fellow with Joan Miller, then a clinical fellow with Don Gass, Evan Gregutis, and then a resident and fellow at Mass Eye and Ear. She became a faculty member at Boston University, followed by a faculty member at Harvard, where she is today. She has gleaned numerous teaching awards, both at Boston University, as well as at Harvard, where she recently received the Harvard Mentoring Award in 2022. She's also received the AAO Achievement Award and the ASRS Honor Award. And today, Dr. Husain will be speaking to us on metabolomics and the pathogenesis of AMD. Deeba?
Thanks, Jenny, for that kind introduction. I'm excited to be here in Chicago for the Retina Symposium. And this morning, I'm going to talk about our work on metabolomics and what its role is in the pathogenesis of age-related macular degeneration. I have no relevant financial disclosures. So as we know, AMD is a leading cause of blindness in the developed world, and the third leading cause of blindness in people over the age of 50 in all over the world, worldwide. AMD is a multifactorial disease where genetics, our diet, our lifestyle, and exercise play a role in the manifestation of the disease. A lot has been discovered in the area of genetics and AMD. There's a large genome-wide association study organized by the International AMD Genetics Consortium, which has shown very significant correlations of several genes with AMD. In fact, AMD is one of those diseases with most significant genetic associations as compared to any other multifactorial disease. And this study has shown that there are significant contributions of rare and common variants, 34 loci and genes with rare variant burden to age-related macular degeneration. And yet, the interaction among these genes and how they promote AMD is not fully understood. We believe that molecular traits on a quantitative scale may be more associated with causal genes for the disease process and better contribute to the pathogenesis of AMD. So what are the major unmet needs of this blinding disease? First of all, there is limited understanding of the pathogenesis. Secondly, there is lack of accessible and reliable biofluid biomarkers of this disease, both for diagnosis as well as for prognosis. And then there is a limited amount of treatments. We have only the first approved treatment at this time for dry AMD, including both the early and the late stages. So we believe that metabolomics may be an appropriate approach to address these needs. Metabolomics is the qualitative and quantitative analysis of all the endogenous small molecules called as metabolites, which are less than 1 kilodalton in our body fluids as well as in body tissues. And it reflects the cumulative interaction of the genome and the environmental exposures. That is the reason the metabolites are supposed to be very closely related to the phenotype in complex diseases. So here is the same concept in a diagram form to help explain it. So the study of genes is genomics. Downstream from the genes is the mRNA, which the study of which is transcriptomics. Downstream from that is proteins, which is proteomics. And downstream from that is metabolites, which is closest to the phenotype of complex diseases, specifically where we are worried about environmental exposures, both for the causation of the disease and the metabolites. So metabolites can be measured in two ways. It can be measured by mass spec, or it can be measured by NMR. Mass spec is used for both targeted and untargeted approach. It's very sensitive, but the disadvantages are that it requires a sample preparation, and it's difficult to diagnose new compounds with this method. The NMR, on the other hand, is a better initial untargeted approach, which is more reproducible, but the disadvantage being that it's less sensitive. So in our work, we decided to use the mass spec-based metabolomics in AMD cases and controls with the aim of finding or exploring pathophysiology of AMD and establishing novel potential biomarkers. This is a longitudinal ongoing study where we are doing annual follow-ups on our cohort. And at this time, we are collecting year 8 data on our cohort. We have prospectively recruited patients with AMD and a control group of more than 50 years without any vitreoretinal disease. We're collecting our own patients from Boston at Mass Eye and Ear and our collaborators in Coimbra. At this time, we have 900 patients, and it's still open to enrollment with about 300 controls and 600 AMD patients. We've collected extensive information on our patients, which includes their medical history, their physical activity, the food frequency questionnaire. We've done dilated eye exams. We've done multimodal imaging of both the macula as well as the periphery using color photos and autofluorescence. And then we have done OCT with EDI. We've collected fasting morning blood and urine samples on which metabolomics has been done within 30 minutes of collection, and the samples are stored at -80 for analysis. And we've done genomics on all of our samples. The metabolomics has been done using the ultra high performance liquid chromatography mass spec, which was done by at Metabolome Inc. Some patients, there's optional extra testing, which includes both functional testing, such as dark adaptation, microperimetry, and contrast sensitivity, and then some extra imaging, which includes more swept source, peripheral OCTs, OCTA, and macular pigment density. So the first analysis, the exploratory analysis we did a few years ago, was to look whether there was any difference in the metabolomics between AMD and controls. And this was a pilot study done with 30 patients in each group, 30 controls, 30 early, 30 intermediate, and 30 late. These patients were pretty well balanced, except for the age, the late AMD patients being older than the other cohorts. And we found 878 metabolites in our cohort. The statistical analysis was done in collaboration with our collaborators over at the Channing School of Network Medicine at Brigham's by Professor Jessica Su and her postdoc Rachel Kelly. They helped us with principal component analysis to check for clustering, whether there was any clustering of AMD patients, which was different than the controls. They helped us with multivariate logistic regression analysis, accounting for the known confounders of AMD, such as age, BMI, smoking status, to identify AMD-associated metabolites. And we did receiver operator curves or rock curves for discriminatory capability of significant metabolites. And to look for stage of disease, ANOVA tests were done. So here's what we found. Accounting for the confounders with a p-value of less than 0.05, there were 87 metabolites that were significantly different between AMD patients and controls. And as we got more stringent, even with a p-value of 0.01, we had 7 metabolites that were significantly different between the two cohorts. And interestingly, when we looked at what these metabolites were, majority, 83% in our initial analysis were lipids, with a small amount of amino acids, peptides, cofactors, and purine and pyrimidine amino acids. Then we looked at what pathway these metabolites were in. And there was one pathway that stood out very significant and separate from the other pathways. And this was the glycerophospholipid metabolite pathway with a p-value of 4.7 10 to the power minus 9. We then looked at stage of disease and found that 48 metabolites were different across different stages, early, intermediate, and late. And the most significant metabolites again showed up in the lipid pathways, specifically glycerophospholipids was most significant in this cohort. So what are glycerophospholipids and what may they be doing in AMD? So glycerophospholipids are the major components of cell membranes. They initiate the oxidative stress, and it's persistent. That has been shown in various neurodegenerative and neurological conditions. They play a role in neural cell proliferation, differentiation, and apoptosis. And actually, very interestingly, in Alzheimer's, there was some recent work at that time that was showing a major role in pathogenesis, Alzheimer's being a neurodegenerative condition, just like we believe AMD is a neurodegenerative condition. And actually, some early sort of plasma lipids were shown to have predicted values in conversion of the mild form of Alzheimer's into the more severe form. So this was exciting for us to find these correlations between our work and the work in neurosciences. So this was published in ophthalmology about 6 years ago now. And this encouraged us to continue this work. By the time this work was analyzed and published, we now had 500 patients in our cohort. And we needed to validate the initial pilot work we did with a larger cohort. So we looked at our 500 patient cohort, which were 200 patients from Boston, 300 from Coimbra. We did 2 types of analysis, similar to the first analysis where we looked at AMD versus controls, and then stage of disease using the 2I models. We analyzed each of our cohorts separately and then did meta-analysis in them and then looked at pathways and did ROC curves to look at the significance of the metabolites we were looking at to find metabolomic profiles in age-related macular degeneration. So what did we find in this study? Looking at AMD versus controls, again, controlling for the known confounding factors, we found 69 metabolites that were significantly different with a p-value of 0.05. In even a more stringent way with accounting for significant Q values, which is multiple comparisons for multiple testing, we still found 28 metabolites to be significantly different. And interestingly, again, the largest signals were lipids, 35.7%, and amino acids, which are 28.6%, and then some other carbohydrates, cofactors, and peptides. And when we looked at the pathway analysis for these, the purines, sphingolipids, taurine and hypotaurine, and glycerophospholipids were the major signals that were significant that were seen in this work. We then looked at stage of disease, early, intermediate, and late, and again, accounting for the known confounders, we found 147 metabolites were significantly different, 67 of which were still different with very stringent Q values. And 22 of these were the ones we had seen with AMD versus controls. And again, 50% of these metabolites were lipids, with 30% being amino acids, and then a smaller portion of carbohydrates, peptides, energy-related, and cofactors. That we found many more pathways looking at staging of disease, which are noticed here. So to summarize that work with a meta-analysis, we found that there were 4 different metabolite pathways that were significant between AMD and controls and stage of disease, including glycerophospholipids, which was seen in our pilot work. We also saw purine metabolites, and these are play a role in energy metabolism. They're responsible for photoreceptor integrity, and they have been shown to be increased in advanced AMD in previous work. We found significant levels of taurine and hypotaurine, which are most abundant in amino acid within the retina. They are neuroprotective, and they are stabilizers of the photoreceptor membranes. And then we found glutamate, especially the nitrogen metabolites, which play a role in oxidative stress. The next logical step was to correlate the metabolomics with genomics, because that is the upstream of where the metabolites are formed. And these associations can provide unique insights into the biological significance of both the genes as well as the metabolites. So they kind of validate the both findings. SNP metabolite associations can overcome limitations of genome-wide studies. And we know that we have very significant genome-wide results in AMD. So this was trying to find the biological significance of the AMD risk genes by trying to understand more the functional role of these genes. And to do these, we did association with the SNPs in our population and the metabolites that we found in our cohort. The term used is metabolic qualitative trait loci, or MQTL, which is the DNA locus that correlates with variations in metabolites. To do this, we did metabolomics on our cohort, like we had done before, and then we did genomics on our cohort using the OmniExpress array, which was done at the Broad Institute, and we found 4,795 AMD SNPs in our cohort. This analysis was done with the help of our geneticists and biostatisticians at the Harvard School of Public Health. They did linear regression models on these MQTLs, again, adjusting for the known confounders, and for the first 10, accounting for the first 10 SNP principal components and first 10 metabolite components. And they did Benjamin-Hochberg procedures to account for the false discovery rates. Again, just like in our last cohort, each cohort was analyzed separately, and then meta-analysis was done to look for the significant MQTLs. And in this work, we found 2 genes that were significantly associated with metabolites. One being the ASPM gene, which was associated with the amino acid group, leucine, valine, and isoleucine group, and LIPC, which again is a lipid gene, which was associated with 25 MQTLs, and several metabolites, which were all in the phenylethylamine, or which is really the glycerophospholipid pathway. It is a subpathway of the glycerophospholipids. So this was exciting to see that this was kind of all pointing to the same direction of the lipid pathways in AMD. This work was published in Ophthalmology Science. So there is still a lot to learn and know. There are several limitations of our work. Most of our cohort patients are Caucasians. The sample is about 1,000 now, but still it's small. For genetic studies, you need hundreds of thousands. But for metabolomics, larger the study, the better it is. For now, most of the work is plasma measurements. There are other body fluids and tissues that can be looked at. And there's also a constant criticism that metabolites vary with various conditions, and they do. And that's why this work kind of needs validation at every step. It's exciting to see that there is a lot of interest now in metabolomics and AMD, and these are some of the larger groups that are now working on it, the large European IRIS consortium. And I'll talk briefly a little bit about their work, which is very exciting. Singapore is doing some work on their version, the polypoidal version of AMD. And then the National Eye Institute is just starting with their metabolomics AMD program in the AREDS population. So talking about the IRIS consortium, this is actually the largest metabolomic genomic association ever published in AMD. It was recently published in 2020. This is a meta-analysis of 5 European cohorts with about 2,000 patients and 4,000 controls. The way they have checked the metabolomics is with the NMR process, which is different than ours. And they have used just the AMD SNPs, the 52 SNPs, to look at the associations. They have used univariate logistic regression analysis, so it's a little bit less stringent than our analysis. And they found 7 AMD variants, which were associated with 34 metabolites, and the strongest associations were all near the lipid metabolism genes. So ABCA1, CETP, ApoE, and LIPC that we found. And the metabolites that they were associated with were also in the large and extra large HDL. So the lipid classes is what was showing up in this. So I'd like to summarize by saying that metabolomics in AMD is an interesting field. It can help better understand the pathogenesis of AMD, specifically when we are using it in the multi-omics area in combination with genetics and hopefully microbiomes. It points to potential biomarkers of AMD and therefore also point to the druggable targets for disease and therefore ushering the era of precision medicine in the management of age-related macular degeneration. We are continuing our work in this area, and I'd like to thank our team. No work is possible as an individual. It's the village of people who help us do this work, both retina specialists, comprehensive ophthalmologists, the biostatisticians and epidemiologists, and of course, the core people, our research fellows and assistants who help us do all the work. And these are the institutions involved and the financial support without which this work would not have been possible. Thank you.
Deeba, that was fantastic. You're really bringing us to the forefront of the research in AMD. And it's really wonderful to see how it correlates with things that we know are associated. Some of the genes that we know are associated, the lipids and all of these other components. I'm curious, do you think that you would use this metabolomics in the clinical trial setting? Or is the number not, big enough to see whether some of the patients who respond better to some of the drugs or who have are harder to treat per se have a different profile? Can you address that, please?
That's a very interesting question, and this work is actually ongoing. We are looking at our own cohort at the Retina Service of patients that respond to anti-VEGF therapy and non-responders. So this is, and actually there's Singapore Eye Group has published their work on PCV responders versus non-responders. So this is an excellent question. This is something that needs more work to really understand whether we will be able to incorporate it in clinical trials, but it's something that we are certainly looking at.
Great, thanks so much. Are there other questions for Dr. Husain? Dr. Agarwal, can you give her a mic, please, or come up here? Give her a mic. Sorry, 1 second, Anita, so we can hear you.
Do you think repeating, resampling the same cohort again, maybe a year later or something, will that make a difference? Have you tried something like that?
Yes, so we actually are collecting data every year. We just published on our 3-year, and we are in the process of analysis of our 5-year. That's an excellent question because metabolites change so much day to day, depending on our diet, our exercise. So, first of all, there is some component of just the variability causing a change, but we are also actually looking at progression of disease to see what progression of disease will do to the metabolomics. The year 3 data is actually showing that, interestingly, it's not the lipids, but it's more the amino acids and the proteins that are changing in those people where progression happens. It's a very small cohort still. At the 3-year, we have only 100 patients with only like 15 that actually changed. But we do have that data, and we are in the process of looking at it.
Can I ask a question?
Sure.
Yes, Dr. Skandra, question? We can hear you.
Anyway, Deeba, it was great talk. I was excited to see your data. We started analyzing some microbiome and fecal metabolomics profile for my patients. And actually, we start seeing a separation between the GA-only patients and those that are predominantly wet. Do you see any pattern in the subtypes of late AMD with the metabolomics, the untargeted metabolomics?
That's a great question. Everything is about numbers, and the cohort, the way it is, majority of our patients are intermediate. To start with, we were focusing more on the early part of the disease rather than the late. So the initial cohorts, we did not have the numbers enough to look for any significance. But actually, now that the study has opened up, we are looking at that difference. But I don't have the results yet of that data.
Any other last questions for Dr. Husain? If not, thank you so much, Deeba. That was fantastic, and we're really pushing the field forward. It's now my pleasure to introduce our next speaker, Dr. Amani Fawzi. Amani is the Cyrus Tang and Lee Jampol Professor of Ophthalmology in the Feinberg School of Medicine at Northwestern. And Amani actually was one of my former fellows at USC. Amani came to us from Egypt after graduating there and completing her ophthalmology all with honors. And she did a research fellowship at UCLA, loved it so much in LA that she decided to do an ophthalmology residency and then a fellowship with us. I say us when I was at the Doheny Eye Institute. Did a fantastic job. We kept her on faculty. And then eventually she came to Northwestern. So they're very fortunate to have her. So Amani spends her time between clinical and surgical practice and her NIH-funded lab studies of animals for ischemic retinopathies, and her clinical research focuses on novel functional retinal imaging approaches, including OCTA, visible light, OCT, and hyperspectral imaging. She has been awarded the Honor Award by the ASRS, the Achievement Award by the Academy, and the Young Investigator Award from the Macular Society. It's my pleasure to introduce Dr. Amani Fawzi. She will be speaking today on biomarkers for AMD progression, looking for clues on ocular imaging. Amani.
Well, thank you so much, Jenny. It's a great pleasure to be back here at the UIC for this session. It's been great hearing Deeba's talk and trying to tie this back to imaging. So my task from Jenny was to sort of think of clues that we could see on imaging that could predict for us the patients who are going to go on to have late AMD. So I thought about the highest sort of yield, and I have no relevant financial disclosures. I thought about the highest yield markers that I see in my clinic that really clue me into patients who are getting sort of hitting that point of no return. And it's those, I started to think of them in clusters. So for atrophic AMD, those are the top, I'd say top 4, top 5 reasons to worry about a patient's eye going on to atrophic AMD. And for neovascular AMD, it's a little harder because we don't really have great biomarkers, but I think double layer sign and subclinical non-neovascular AMD are probably the best ones. But as you can see here, drusen volume and hyperreflective foci show up in both. So it's a little confusing sometimes to figure out who's going to do what, but walking you through this talk hopefully will give you some clues. And that's the purpose of this talk. So I'll start with this idea of nascent GA. This was something that Robin Geimer first proposed. And it's that as drusen are regressing, the retina on top of them are sometimes already going into atrophic AMD. And there's some subtle clues on the OCT that couldn't clue us in. So for example, here's an example of sort of an earlier stage where you could see this diving in of the outer plexiform layer into the top of the drusen. And that basically tells you that the outer retina is almost completely gone there. So when you see that, you know this area, as the drusen regresses, is probably going to go into atrophy. And this is what nascent GA is. It's basically atrophy related to drusen. That's sort of the definition of this entity. And as the drusen continue to regress, you see the sagging of the drusen right there. And you see this sort of hyporeflective area in the outer plexiform, this wedge-shaped area. And that's the next clue of continuing progression into atrophic AMD. So when you see that, especially when you see this underlying hypertransmission into the choriocapillaris, into the choroid underneath, and you can see that even in the earlier stage, you know this region there is going into atrophy most likely. I don't know if there's a point of return here. This eye is unfortunately going down that path. The next two features are hyperreflective foci and drusen volume. And that's again sort of a shared theme here. This is a paper from Vasada, probably the year I left Doheny, 2013. But this is a large database of the eyes at Doheny, where they basically looked at biomarkers that predicted progression to atrophy. And this hyperreflective foci showed up back then. And it's basically these white spots that are, you could think of them as RPE cells running away from the RPE layer, which sort of helps me understand what's going on there. They're probably not RPE cells. They're probably macrophages diving down and eating RPE cells or inflammatory cells or something else. But just to sort of think about it, sort of conceptualize it, you may want to consider that idea. So these hyperreflective foci really tell us that something bad is going on in this eye. And as you can see here, the odds ratio of going on to dry atrophic AMD is 30. So that's pretty high if you have these hyperreflective foci. The other high yield lesion here is this heterogeneous or versus homogeneous drusen. And that's a concept that's also going to show up over and over again. So I wanted to illustrate that. And this is basically drusen that's got different reflectivities within it. And so you have a hyperreflective area. I think the mouse is working. So there's a hyperreflective area right there and a hyperreflective area next to it. And that's a type of drusen that really should clue us into something bad is going on. It's a drusen that's collapsing. It's a drusen that's dying. Some people call this feature ghost drusen, basically a drusen that's hollow inside. And sometimes they have this hyperreflective line underneath them, and that's a sign of calcification. So these are probably, I'll tell you later what they are, but my thought is that they're probably calcific drusen in evolution. So you have this hyperreflective cap, hyperreflectivity inside, and eventually they become totally hyporeflective and then collapse and you get atrophy. So be on the lookout for these lesions because they're also very high yield for progression to atrophy. And then maximum drusen height increases the odds by 4 times. So drusen volume is important, and we'll get into that a little bit later. But this hyporeflective, mixed reflectivity drusen and the hyperreflective foci are really important clues. So next, I'll get into drusen volume. And it was Voss again that looked at this series of eyes shared between his group, the Boyer group, and Phil Rosenfeld's group, looking at sort of this volume question, right? What drusen volume is bad? And so here they came up with this idea that for every 100 micron increase in drusen volume in the center 3 millimeter, there's an increased odds ratio by 1. But the important number to remember is this, is a threshold volume of 0.03 millimeters in the central 3 millimeters sort of circle. And that you can come up with from software on Cirrus usually. So if you're using the Spectralis like we do, it doesn't spit out that number. But if you have a volumetric center macular scan on the Cirrus, it spits out that volume. So if you have that threshold volume of drusen, your risk is really high. In their series, 19 out of 22 eyes that had that threshold volume metric developed either atrophy or neovascularization within 2 years, 24 months. So basically a very, very high yield number if you use the Cirrus to look for that. And it may be a good reason to use the Cirrus, one of the reasons. So Ursula's group looked at what happens to drusen over time. And if you could look at this graph here, it sort of shows you that drusen keep growing, and then at some point they collapse, right? When they reach sort of a threshold of they can't get bigger anymore, they collapse. And then they looked at that collapse point, and very important things happen at that collapse point. So 22 eyes had this drusen collapse, and 30% of those eyes, so 4 developed macular neovascularization, and 3 developed GA. So when drusen go away, it may not necessarily be a good thing. So you need to be on the lookout for, as drusen grow, you have to worry, but also as they go away, you have to worry because 30% of the eyes that have drusen collapse or drusen disappearance can develop an end stage sort of atrophy or neovascularization. So we always think of drusen leading to atrophy, but in this series, they opened our eyes to this idea of drusen collapse leading to neovascularization. Here's an example of those eyes, sort of a drusen volume here by red getting bigger and bigger, drusen collapses. And then 3 months later, this eye develops neovascularization. So you really have to be keyed into these eyes and watch them closely. If drusen is getting bigger, watch them closely. If it collapses, you watch them even closer because an end stage may be coming up. So next is this very cool paper from Robin Geimer and Ursula, where they basically looked at competing theories, right? Is color photography better, or is OCT better, or are combining them even better to predict progression? So they started out with color fundus photo grading, and here it was simple: pigment present or absent, a binary outcome. That is model 1. Model 2 is minimal OCT biomarkers, and that's the two that we already know: hyperreflective foci and drusen volume. Is that predictive? And then number 3 is extended OCT biomarkers. So they looked at 7 different biomarkers that Ursula's group had come up with, complicated OCT biomarkers. And then number 4 is a combined sort of 1 and 3. And looking at competing these models against each other, which of them is more predictive. And the result is really cool because it tells us that OCT is probably enough. So if you look at model 2, the OCT parameters, which is just hyperreflective foci and drusen volume, and they added age in the model, it had a very, very high AUC. So there are some cases where color may be helpful. So here's sort of the top 2 examples are cases where color photography or OCT were equivalent because they had pigment and they had high drusen volume. Pigment and high drusen volume makes the 2 models equivalent. But if you have an eye where there's no pigment and there's a high drusen volume, then OCT becomes more reflective of the outcome, more predictive. If you have the opposite, an eye with more pigment and barely any drusen, then color photography is more predictive. But overall, as you can see here, the OCT, minimal OCT biomarkers, hyperreflective foci alone and drusen volume had a very high AUC, 0.82, which was equivalent to all the other sort of complicated markers. So if you just use OCT, hyperreflective foci, drusen volume, it'll get you there. You can predict really a poor outcome for these eyes. Now, this is a study from the AREDS group. Cindy Toth and Emily Chu collaborated on this. So here they're looking at how early are these markers predicting the sort of atrophy outcome. So they looked at 4-year precursor lesions, 3-year precursor lesions, and 2-year precursor lesions. The interesting thing is there's drusen complex volumes can predict even 4 years ahead of time. So this is interesting. The other interesting thing is photoreceptor loss. If you do it volumetrically, if you look at photoreceptor volume below the 95 confidence interval for a healthy eye, that can also predict as early as 4 years that this specific area is going to develop atrophy. If you look at hyperreflective foci and drusen substructure, those show up later. So 3 years predictors, 3 or 2 years predictors, but photoreceptors and drusen complex volume start out very early. So I'll talk about this drusen complex features that sort of keep popping up. So here you see that again, the empty drusen, that drusen with no sort of reflectivity inside, the hyperreflective drusen, which we have called ghost drusen or I don't know, low reflective drusen. Those are very, very strong predictors of atrophy. So you could see here, this is where that drusen was before, now it's atrophy, and here's a neighboring drusen that's evolving into this. And these, if you look at the colors, are all calcific drusen. So these features on OCT are very well correlated with calcific drusen. Calcific drusen, we've known for years that they predict atrophy. So basically, we're talking about calcific drusen here. And then this next paper tried to tease out this question about hyperreflective foci and drusen volume? Do they predict atrophy or do they predict neovascularization? And what are the specific features? This is a very cool paper from Ursula again, where they looked at the Harbor fellow eyes and looked at sort of baseline drusen volume and baseline hyperreflective foci volume in the middle of that 3 by 3 millimeter circle. And so here's an eye that went on to develop macular neovascularization. So you could see here the drusen distribution and the distribution of hyperreflective foci. Now this eye developed atrophy. So now the drusen distribution looks different and the hyperreflective foci looks a little different. And here's an eye that did not have any progression. So the lower drusen volume and lower hyperreflective foci. But when they looked at the distribution, they saw some very interesting clues. So if you have a high drusen burden in the center 0.5 millimeter in the center point, these eyes develop macular neovascularization. Versus if you have a high hyperreflective foci volume in that center 0.5 millimeter, these eyes develop atrophy. So basically by the location, if whatever is going on in that center 0.5 millimeters will tell you which endpoint this eye is going to have. And so if you pay attention here, I have a question at the end. So stay awake. And then they looked at progression of these biomarkers. And again, they saw that faster increase in drusen volume can basically predict progression to neovascularization. And then this fluctuation of hyperreflective foci volume can predict atrophy. So basically what is happening over time to those two biomarkers, if you pay careful attention, you have a volumetric map, can actually predict who's going to go on to have which outcome. And finally, for the next 2 minutes, I'll talk about this non-neovascular AMD or non-exudative AMD outcome. And Dr. Rosenfeld, I was hoping he was going to be here, really opened our eyes to this feature by showing us that a lot of these low lying sort of double layer signs can be harboring new blood vessels if you look in swept source OCT. And why do we care? Because these eyes have a very high risk of developing active neovascular AMD within 2 years. This is their paper showing that their cumulative risk is about 24% of progressing to neovascular AMD. So this is almost as close as you can get to knowing who's going to go on to develop neovascular AMD. If you have this low-lying sort of hyporeflective double layer sign, and if you don't have, even if you don't have OCT and geography, you could sort of keep an eye on those eyes because they could develop neovascularization. Here's an example of one of our patients. He had a very, very shallow double layer sign, but it was vascular. And this specific patient ended up 5 years out now, no neovascularization. So remember, it's 24%, not 100% predictive. So they're usually asymptomatic, but they need to be watched very closely. This is Mike Hyperman when he was a resident with us. We looked at sort of the entire literature, and it's the predictive sort of value ranges from 6% in 2 years to 25% from that Phil Rosenfeld paper. So really wide range of progression, probably depending on the other risk factors for this eye in this patient. So what if you don't have an OCTA? What if all you have is an OCT, right? Every time, can you predict by looking just at the OCT, how many times are you right by looking at the OCT and predicting neovascularization? This is another nice paper by Phil, where they looked at the predictive value of just having double layer sign for the presence of neovascularization within that lesion. And they saw that in early AMD and intermediate AMD, the predictive value is very, very high, almost 90% from a senior grader. But where it really becomes a little problematic is when you have sort of atrophic AMD, the predictive value falls a little bit. So there's other lesions that can look like this in late AMD. So not all of them will be neovascular. And here's a paper which explains why. So this is a nice paper from Christine Curcio, where they had a histopathologic sort of donor eye confirmation after the patient passed from an OCT. So here you have a very long double layer sign, so that extends from here all the way to that yellow area. And as you could see from this paper, some parts are just basal laminar drusen, especially if you look here, there's what looks like a hyporeflective area here, but this was only basal laminar drusen on histopathology. But some parts actually harbor subclinical macular neovascularization. So sometimes it's really hard to tell. Voss talks about this thick double layer sign or high volume double layer sign. As you can see here, there's two reflectivities, a hyperreflective area and a hyporeflective area. And that may be sort of the better clue. The bigger the double layer sign, the more likely it is to harbor neovascularization. So as you can see here, two reflectivities, one hyperreflective, one hyporeflective. These sort of thick double layer signs are more likely to harbor macular neovascularization. And why do we care? Even more now we care because of the treatment for atrophic AMD. This is the Philly phase 2 trial of complement inhibition in geographic atrophy. This is a very nice study trying to explain why there was such a high risk of neovascularization in these eyes. They looked back at eyes that had the double layer sign, and of those eyes that had at baseline double layer sign, 50% developed active neovascularization when they were treated with complement inhibition. So if you have a patient with geographic atrophy and you're considering starting complement inhibition, be on the lookout for double layer sign in these eyes, because if you're treating an eye with double layer sign with complement, the chances of developing active neovascularization are super high, 50% according to this. So really important to think about this. And then finally, I was going to talk about this calcific drusen story. So this is a nice paper that just came out from Phil Rosenfeld, where he looked at what happens to drusen on sort of volumetric autofluorescence, this slab below the RPE, and the volumetric drusen volume. So here's an example of a soft drusen that went into calcific drusen. So if you look at the infrared, the calcific features become hyperreflective on infrared, and they become hyporeflective because they mask the signal on the slab below the RPE. And over time, that sort of hyperreflectivity and the blockage of signal becomes more intense. And if you look at the OCT in these eyes, you could see that what we've been talking about, the different reflectivities within the drusen in the calcification. And so finally, here's my question to you. This is one of my patients that sort of is interesting, tries to tie together everything we've talked about. So this patient saw me when she was only 56 years old. She had a family history. Her mom had AMD, and this was 2015. So she had big drusen in both eyes, right? So I was on the lookout. 4 years later, her drusen got even bigger. So I'm really on the lookout now. I see Jenny cringing. She's young, but her drusen are giant. So giant drusenoid PEDs, and here's some sort of hyperreflective features. It's not quite dense yet, but I'm really worrying about her, and I'm bringing her in every 4 months because I'm predicting sort of a poor outcome, and the drusen become bigger and bigger. And this is December 2019. There's some slit-like feature here, and I don't know what that is, except that the drusen is growing fast. And so in September 2020, the drusen regressed completely in her right eye. So here's the right eye. So what do you think happened? I won't tell you. I want you to predict from all I've told you. What do you think happened in the right eye? So this eye developed drusen regression. So the things that are in her favor is she doesn't have any hyperreflective foci here. I don't see any of those calcific drusen, and she's young, okay? So the drusen regressed completely without anything happening. So this is one of those 75% of drusen regression without anything happening. So yet, okay, but this is 2020. I have more information. And then there were some features that looked like they were calcific in this eye, this right eye. But then over time, the right eye stayed good, right? Nothing happened, but the left eye started to develop hyperreflective foci in the middle, in the fovea, right? Dr. Skandra, remember? So, and they kept getting worse. This left eye kept getting worse. The hyperreflective foci were everywhere. There was this sort of thickening on top of the drusen, and the drusen kept getting bigger and bigger and more hyperreflective foci. And guess what? Is it last month? Last month, the drusen regressed in the left eye just for this talk. And now what happened, Dr. Skandra? What is it? Statin. Ooh, no, didn't look. And I don't know if she's on the Mediterranean diet either. So nothing, nothing happened. I guess I don't know yet because there could be some atrophic lesions there because, she has a ton of hyperreflective foci, the drusen regressed. There's probably a little bit of atrophy, but at least centrally, she didn't go too bad. But the story's not ended. I'm following her. This is only 3 weeks ago. The right eye has stayed stable now, 4 years out. Absolutely nothing happened. She's developing some hyperreflective lesions there, but still no end stage progression. And so in summary, drusen volume and hyperreflective foci, these are your friends. Threshold drusen of 0.03 millimeter cube. High drusen burden in the center 0.5 millimeter could predict macular neovascularization. Drusen regression may herald late AMD in 30% of the eyes, and I showed you an exception to that rule. Hyperreflective foci in the center 0.5 herald atrophy, so keep an eye for that. And of course, multirefractive drusen, ghost drusen, calcific drusen, these are bad players, and the subclinical sort of double layer sign as a predictor for neovascular AMD. I thank you for your attention.
Thank you so much, Amani. That was a wonderful overview, quite excellent. So my question to you is, in the clinic, do you actually use this in determining when you're going to see your patients back? And can you inform us about that? And then whether AI can also be helpful in this regard?
Yeah, for sure. For sure. I mean, I'm waiting for Ursula's AI to come up on our Spectralis so we can all do that in our clinics. But definitely when people's drusen grow bigger, I watch them closely. So instead of every 4 months, I might say, here's the Amsel grid. Can you please pay attention and, see me in 3 to 4 months. And if anything happens, I sort of have an extra heart talk with them about the progression. And if drusen disappear, I also watch them closely. And of course, with the double layer sign, I'm always sort of cautious.
Mani,
Anita,
can you
Dr. Agarwal.
correlate any of this to what's happening in the capillaries? Because that I think is really where the meat is. Right?
Great,
the drusen
great
is only what
question.
we're seeing up on the top,
yeah.
and what's happening underneath is really what's.
That's a great question. I'm looking forward to Phil's talk tonight in the afternoon because I think he's done a lot on the choriocapillaris and progression. The problem when you have these hyperreflective drusen and they're casting shadows below them, it's very hard to know what's going on in the choriocapillaris. But Phil has done a lot of work on the choriocapillaris, and I'm excited to hear his talk later.
Dr. Roxana Mirza, you have a question?
Great talk, Amani. Did you see anything about the rate of drusen regression? Sometimes we see draping and it gets confusing. Is there neovascularization starting there, or is it? And oftentimes there's not. It's just that draping. And I think that happens because the rate of
Yeah, no,
is
that's
more.
a great question. I think Saraf has written a couple of papers about this fluid and dry AMD. So when you have drusen and what looks like fluid on the OCT and how to tell if there's neovascularization or not. And I think the jury's still out on these eyes. So when I see that fluid draping, whatever, I bring the patient back in a month because I really cannot tell a priori if this is just draping or if it's if it's neovascularization happening. If the drusen is super solid and there's not a single evidence of fluid under the RPE, I'm less worried because there's unlikely to be neovascularization if there's no hyporeflectivity within the drusen. But I still bring those patients monthly. And if they continue to be asymptomatic for 2 or 3 months, then I go back to the regular follow-up. But great question.
Great. Any other questions for Dr. Fawzi? Great. Thanks so much, Amani. If not, we will move on to our next speaker, Dr. Larry Ulanski, who is one of my valued colleagues here at University of Illinois. Larry is clinical assistant professor in ophthalmology here at UIC, and he's also chief of ophthalmology at the VA at the Captain James Lavelle Federal Healthcare Facility, which is the North Chicago VA. Larry hails from Canada, and he actually did a fellowship in Canada. He's originally from Detroit, though, so we'll let that one go. And he actually also did a uveitis fellowship here at UIC following his tumor fellowship. And we were fortunate that he chose to stay with us as part of the retina faculty. Prior to going to North Chicago VA, he was at the Jesse Brown VA. Larry was the PI of the ARED study here at UIC and was also involved in multiple clinical trials. He'll be speaking to us today on what is photobiomodulation and does it work? Many of you have probably heard about this. It sounds a little bit like out there, but if you were at the COS ICEPS meeting, there is some scientific basis for photoreceptors in other parts of our body that do respond to light. So Larry, go ahead and inform us about photobiomodulation and does it work?
Thank you for the introduction, Jenny. Let me just make sure there was a comment on the chat about not seeing the screen, not seeing, can everyone give me a shout out that you are seeing what you're supposed to be seeing? We did some tweaks here. Let me put up the chat here so we can see the chat. Yes, thank you. Thank you, Dr. Rosenfeld. All right. So Larry Ulanski, I feel kind of like I'm squished between Amani, Fawzi, Dr. Rosenfeld, Jenny. And so I think Jenny gives me these talks either for comic relief or because she knows I'm just going to get the schedule back on time because I kind of, that's the way it works. So I appreciate the offer to speak, Jenny. So the photobiomodulation doesn't work. My disclosures, I have no disclosures. I'm a federal employee, and that's all I can say about that. Photobiomodulation, when I saw the title, I was like, oh, Jenny, photo, okay, must have something to do with light. Biomodulation, okay, must change someone's biology. I'm like, but we do that every day in the clinic, Jenny. I can't believe Jenny's going to give me a talk about PRP and destroying people's retina. So that was my first initial thing, but then I actually did some research, and photobiomodulation is defined as a form of light therapy that uses non-ionizing light source, including lasers, light-emitted diodes, or visible light to change our physiology. It elicits a photophysical and photochemical event at the cellular level. And this is a little diagram here of the light, near infrared light, breaking up some bonds and stimulating cyclic AMP. So in our world of retina, this is our curve of light. This is argon laser versus krypton laser versus green laser. This is where we live, and this is ionizing radiation. It destroys tissue, it creates burns. It's what we do. It's how we live in retina. But this is not what this is about. This is about these ultra more near infrared, high short wavelength or longer wavelength type of laser frequencies that don't necessarily cause the same effects that we use in retina. Photobiomodulation is a therapeutic dose of light to impair or dysfunctional tissue leads to a cellular response mediated by a mitochondrial mechanism. So the key is the mitochondria. The promise of this is it alleviates pains, it reduces inflammation, it promotes wound healing, it promotes tissue regeneration, reduces apoptosis. It sounds like one of those wonder drugs that you get at the pharmacy. PMB, photobiomodulation, replaces low-level laser light therapy, cold laser, or non-ablative type of laser therapy. It uses anywhere from red to near infrared light sources, so 670 up to 1,000 nanometers. The power density is very low, so you're not trying to create a thermal effect, you're just trying to stimulate the cells. It stimulates the cytochrome C mechanism. That's the primary target is the cytochrome C complex found on the inner membrane of mitochondria. It's vital for electron transport chains. It's kind of like stimulating the engine. Let's kick it up a little bit. Increases the production of ATP, adenosine triphosphate. Light absorbed by the cytochrome C oxidizes and stimulates this transport chain. So, quick quiz. How many ATPs are created in the Krebs cycle? I got a 34. Come on. This is like the one thing I remember from like college, bio. All right, the answer is, no more guesses? 36. 36 in aerobic and 38 in anaerobic conditions, because you get 2 extra ones when you're in an anaerobic condition. So it modulates the reactive oxygen species. It supposedly produces free nitric oxide, which is a powerful vasodilator to help blood flow. And this photobiomodulation in the eye, what is it doing in the eye? So when you treat the eye with this type of light therapy, it's supposed to stimulate complex 4, which co-localizes on the retinal layers, rich in mitochondrial cells, such as the nerve fiber layer, the ganglion cell layer, the inner and outer plexiform layers, the photoreceptor inner segments, the basal surface of the RPE. It stimulates everything. And this complex 4 is everywhere. And it is activated by near infrared light, and it increases this respiratory chain. So in real life, there is real life photobiomodulation going on every day in our bodies that we don't really think about as a therapeutic system. The visual cycle, rhodopsin. Rhodopsin undergoes cyclic decomposition from cis to all trans forms with the acceptance by a photon. We all know this very well in ophthalmology, but it is a form of photobiomodulation. Vitamin D synthesis. You convert the pro form of vitamin D to the pre form, and then ultimately becomes activated through the use of light. And rickets is the cause of not having enough sunlight. Tanning. UVB damages the DNA and keratinocytes, which upregulates the pro-opiomelanocortin gene, which then stimulates the whole cascade of releasing melanin to protect your cells. So we have real examples of photobiomodulation within our own living tissues. But in the eye, what about the eye? So macular degeneration and diabetic macular edema are the 2 areas where this has been kind of shown to have interest. There are about 60 publications out there that were reviewed as a grand review. 14 studies were reviewed that seemed to meet the criteria of something that would be worthy of looking at. There's the TOPRA study, TOPRA 1, TOPRA 2, LightSight 1, Electrolyte, Grewal had 42 eyes. So these are small number of eyes. They weren't really kind of like true studies to kind of base anything on, but they're more introductory studies, preliminary studies. We're going to go to the LightSight 2 study. And this was for macular degeneration. It was a safety trial to study the use of photobiomodulation for the treatment of visual impairment in dry AMD. The Alida light system was the system used by lumitherapy. 44 patients were enrolled, 53 eyes, 17 were men, 27 were women, and the mean age was 74 years old. It was dry AMD, non-central GA, with actually good vision at the time. The light treatment was 3 times per week over 3 to 4 weeks. And the photobiomodulated treated eyes showed a significant improvement in visual acuity compared to baseline visual acuity at 9 months. Approximately a 4-letter gain was seen in the treated patients versus a 0.5-letter gain in the sham group. So this stimulated more discussion and more interest in this. Greater than 33% of the patients treated with photobiomodulatory therapy gained 5 letters at 9 months. Conversion from dry to wet was only 3.1% in the treated group versus 5.6 in the sham and 4.8 in the non-study eye. So this generates more interest. And the LightSight 3 study is currently we're waiting for results. It started in September of 19, supposed to complete in June of 22. Of course, they got hit by COVID, like a lot of studies. And so I think there's a delay in the results being sent out, or maybe the results aren't going to be sent out ever. We'll find out. Now, talking about diabetic macular edema, there's Ellis published 10 patients. Shannon group had 42 eyes. The DRCR-net had center involving diabetic macular edema with good visual acuity. And this is the paper that we're going to take a look at as kind of maybe a gold standard of what we should be thinking about and how we evaluate this therapy. So the protocol AE, and this was published with Judy Kim and the DRCR-net group. Dr. Jampel was one of the authors on this paper too. determine the effect on central subfield thickness compared placebo versus treatment. They used a 670 nanometer light emitting source for their phototherapy. It was an eye patch. So they had one patch that was a 670 nanometers. The other one was a white patch that was their control. And they treated for 90 seconds twice a day for 4 months. So a good amount of time, a good amount of exposure. Started in 2019, ended in February 2020. They had pretty good enrollment, 135 participants, 69 of which had treatments, 66 who had placebo, mean age 62 years old, all pretty much Caucasians with majority of men. The compliance was 92% with treatment and 95% with placebo. So photobiometric modulation in lab studies showed significant reduction in retinal microvascular leakage observed in the interplexiform layers, internuclear layer, and outoplexiform layer. That's the theory behind why it should help in diabetic macular edema. In this study, treatment it was given, it was safe and well tolerated, but they had some interesting results. Central thickness, central subfoveal thickness increased in 13 treated eyes versus 15 in placebo. In those patients with center involving diabetic macular edema, 90% were seen in the treated group versus 86 in the placebo group. So there really wasn't much difference between the two. Visual acuity decreased equally between the two groups, 0.2 letters and 0.6 letters in the control. So there was not any statistical difference between the two treatment arms. So they said, while it's safe and well tolerated, it was not found to be effective for the treatment of central involving diabetic macular edema in this treatment trial. So, I'm just a retina guy doing my retina thing in the retina world, but, experts have opinions and I have to defer to the experts. And what if this was true? What if this was a really viable therapy? And I think we have to listen to the experts. So here we go. Ooh. Here are the eyes. Oh, it gets better. Oh, yeah. You didn't test that. Oh, medical doctors. 1 minute. You did 90 seconds. The man is a genius. What can you say? So this is what you can do at home. This is called the Novothor. The Novothor is a real product, and it actually does whole body therapy. Now, I don't know where I can get this, but it certainly does look like a tanning bed to me. Anyway. Thank you. Appreciate your time. I hope that was educational.
Thanks so much, Larry. I gave you this topic because I know you would entertain us. I knew that as well as educate us. That was excellent talk. I think it's very interesting that some of these things are kind of out there, become mainstream, and there is actually some scientific basis for it. Some people are using this for inflammatory diseases and showing that there might be some reduction. So who knows? I just wanted to bring something that's being tossed around and even tried by the Diabetic Retinopathy Clinical Research Network, and as well as, light sight being done in AMD. So we'll see what that shows us in the future. Are there any questions for Dr. Ulanski? We give Anita the microphone, please. Dr. Agarwal has a comment here.
I'm going to go out on a limb and I'll quote Don Gass. I wouldn't think light into retinal disease is going to be of any benefit. So we'll see. I can't believe that stimulating the retina that's already been overstimulated by us causing AMD, longer we live, we get AMD, the less we sleep, we probably get AMD. And with all of that stuff, I can't believe we're going to use light to treat AMD.
Exactly. And you know, that's also my feeling. But again, it's blue light, right? That's more damaging, lower wavelength. And then we're talking of higher wavelengths. So we'll see. I think the jury's out. Thanks so much, Larry, for entertaining us as well as educating us on this very controversial topic.
He's going to share his screen.
So we're going to move on next. But before we get to, hang on one
You're-
quick
you're up,
second
Marty.
here.
You're sharing.
No, not yet. One second. I want to get to here. So we come to the highlight of one of our highlights of this meeting, and that is the Cless speaker and the Cless Award. So Phil, I see you there. I'm waving at you. Is there a way
Hey,
we can
Jenny.
see Phil? Hey, Phil, we can hear you. Awesome. I love
Hey,
this new
Jenny.
technology. Everyone can hear you, Phil, and I think we can also see you as well. So really, thank you so much for doing this, Phil, and for doing this via high tech as well. So our 2023 Cless lecturer is Dr. Phil Rosenfeld. He's a distinguished professor of ophthalmology at the Bascom Palmer Eye Institute, Miller School of Medicine at the University of Miami. As we all have heard already this morning, Phil, not yet, I have some words to say about you. I know you're excited to give us your talk. And
No, I
very
love the picture. It's a great picture.
Oh, okay. It's a younger picture, I might add. Phil's a
Quite a
good
bit.
friend, so I can rib you on that one. Phil is an honored researcher, educator, clinician, and mentor. He has multiple research discoveries that impact our practice today of retinal treatments of diseases. He, for instance, discovered the use of bevacizumab and pioneered it for treatment of AMD. And I do remember, Phil, when we were doing that, you were doing that study where you were injecting initially intravenously, right?
That's correct, yeah.
Bevacizumab, and I was at that meeting at FARVO, and we were getting the, issues with blood pressure, and then you decided, hey, why not? Let's just inject it into the eye. So you went ahead and took that leap, and lo and behold, millions of injections later, and millions of eyes saved with vision in multiple diseases. Phil has also developed the clinical development of OCT instruments, pioneering the use of OCT-guided therapy using VEGF, anti-VEGF drugs. I believe this started with the Pronto study, which you did with Ann Fung many years ago.
Mhm. Right.
We've also developed novel therapies for treatment of non-neovascular dry AMD, particularly with an emphasis on complement inhibitors. And you've also developed swept source OCTA and novel OCT algorithms for the diagnosis and study of AMD and for its use as a clinical anatomic endpoint, particularly again in dry AMD. So how did Phil get to where he is? Well, Phil has a stellar background, pedigree, if you will. He is an MD PhD. He graduated with a BA from Case Western Reserve, summa cum laude, in May of 1979. So I think, Phil, I gave away your age just now.
Yeah.
And then he went to Johns Hopkins Medical School, where he was in the MSTP program and got an MD PhD in 1988. Now, you might not know this, but he actually was an OB-GYN resident, and he did this between 1988 and 1991. And in 1991, Phil wandered up to the Retinal Vascular Center, where he met Carl Chalky and me. And Carl and I were medical retina fellows at the time, working with Stuart Fine, the Bresslers, Bob Murphy, Dan Finkelstein. And Phil came up and said, I really like this retina stuff that you're doing. And we're like, oh, why don't you, come join us, become an ophthalmology fellow? And he's like, well, I'm kind of in the middle of my OB-GYN residency. and we're like, it's never too late. And Phil goes, well, I've already gotten an MD PhD. So I've already taken the long way around, and I've already done this OB-GYN. And both Carl, who's an MD PhD also, and I looked at each other and said, what's the rush? And thank God, Phil, you decided to switch. So Phil then did a research pathology fellowship in retinal degenerations, working at Harvard with none other than Dr. Ted Dryja. And then he became a resident at Mass Eye and Ear at Harvard, followed by a vitreoretinal fellowship at the Bascom Palmer Eye Institute. Since then, he has been at Bascom the entire time of his stellar career. He was an assistant professor until 2002, rapidly rose to associate professor at that point, and then in 2006, again, very rapidly became professor in the Department of Ophthalmology at the University of Miami. Now, Phil is very decorated, if you will, with multiple awards, and these are just a small slice of these from his CV. Of course, he has AAO Achievement and Secretariat Awards. He also received the Paul Ehrlich Award in Basic Science Investigation from the Johns Hopkins School of Medicine in 1987. He was the American Society of Retina Specialists, or ASRS, Senior Honor Awardee in 2005, and he received the Founders Award from the ASRS in 2007. He is also the Richard and Hinda Rosenthal Foundation Awardee and gave the lecture in 2007. This is also better known as the Macular Society Young Investigator Award today. He was given the John Gass Award and Lecture by the Retina Society in 2009, the Arthur and Esther Kane Lectureship at Northwestern in 2012, the Sanford Gifford Memorial Lecturer for the ICEP COS Joint Conference in 2013. He is a Silver Fellow as well as a Gold Fellow for FARVO. He was the 75th Edward Jackson Memorial Awardee and Lecturer in 2018. He's also a top 5 finalist for Bausch & Lomb's Icons of Ophthalmology in 2020, and of course, a top doc and a best doc. And I will highlight that he was a Charles Scapins Awardee and Lecturer from the Retina Research Foundation and the Scapins International Society at the recent Academy of Ophthalmology in 2022. Above all this, Phil is a very valued colleague by many of us and a very valued friend. He is a loving spouse to Julie, as you see here in this beautiful dress that she's wearing, and a great dad to Maya and Shuli, whom I've had the pleasure of meeting all three, as well as, of course, Phil. So Phil, congratulations, and thank you for being the Cless Lecturer in 2023. Dr. Rosenfeld will speak on rediscovering AMD with swept source OCT imaging. Phil?
Wow, Jenny. What a sensational introduction. I'm so overwhelmed. Thank you so much. What I want to do, actually, I wanted to drive the slides from my computer. Can I do that? Or no?
Sure.
Okay,
I'll have
great.
our AV person, AKA Dr. Ulanski, switch you over.
Yeah, I just need the screen sharing. You have no idea what our friendship has meant to me, Jenny. You know, when I was this OBGYN resident, that's a whole other story of how I got started with that. And I enjoyed it, but I fell in love with ophthalmology late as a medical student. And lo and behold, I go talk to Stuart Fine, and I get to spend my elective time in my second year with Stuart Fine. And I go up, and lo and behold, Carl Chalky and you were in the room. I didn't know you before then. And it was the most sensational experience I could ever imagine. And you convinced me right then and there that ophthalmology, in particular retina, I didn't mention to you that Bert Glazer did a postdoc in our lab, and he introduced me to the whole world of VEGF at that time. But retina specifically was going to be my future. And as you mentioned, that led to the Massachusetts Eye and Ear Infirmary, getting to work with Ted Dryja and Elliot Burson. And then at the Massachusetts Eye and Ear Infirmary, getting to learn about photodynamic therapy and VEGF and OCT development with Carmen Puliafito. And one thing led to another. And I was going to be a retinal degeneration disease expert. And then photodynamic therapy got started, thanks to Joe Miller. And no one wanted to do it at Bascom Palmer. I said, heck, I'll do it. And one thing led to another and another and another. And so, Jenny, without your inspiration, I wouldn't be talking to you today. So thank you so much. You've been a great friend and colleague over the years. And it's a great honor to be here today to give the Gerhard Cless lecture. He was a phenomenal benefactor to your department. And it's a pleasure to talk to you today about rediscovering AMD with swept source OCT imaging. And thank you, Amani. Amani Fawzi just gave you a sensational introduction to the whole field of AMD, OCT biomarkers, but I'm going to really narrow it down to using swept source OCT and kind of rediscovering all the anatomic features that we've learned over the years. Now, I do have a number of financial disclosures, and the only relevant one is that I do receive research support, and I'm a consultant for Carl Zeiss Meditech. Before I start, I have to acknowledge Bill Foyer. Many of you who have ever spent time at Bascom Palmer or collaborated with us know Bill. Bill was a beloved biostatistician on our staff for 35 years. He was part of my research group for 20 years. He passed away unexpectedly last summer. And all the work I'm going to talk to you about today really Bill was involved in it, and without his contribution, I don't know if it would have been possible. This is my team, my recent team of phenomenal fellows. And in particular, I want to shout out Giovanni Gregori, who's been my faculty collaborator and colleague for 20 years. Without their extraordinary effort, none of the work I'm going to show you today would have been possible. And then I want to give a particular shout out to Carol Chung. Many of you know her. She's a consultant biostatistician. She took over after Bill Foyer unexpectedly passed away. Ricky Wong, many of you know Ricky. He's professor out at the University of Washington, Seattle, Biomedical Engineering Department. Unbelievable engineer, phenomenal algorithm and hardware development. And without his graduate students and postdocs, once again, none of what I'm going to talk to you about today would have been possible. My other collaborators include Jim Fujimoto and Eric Molt from MIT, Nadia Waheed from Tufts, Robin Geimer from Australia, Xiangyong Yu from Seoul, Xiaodong Sun and Fenghua Wang from Shanghai, and Aaron Lee from the University of Washington, Seattle. And I have to give a shout out to the engineers and scientists at Carl Zeiss Meditech that made the swept source OCT possible, and we work with on a weekly basis. So if you remember nothing else from this talk, this is the most important information. You can extract both structural and angiographic information from OCT angiographic scans, and it provides all the information needed to diagnose and manage patients with age-related macular degeneration. Why is this important? It's important for you clinicians out there, because in 2025, for the first time, we're going to have a code for OCT angiography. But if you perform OCT angiography, you're not going to get reimbursed for standard OCT. What I want you to understand is if you get OCT angiography, the structural information contained within that scan is just as valuable as the structural information that you can obtain from a routine structural OCT scan. Now, this is the instrument that we've been using. It's called the Plex Elite 9000. It's a swept source OCT with a center wavelength of 1050 nanometers and scans at both 100,000 now and 200,000 A scans per second. And there are a number of different scan patterns. There's a 3x3 millimeter, a 6x6, a 9x9, a 12x12, and we can auto montage, a 15x9 with an auto montage feature, and now a 15x15 with an auto montage feature. But in age-related macular degeneration, it's really the 6x6 and the 12x12 raster scans that are so valuable. And Ricky Wong's optical microangiography algorithm really made angiographic imaging possible. And what we really value this method for is the imaging of macular neovascularization using what we call boundary-specific segmentation, which was really developed originally, this boundary-specific segmentation by Zeiss for the Cirrus, and Giovanni Gregori in our group actually first patented that technology. So in 2017, we developed this slab called the ORC slab. It goes from the outer retina to choriocapillaris, and it does a beautiful job of imaging macular neovascularization. And you can see the lesion, but you also had a lot of projection artifacts, which we're able to move using an algorithm developed by Ricky Wong, and we can also measure the area of the lesion as well. So we first used this to image type 1 macular neovascularization. Now you could see these lesions using the ORC slab, the outer retina to choriocapillaris slab. You can see the lesion quite nicely, but it's really the boundary-specific segmentation with en face imaging that Giovanni developed, which allows us to image these lesions so beautifully that reside between the Brooks membrane and the retinal pigment epithelium. So you can also use this to measure every component of the neovascular complex. So we can measure the total lesion using the ORC slab, total macular lesion. But as you know, type 1 lesions, which are below the RP and Brooks membrane, can be visualized, and they're part of the type 2 lesion that resides above the RPE. So using boundary-specific segmentation, you can see what's below the RPE, what's above the RPE, and you can also visualize the total lesion. Now, polypoidal choroidal vasculopathy has been a type of neovascularization that's really relied on color, fluorescein, and really ICG angiography to identify the polyps and the branching vascular network. But swept source OCT is great. You have the structural B scan. You can see the polyp. And as Amani mentioned, this double layer sign representative of a branching vascular network or type 1 lesions. And the ORX lab shows it quite nicely. You can see the entire lesion, the polyp, the branching vascular network, or you can do the boundary specific segmentation. And you can see the polyp and the branching vascular network. You see all the polyps, and these polyps are actually tangled vessels. They're not aneurysms, they're tangled vessels. And this work came out of a collaboration with the group in Shanghai, Fenghua Wang and Jiaodong Sun, in which they showed using swept source OCT that these polyps are tangled vessels. Now, what's the best imaging strategy to visualize PCV? Well, in collaboration with Seung-Young Yu in Seoul, Korea, I think it's pretty definitive that swept source OCT really is as good as ICG angiography for the detection of PCV. And moreover, in a recent paper that was just published in Ophthalmology, again with the Shanghai group as collaborators, we've been able to show that changes in the polyp actually predicts near-term onset of exudation using swept source OCT angiography. Not only that, swept source OCT angiography can show the evolution of polyps, and this comes from the work of Mengxi Shen from our group in collaboration with Shanghai. And this is an case of a polyp here underlying a large pigment epithelial detachment. And after 26 anti-VEGF injections, look, this polyp actually progresses to a branching vascular network or type 1 neovascularization. And this work came out in AJO case reports in 2021. How about type 3 macular neovascularization? It has all the classic signs on structural B scans. You see the cystic maculopathy, the PED, the kissing sign between the RPE and the outer plexiform layer. But on the ORX lab, you can see the entire lesion here, and you can get rid of the projection artifacts. You can see the lesion isolated within the retina. And now, as Amani mentioned, one of my favorite topics, which are these treatment-naive, non-exudative macular neovascular complexes, which we first identified when we started imaging patients with swept source OCT. Here on spectral domain OCT, you see what you think is just this drusenoid low-lying pigment epithelial detachment. But this is actually what Amani mentioned as a double layer sign, just as in the branching vascular network in PCV. So here's an ICG of this lesion, and now we blow out up to a 3x3 region from this ICG, and here's the OCT angiographic image. We were blown away by the detail on how much better it appeared compared to the ICG angiogram. And now we followed this case. This is one of the first cases we ever followed in our clinical trial. We followed this patient for 73 months, and the lesion grew, but there was never any exudation over 6 years. Here was another case we discovered around the same time. We thought these were drusen here. They're not. The ones on the side are drusen. This is a double layer sign. Notice the difference in the internal reflectivity, as Amani mentioned. The ones on the side are drusen. And this lesion was quite obviously a neovascular complex. And it grew over 23 months. And lo and behold, some fluid developed, exudation developed. The patient was symptomatic. So we treated this patient. And one of the take-home messages from this study, as well as the current work with pegcetacoplan, is if you catch these lesions early when exudation developed, they do really well. 7 anti-VEGF injections, 20/20 vision. This patient did exceptionally well. And then we followed this patient. And over 4.5 years later, the lesion clearly is growing. And the PED is collapsing to a low-lying double layer sign, but no exudation. So if you catch them early and you treat them aggressively, you can watch them and you can see with swept source OCT angiography, the lesion growing. So as Amani mentioned, we've performed natural history studies on these lesions. So we have 1-year data on 160 patients, 2-year data on 227 patients. And lo and behold, 14% of patients with intermediate AMD or GA had these non-exudative lesions. So it's not uncommon. And if you look at the natural history, this Kaplan-Meier plot, if you have one of these lesions, by 12 months, a 21% risk of exudation versus a 3.6% risk. And by 24 months, 34.5% risk versus 7.3% risk. So that represents a 14-fold increased risk of exudation if one of these lesions is present. And it doesn't matter if it's intermediate AMD or geographic atrophy. So what's the best imaging strategy to identify these lesions? You guessed it, OCT and geography. But we did a study way back in 2016 showing that swept source OCT and geography is as good, and I think better because it doesn't require the infusion of a dye, than ICG and geography. So as Amani mentioned, with the recent FDA approval of pegcetacoplan, detection of these non-exudative lesions will be a very, very important. And as we published in the paper from a couple of years ago, in eyes with double layer signs at baseline, there's a significant risk of exudation that's statistically significant if one of these double layer signs is present. And this work came out in September 2021. And this is all from the phase 2 trial. And you can imagine the same type of study is underway from the phase 3 trial with pegcetacoplan. So now, before treating any of these GA patients with pegcetacoplan, my recommendation is you perform OCTA imaging. And if those patients contain these non-exudative neovascular lesions, I plan to follow them monthly with OCT when they come in for injection. I'm not going to do a full exam. One of my strategies since COVID has started, and we published on this, I do a lot of OCT only visits with injection. So I'm planning to do the same thing here because I know these eyes are going to be at increased risk of exudation. And I want to emphasize pegcetacoplan probably doesn't increase the risk of neovascularization, but the risk of existing neovascularization to develop exudation. So it's that trigger. So we can talk about that after the talk, but there's something about complement inhibition that triggers non-exudative lesions to become exudative lesions. So swept source OCT isn't just for wet AMD. We use it all the time for dry AMD. And I think it's even more valuable because we can follow the disease progression, as Amani mentioned, from dry to wet. So let's talk about drusen. So years ago, Giovanni Gregori in our group developed this algorithm for the serous OCT using the segmentation strategies that we use here. So we do dense raster scans using the swept source OCT, and we can do boundary specific segmentation of the RPE and Brooks, and we can then view the segmentation in an en-face strategy, get a volume and an area map. You can see how beautifully it superimposes on top of what you see on a color fundus image. And this work has been validated, and it came out in 2021 in TVST. So we use this for following disease progression in our ongoing clinical practice and in what we call the impact swagger studies that I've been doing in collaboration with Nadia Waheed, because we're really interested in understanding what precedes the formation of geographic atrophy. Now, en-face imaging of atrophy is something that we started way back in 2009, again, with boundary-specific segmentation. So this is a dense OCT scan, and we're looking at what we call the sub-RPE slab. We're looking at this hypertransmission defect that develops as atrophy develops. And this is a 64 to 400 micron slab below Brooks membrane. And mathematically, we just remove what's above and what's below, and we look at this region in an en-face projection. And anywhere that it's bright, you've lost the RPE, or it's greatly attenuated, so you get these hypertransmission defects. We don't do autofluorescence. We don't do routine color. All we do is follow our patients with this sub-RPE en-face slab to follow the development of atrophy. So we asked a very simple question a few years ago. What's the smallest hypertransmission defect that persists and progresses to form typical geographic atrophy? This is a variation of the IRORA-CRORA nomenclature. And we just look at the hypertransmission. So here we have a case which was thought to have intermediate AMD. And there are these bright spots here. And you can see on individual B scans through these regions that wherever there's a bright spot, you can see this hypertransmission defect developing. We think this is the earliest manifestation of disease progression. And we proved in a natural history study in collaboration with Robin Geimer that once they hit a greatest linear dimension, not a horizontal, not a vertical, not a diagonal, but any greatest linear dimension of 250 microns, they persist. They don't vary. They don't regress. They persist. So do persistent hypertransmission defects develop into typical GA? Yes, they do. So once we see a persistent hypertransmission defect, we think of it now as analogous to geographic atrophy. And can it be graded? So we performed this grading exercise. And I want to thank Jeremy Liu. He came up with this idea that why not just do a simple grading where here's one of these hypertransmission defects and you have a circle with the greatest internal diameter of 250 microns. And by grading, you just put the circle in the lesion. And if the lesion is at least as big or bigger than the circle, then it's a persistent hypertransmission defect. And you can move the B scan to the region, and you can see the outer retinal changes and the hypertransmission defect. So we put together this grading group from Baskin Palmer and Federico Corvi, Rick Ferris, Tak Han Lim from the CAM group, Singapore, Vasada, and Nadia Wahid. We performed this grading exercise. And long story short, 97% agreement, positive predictive value of 99.2%. This work came out last year in Ophthalmology Retina. So can these persistent hypertransmission defects serve as a clinical trial endpoint? So we all want to develop therapies to study disease progression, but we got to come up with endpoints in clinical trials. So look at this drusen volume map here. This is a sub-RPE en face image, no hypertransmission defects here. And then 18 months later, notice that the drusen volume has increased, but lo and behold, you get these hypertransmission defects already appearing before the drusen go away. And that's a take-home message for everybody. The drusen don't have to go away before you form regions that are already destined to become atrophy. And then 24 months from baseline, you start losing some atrophy, some drusen, but you've already developed more areas of these hypertransmission defects, and they go away further, and more hypertransmission defects form, and they coalesce and they grow. This is how atrophy forms. Little foci of hypertransmission defects then coalesce into larger areas. So look at this video that Jeremy put together. This is amazing. You're going to see this a couple times because this is how atrophy forms. So this is a sub-RPE slab, and you can see these regions, and this is pigmentation. I'll get to that in a second, but regions with hypertransmission defects and pigmentation grow, and they coalesce. And now I'm going to show you, after 5 years, what these regions look like relative to the drusen that are created from the exact same slab. So here's the drusen map. Here we're starting to see, oh, look, there they are, but the drusen are still there. Oh, there's some, now the drusen start going away, but it's already forming. The hypertransmission defects form long before the drusen go away. And as Amani mentioned, it's about drusen volume predicting this progression and these areas of hyperpigmentation, really, really important. So you get these areas of hypertransmission defect, and every place you see a B scan, you can see this hypertransmission signal into the choroid. Now I want to introduce you to a new concept thanks to Ricky Wong called optical attenuation coefficient. Now, a little bit of optical principles here. So the optical attenuation coefficient, what's called mu-T, is a property of biotissue. It has to do with the light absorption, the light scattering, the light back scattering, and how it varies depending on different dimensions of the tissue. So this optical attenuation coefficient is a characteristic of the biotissue. And in OCT, remember, light hits the biotissue, the retina and the choroid, and it gets attenuated, and then it gets reflected back. So it's this doubly attenuated light that gets back to the detector, this backscattered light. And the property of the tissue depends on how much light gets back to the detector. And if you look at a typical OCT signal, this is signal strength versus depth. In the instrument, the original signal has this exponential decay. But what in the commercial instruments, there's this logarithmic transformation that linearizes it. And that helps the signal in the shallower depths, but there's still a lower signal deeper. But mu-t, I just want to bring out this mu-t factor is a component of the slope. It's a constant. Well, it doesn't have to be a constant. One of the interesting things about optical attenuation coefficients is that it can be calculated for every pixel along the depth of the scan. And not surprisingly, the RPE has the greatest optical attenuation coefficient. And Vermeer in De Boer's group deserves the credit in 2014, coming up with the idea of let's replace the signal intensity in the OCT, typical OCT, with the optical attenuation coefficient. And this really accentuates the RPE because in an OAC BP scan, the RPE jumps out. And as you start to lose the RPE signal, it disappears. So it's a really nice strategy for highlighting the RPE and allowing even better segmentation that we can combine with other segmentation strategies. So what we've done with Ricky's group and Aaron Lee with artificial intelligence is we've combined the sub-RPE slab with what's called the OAC sum map. And now we have the OAC elevation map, which shows the elevation of the RPE layer above Brooks membrane. So it identifies drusen. And now we have an algorithm that automatically calculates the OAC composite map. So this is the typical drusen map I showed you previously, and the slight variation in color intensity, but look at the volumes here. This is the OAC elevation map, which is equivalent to the drusen map. And this is combining this single algorithm that gives us this composite map and the OAC elevation map. And now we can see where we have automatic algorithm that can see where the hypertransmission defect is appearing, and we can get the area of these lesions, and we can get the area of the individual lesions as they grow and coalesce. Why is that important? Because now we have an algorithm to follow disease progression. And we can now use persistent hypertransmission defects as a clinical trial endpoint. And for it to be used to study intermediate AMD, and right now we don't have any clinical trials that allow us to use an endpoint in intermediate AMD. So to use it as an endpoint, these hypertransmission defects have to be absent in intermediate AMD. It has to be a precursor to typical GA. They have to persist, which we know they do by definition. They have to be easily and reproducibly detected and measured. And the growth rate has to be easily measured like we do with GA. But we had to know, is the growth rate reproducible? So we did this study where we had two groups of patients, 171 eyes with intermediate AMD, 68 developed at least one hypertransmission defect over time, and another 59 eyes with really, really small lesions. These lesions would never get into any clinical trials. And we measured their growth rates, and both groups grew at a growth rate of 0.22 millimeters per year. So now we have a new clinical trial design, which you're going to be hearing about. We start with eyes that have a certain drusen volume, which you can see here, just like I showed previously, no evidence of these hypertransmission defects, central drusen volume. Here we're starting a little bit larger, not 0.03, but 0.2 millimeters cubed. You randomize 100 eyes into each group, and this ensures the adequate number of hypertransmission defects after 1 year. After 1 year, if you start with 100, you should get about 72 hypertransmission defects in your sham group. And the possible treatment effects, imagine the amplification effect if you can decrease the number of hypertransmission defects and decrease the growth of these individual hypertransmission defects. So your endpoint is at 2 years, and the overall growth of the hypertransmission defects is what your endpoint is. But imagine if a treatment can prevent the hypertransmission defects from forming as well as growing. And this clinical trial design has been accepted by Wiley Chambers at the FDA. And just yesterday, we got word that the AJO has accepted our paper. So as Amani mentioned, there are other OCT biomarkers that are very, very exciting. One is the hyperpigmentation or the hyperreflective foci, calcified drusen. All these biomarkers are going to increase the likelihood of disease progression, but you always have to be careful that you don't increase the number of biomarkers to the point that you reach the point of no return. Choriocapillaris flow deficits, basal laminar deposits, photoreceptor thickness, reticular pseudodrusen, and choroidal thickness and choroidal vascularity are going to be important too. And I know I'm probably running late, and Jenny, I want you to yell if I'm already too long, because I don't have the timer in front of me. But let's talk about hyperpigmentation. Amani mentioned this. These are these black spots. These are RPE cells clumped together, maybe with some inflammatory cells. And as Amani mentioned, once you get these hyperpigmentation areas, they generate these hypotransmission defects, and they appear black on the sub-RPE slab. Well, this is great because it gives us a way now to quantitate these hypertransmission defects. And we published this in Ophthalmology Science last year, how these sub-RP slabs are really useful. But guess what? Ricky Wong's come up with an optical attenuation mapping procedure that allows you to look at the RPE as well as these hyperpigmentation areas in the retina, as well as hyperpigmentation along the RPE. This is a totally automated algorithm. It works for both swept source OCTA and spectral domain OCTA. And this is a summary of some of his figures showing an eye with and without drusen. And the red represents the hyperpigmentation along the RPE, and the blue represents the hyperreflective foci in the retina. And this work came out in Biomedical Optics Express last August. How about calcified drusen? As Amani mentioned, Fassato was the first to mention these drusen with weird internal reflectivity and these hyperreflective cores. And Rick Spade published a paper showing these things, which he called refractile drusen, but they're calcified drusen being associated with areas of hypoautofluorescence or atrophy. And then Christine Curcio demonstrated beautifully that these refractile drusen or calcified drusen are actually calcium phosphate spherules or nodules. And she showed once again, these weird drusen with hyperreflective cores correspond to these refractile calcified drusen and hypoautofluorescence. So we performed the natural history study, as Amani mentioned. We followed drusen over time as they developed these calcified drusen. And once again, the sub-RPE slab, you can see weird internal reflectivity, but also these hypotransmission defects. And using the same on-phos strategy, where you see these hyporeflective hypotransmission regions corresponding to calcified drusen. So this is a great way to quantify calcified drusen. And not surprisingly, Ricky Wong just published the paper coming up with an automated algorithm to quantify calcified drusen. But this is just showing how a typical drusen causes some attenuation of light in the sub-RPE slab. But these calcified nodules, these spherules, cause a great deal of reflectivity, causing a black spot. And what's really interesting is we performed this natural history study looking at the progression. And as atrophy forms along the margins, you form these donut lesions on the sub-RPE slab. And as more and more of the calcified spherules go away, you develop this target lesion, which is very important for following disease progression. And we describe in this Ophthalmology Science paper from last year. So are these focal hypertransmission defects really calcified drusen? There's a paper that just came out in March 2023. Now, this is a collaboration I started with Jerry Luddy, who has unfortunately passed away, but he was a good friend. He and I were graduate students together in the BCMB program at Hopkins. And I imaged two of his brothers who were living in South Florida back in 2016. And Malia Edwards, who took over his lab after he passed away last year, continued the work. And what we did was we looked at these two brothers. This is the same eye, one showing with and without B scans. And in this brother with a large area of atrophy, this is the sub-RPE slab, you've got these dark areas where these are calcified drusen. And you can see them here, the drusen with the hyporeflective cores. And lo and behold, she actually performed alizarin red S stain of these lesions. And sure enough, they are calcium spherules, as you can see here. And then she pre-treated some of the slices with EDTA and showed that they went away. So thank you, Malia. Jerry Luddy's work lives on, and they are calcified drusen. And now let's talk about choriocapillaris flow deficits. Again, this is an algorithm we developed with Ricky Wong's group. This is looking at a natural history progression of choriocapillaris flow deficits in the central macula. This is using a 6x6, a 3x3 scan. And just as you age, you lose the white dots are flow deficits, areas where you don't have flow in your choriocapillaris. As you age, you lose choriocapillaris flow, predominantly in the center macula. Isn't that fascinating? Oh yes, and the age-related macular degeneration affects the center macula. So could there be something going on here? As eyes get older, you lose flow in the center, and then those patients with genetic risk of progression are losing their choriocapillaris flow in the center. Double whammy, right? So we published this work on the normative database in 2019 in AJJO, and somebody asked, does it correlate with disease progression? And I'm happy to answer questions if there's any time, which I doubt. But this is geographic atrophy imaged using the 6x6 lab. And here we've outlined the area of atrophy, okay? And what we've done here is we've whited out the area of atrophy because we're not interested in the area that's already lost RPE and choriocapillaris. We're looking at the region around the atrophy. And lo and behold, when you look at the choriocapillaris, there's this dark halo around the atrophy. Sure enough, there's more choriocapillaris loss around the atrophy. And we were able to quantify it. We do this by looking at 1 degree rims or 300 micron rims. And you can see in that 600 microns around the atrophy, the choriocapillaris flow deficits are the highest. So does it correlate with growth? And summarize a lot of work. We looked at all the choriocapillaris flow deficits in these rims and in the outside region, and it's the entire area of the macula that correlates with the growth of geographic atrophy. And the choriocapillaris flow deficits can predict about 36% of the GA growth. We published this in AJO in April in 2021. So what else contributes? What other biomarkers contribute to GA growth? And this is an example of a hypotransmission defect. And these are the en face images, which I've shown you previously. Here's a mass showing the atrophy. And lo and behold, as Amani mentioned, there are these things that also cause double layer signs called basal laminar deposits. And using the OAC algorithm, we can measure the distance between Brooks membrane and the OAC max, which is the RPE. And sure enough, all around the atrophy, you have these basal laminar deposits that have accumulated. And the Bond group, Frank Holz's group, showed that in fast-growing lesions, there's this splitting between the RP and Brooks. But once again, Christine Curcio, who has contributed so much histopathologically in AMD, demonstrated that this splitting, when it's not neovascularization, this splitting is caused by basal laminar deposits. So here's a slowly growing lesion over time. The dotted line is baseline. The solid line is where it grows after 1 year. We're looking at those rims around the slow growing lesion, and they look pretty benign, but lo and behold, an average growing lesion has these basal laminar deposits that dominate in this region, and then a fast growing lesion has basal laminar deposits which have accumulated all throughout the region. And sure enough, once you factor in the basal laminar deposits and choriocapillaris, you can account for 56% of GA growth. What's responsible for the rest? How about photoreceptor loss? So this is another one of these examples. You can see the basal laminar deposit here. Here's the normal outer nuclear layer, but notice the outer nuclear layer is thin here. Well, Ricky Wong developed an algorithm that measures the outer plexiform layer to RPE thickness. And sure enough, you got photoreceptor thinning all around the lesions that grow quickly. And you can see it in the average growing lesion here and in the fast growing lesion to the right. So when you factor in choriocapillaris flow deficits and basal laminar deposits and photoreceptor thinning, you can account for 62% of the GA growth. And this work came out in December issue of AJO. So what predicts the remaining 38%? Well, you can round up the usual suspects. We expect reticular pseudodrusen are important. These are these deposits under the retina. And once again, using swept source OCT, you can look at a slab just above the RPE, so you can pick up these subretinal drusenoid deposits. And I think you actually see them better than you do on autofluorescence. These are the dot images. Here's another case, reticular pseudodrusen. You can see them here, and there's a more the ribbon configuration. You can see it here. So you can see on a single slab, you can see an area of atrophy based on your sub-RPE slab or an area of reticular pseudodrusen. And we actually did a mass grading of 307 eyes comparing OCT imaging with color, autofluorescence, and infrared imaging. And sure enough, it was as good, and I think better, because you only need one imaging modality. And finally, I'm going to end with choroidal thickness. Once again, swept source is a great way of looking at the choroid, use optical attenuation correction now. And basically what you do now is you fix the total optical attenuation coefficient so it's stable throughout the entire depth, and then you adjust the intensity of the signal. So now you get this beautiful, intense choroid. See, if in a normal OCT scan, sometimes the posterior segment isn't visualized, but Ricky Wong came up with this optical attenuation correction, which is routinely used in ultrasonography. And now we have this beautiful algorithm that measures the outer boundary of the choroid. And as you age, your choroid gets thinner. Your choroid's getting thinner, you're losing choriocapillaris. So clearly the choroid, I think, plays an important role in disease progression. And we had a natural history study of normal eyes through the 20s through the 80s. And what you can see here is the choroid is thickest in the center macula superiorly, thinnest under the optic nerve. We weren't the first to show this. This has been well established. This is the 95% upper limit for all eyes and the lower limit. A lot of people would call this pachychoroid. I don't know what pachychoroid is because normal eyes can have a very thick choroid. So I don't even use the term pachy anymore. And these are eyes in their 20s, average, 95% upper limit, 95% lower limit, 30s, 40s, 50s, 60s. You can see it just gets thinner and thinner with age, right? And you can also use this optical attenuation correction to look at the choroidal vessels. You can't see flow in the choroid because the RPE scatters the light, that backscattered light. So what you can use the optical attenuation for, you can use it to actually look at the vessels themselves. So Ricky came up with this algorithm that allows you to actually look at the choroidal vessels. This isn't an ICG image. This is a swept source OCT image. And you can come up with a variant of CVI where you're looking at the volume of the vessels and the volume of the choroid, not the area. And now look at the mean choroidal thickness and the choroidal vessel volume. They go hand in hand. So when you look at the choroidal thickness, what you're measuring is the choroidal vessel volume. But what's so fascinating about the choroidal vascularity index is that it remains constant despite what the thickness is. I think of it as a universal constant. So if it changes, you know there's something pathologic going on. So this is just looking at the average over all the decades. You can see whether it's the mean, the 95% upper limit or lower limit, the maps look identical. So CVI is a great way of following disease, particularly age-related macular degeneration. So optical attenuation correction allows you to measure choroidal thickness and vascularity. It allows you to follow the choroidal vascular volume. The choroidal thickness and volume decrease with age, and the choroidal vascularity index, I propose, is going to be a really valuable metric for following patients with disease, particularly AMD. This is where it came out in the April issue of Ophthalmology Retina. So here are the important take-home messages. Swept source OCT angiography provides both structural and angiographic information. You can use it for both dry and wet AMD. You can identify and quantitate every step in the progression of AMD from drusen to macular atrophy. Dense raster OCT scans are going to be really powerful no matter what instrument you use, because you can perform en face imaging. And we're all comfortable with en face imaging because we use it now for OCTA imaging. And optical attenuation coefficient OCT imaging enhances the power of OCT imaging. And I propose that moving forward, I think we're going to get more and more in the mindset that OCT imaging can provide a standalone strategy for studying the onset and progression of our patients with AMD. Thank you for your attention. I'm sorry if I went over.
Thank you so much, Phil. That was outstanding as always. I learned a lot from you, as I always learn, and I'm sure there are a lot of questions. One of my questions for you, Phil, is with that OAC, is that going to become standard, do you think? Can we extract that from regular non-swept source to guide our treatment and inform us as to maybe which eyes are better suited or less suited to some of the evolving therapies?
So what I often say about swept source OCT, it's provided us a tool to identify OCT biomarkers. And now we're going back to spectral domain and we're applying the same algorithms, the same OCT biomarkers to spectral domain. So I'm delighted to say that we developed these algorithms for swept source, and now we can plug in the OCT angiographic scan patterns, which are really better scan patterns because they're denser scan patterns. And every OCTA machine repeats the B scans because that's the principle of looking for flow. You look at repeated B scans in the same location, and then you look at changes, and that gives you the flow signature. But the repeated B scans also give you the ability to get better structural images too. So everything that we've developed with OCTA, swept source OCTA, yes, we could now retrofit, go back, and use the same algorithms on spectral domain OCT angiographic scan. The only one thing that swept source gives you that spectral domain doesn't is deeper penetration into the choroid.
Fantastic. I mean, you've given us yet another tool again to help us in the clinic and to help us guide our treatments. Are there questions? I think Anita Agarwal has a question.
I have a practical question for Phil. Is the company still making the Plex Elite? When I spoke to them last year, the Academy, they said they were going to kind of take it off the market. Is that true?
No, they were never going to take it off the market. The question was, are they going to continue to produce new instruments or go to the next generation of swept source instruments? So I'm pleased to tell you, they're going to continue to make new instruments and support the existing instruments.
So they don't make any more of the Plex Elite, but they'll make a
No,
new model?
No, they're going to make more of the Plex Elite, but they're always looking to make the next generation of instrument. You know, they want to make it faster. They want to make it, denser. And so all that is underway, but they are making additional Plex Elites.
Okay.
Phil, do you foresee in the future that we'll have AI and we'll be able to extract everything from an OCTA?
Well, I'm biased, and I think you've gotten a taste of what artificial intelligence can do from Ursula Schmidt-Erfurth and Pierre Skeen and the work from Vasada's group and Justin Ehlers. I mean, AI is the future. But I'll go on the record for saying I haven't seen anything that AI has shown me that I didn't already identify using my own eyes and following the natural disease progression. So I don't see AI as teaching us something new. I see AI as a tool for automating the things that we've identified in our clinical practices. So yes, I think what's going to happen in the very near future is based on the algorithms that have been developed, based on our observation and other people's observations. You're going to get an algorithm, AI, that's going to give you the risk profile of any patient with AMD that comes in. I want to reiterate that all the AI algorithms that looked at the risk of wet AMD progression, none of those AI algorithms identified the double layer sign as a risk. And we all know now that that double layer sign, that non-exudative neovascular lesion, is the biggest risk factor for near-term exudation. So I believe in clinical observation. Us clinical researchers are still in business. I think of what we do is feeding the field of artificial intelligence.
Agreed. I think it'll help us screen and maybe have with the throughput in the future. Hopefully we won't have an AI in a box and just have us in another little room injecting as we need to. Because I think
there's always going to be
There's
always going to be new and interesting observations
Right.
for us to make.
Right. Other questions for Dr. Rosenfeld? Dr. Skandra, Bill Dimitra has a question for you. I wish you were here so we can give you a hug. That was a great talk. And then wine and dine
thank you.
you as well.
Thank you for a great talk. Have you looked at any association between genetic risk and biomarkers on OCT?
One of my favorite topics. So years ago, when we did this study called the Complete Study, this was the first complement inhibition study. We did it with the Alexion drug, eculizumab. I had the magic with systemic Avastin and intravitreal Avastin. I thought lightning was going to strike twice. And we did a systemic study with eculizumab, which is the C5 inhibitor from Alexion that was approved for paroxysmal nocturnal hemoglobinuria. And we did the study, but it didn't work. As Bill Foyer said, the study was a success because the natural history was validated, but we didn't change disease progression. But in that study, we showed that in intermediate AMD, the drusen volume, the drusen burden, was directly related to the number of complement factor H alleles someone carried, 0, 1, or 2. And that's now been confirmed in 2 additional studies. So I think that there's going to be a really close association between genetics and OCT biomarkers. And I give a shout out to Nadia Waheed and the Impact Swagger study that we've been doing for over four years. We have genetics on all these patients. So exactly, our goal is to correlate the genetics with these OCT biomarkers that I've showed you today.
Any other questions? Phil, that was fantastic. Thank you for teaching us once again, identifying yet more biomarkers to inform our treatment. Again, I wish you were here. Great big virtual hug from all of us to you. Say hi to Julie.
Thank you, Jenny. Greatly appreciate it. I can't wait to hear your talk.
Thanks. All right, so we'll move on now, and I will be giving my talk entitled New Treatment Options for Geographic Atrophy and Neovascular AMD. These are my financial disclosures, several of which are relevant for these talks. We've heard from Dr. Rosenfeld about the biomarkers for geographic atrophy. You've also heard from Amani, and I'm going to speak today as to new treatments for geographic atrophy as well as for neovascular AMD. Geographic atrophy affects 1 million people in the United States today and 5 million worldwide. So just like everything else, it increases in prevalence as we age. So unfortunately, it's 0.7% at age 70, but increases to 11.29% by age 90. So fortunately, there are some new treatments for geographic atrophy. In the past, when I was sitting where the residents are sitting and the fellows, there was no treatment except for the AREDS formulations. Now we have anti-complement drugs, thanks to the work of Phil and many in this room. In terms of the anti-complement approvals, we have pegcetacoplan, known as APL2 or Syfovre, which was FDA approved in February of this year. We also have avacincaptad pegol, known as Zimura, for which an FDA approval has been submitted, and probably we'll be seeing that in the near future. I'll also touch upon some integrin regulating therapy, and of course we know there's stem cell therapy, but there's limited time, so I'll concentrate primarily on anti-complement drugs today. So why anti-complement drugs? Well, we know that in our lives, oxidative stress combined with genetics and combined with the environment can lead to chronic inflammation. And this chronic low-grade inflammation is mediated by complement and results in complement deposition at the level of the RPE, Bruch's membrane, and choroid. So it makes sense that perhaps if we inhibit complement, we can retard the development of complications from AMD, such as geographic atrophy. Pegcetacoplan is a pegylated pentadecapeptide, and it's a treatment for complement-mediated diseases, such as paroxysmal nocturnal hemoglobinuria, or PNH, for which it was approved in 2021 and is known as Empaveli. This year, in February 17th, we got approval for geographic atrophy. So how does pegcetacoplan work? Pegcetacoplan inhibits the complement pathway by binding to C3 and C3b, and thus inhibits the downstream effects of the complement pathway and the MAC attack complements, and obviously cell lysis in the long term. The Derby and the Oaks are the phase 3 studies that studied the effect of pegcetacoplan as compared to sham. So in these 2 studies, which were parallel randomized sham-controlled studies, pegcetacoplan 15 milligrams in 0.1 ml monthly was compared to pegcetacoplan at the same dose, but dosed every other monthly, and compared to sham given either monthly or every other monthly. The primary endpoint was at 12 months and was the change in total geographic area lesion size based on fundus autofluorescence. And there were multiple other secondary outcomes that were also studied out through 12 and then 24 months. And there's actually a 3-year extension study known as GALE that will inform us of long-term outcomes. So patients who were 60 years of age or older and had visual acuity of 23/20 or better were eligible for inclusion. The lesion size had to be between 1 and 7 disc areas. And in this study, it's important to note that the geographic atrophy lesion could be subfoveal or could not be involving the foveal center itself. There is also the requirement of perilesional hyperautofluorescence around the lesion seen on fundus autofluorescence. Patients who had choroidal neovascularization in the fellow eye were not excluded, unlike the other study that we'll be talking about later. At baseline, the patients were well balanced with regards to the pegcetacoplan arms and the sham arms. So what were the results? Let's look closely here. And we see that in these two parallel studies, Oaks and Derby, that we have the rate of geographic atrophy lesion size plotted across time. In the Oaks study, the results at 1 year were significant for a decreased growth rate for the pegcetacoplan arms as compared to the sham arm. And we see reductions of 16% for every other monthly and 21% reduction in the growth rate is monthly treatment as compared to sham. Now, if we look in the Derby, however, we don't see that same reduction, and this was not yet significant at 12 months. Let's just watch and see what happens at 18 months. At 18 months in Oaks, we continue to see a clinically significant difference between the pegcetacoplan arms, shown in blue and orange, compared to the sham arms, 16 and 22% reductions. By now in Derby, we do see a reduction in the growth rates of 12 and 13%. And these increase out to 24 months, such that there were significant differences at 24 months between the pegcetacoplan arms and sham. So why was there a difference? Well, actually in the Derby study, there was an imbalance of more subfoveal lesions, which grow at a slower rate. And so because of this, the difference in the reduction of the growth rate could not yet be seen at that early time point. So let's just plot out and combine both the Derby and Oaks. And if we do that, we do see that there were clinically significant differences in terms of the reduction of the GA lesion growth rate at 6 months, 6 to 12, 12 to 8, and 18 to 24 months. Now, if we look closely and we say, let's look at the non-foveal and let's look at the subfoveal. Again, we see that the drug was effective for non-subfoveal GA, slowing in growth, as well as subfoveal lesion growth slowing, but it took a little bit longer for the subfoveal difference to occur. What about visual acuity? So we're all wondering, does this affect the visual acuity? Well, actually, because there were non-subfoveal lesions involved, and then of course with the subfoveal where the vision was already decreased, there was no difference seen in the visual acuities between the sham and the treatment arms. Also, in terms of secondary endpoints, there were also no significant differences seen in terms of reading speed and even functional reading speed indices and microperimetry. Now, although there were no differences in the actual microperimetry results for the center, there was a post hoc analysis done of the junctional zone around the area of geographic atrophy shown in yellow. And this area is a lesion area of 250 microns around the border of the actual GA lesion. So from the border out to 250 microns. And this was a special programming that was done in Oaks that looked at microperimetry. Now, these endpoints were assessed in this yellow region itself. And it was found that indeed there appears to be a difference within this junctional zone such that we may be affecting the outcome of the areas that are not yet affected by the geographic atrophy, and only time will tell what that will show. So looking here, you see that the number of scotomatous points are fewer for the treated eyes compared to the sham eyes, and the dB in terms of sensitivity was maintained in the treated eyes compared to the sham eyes by 24 months. So it appears that you need to give this drug for some time to begin to see this effect in that region. What about the safety of pegcetacoplan? Well, in terms of the endophthalmitis rates, it was 0.034%, so no signal there. In terms of inflammation, 0.24% per injection. And importantly, knowing what we saw with brolucizumab, there were no ocular occlusive vasculitis events, and there were no cases of retinitis. Now, as Phil already alluded to in his talk, we do get choroidal neovascularization in terms of eyes with geographic atrophy or just eyes with AMD at baseline that we might not know are there. So that could be macular neovascularization. And then eventually they can become exudative macular degeneration when fluid develops. So what happened in Derby and Oaks? If we look over time, we see that the rate of choroidal neovascularization as diagnosed by the primary investigator was 3% in the sham group. In the ever other monthly group, it was 6.7%. And in the monthly group, it was 11.9%. So there is a signal being seen in this group in which choroidal neovascularization is developing. If we want to try to understand this better, we can divide out in the study eyes that had a fellow eye with choroidal neovascularization and eyes without choroidal neovascularization. And what this tells us is that the eyes that do not have a fellow eye with CNV overall will have a lower rate of progressing on to choroidal neovascularization. We know this from the MPS, we know this from multiple other clinical trials that have been done. And typically too, when we look at the eyes given pegcetacoplan, the rates, although higher, are still not as high as for the eyes in which we include choroidal neovascularization in the fellow eye. So something to think about when you're using this drug and trying to decide what to do. Now, I'd like to show you here an example of how these lesions grew over time. On the left is an example of how a patient who was treated with pegcetacoplan had loss of photoreceptor shown in green or RPE loss shown in the reddish orange color. On the right is the fellow eye of this patient, which was not treated. And you can see that the growth rate here was much faster and the lesion ended up being much larger. That is, the RPE loss grew by 50% in the treated eye with pegcetacoplan versus 225% in the fellow eye by 1 year. So let's turn now to the other drug. Let's look at avacincaptad pegol, which is a pegylated RNA aptamer. Now, this drug, unlike pegcetacoplan, which is an anti-C3, this is an anti-C5 drug. And so it works further down in the complement cascade, attacking the MAC attack complex and lysis at that area. Obviously, the inflammasome and the MAC attack complex result also in cell death, so inhibiting C5 will inhibit the complement pathway. Now, avacincaptad pegol was studied in the two GATHER studies, GATHER1 and GATHER2. Unlike the other drug, pegcetacoplan, in this study, avacincaptad was only studied in eyes in which the foveal center itself was not involved. So these were non-center involving geographic atrophy. In addition, the lesion had to be within 1,500 microns of the foveal center itself. In this study, evidence of choroidal neovascularization in either eye at baseline was excluded. And this is again important to keep in mind when we look at the rates of choroidal neovascularization. So let's look at it now, and these are some examples of what these eyes had to look like in baseline. So here's the 1,500 micron border from the fovea. As long as the lesion was somewhere within this region, but not involving the foveal center, it was included. If it was beyond completely this 1,500 micron circle, as in here, that would be an excluded eye. In GATHER1, the patients were randomized to receive avacincaptad pegol, 1 milligram versus 2 milligrams versus sham in part 1, and then later on 2 milligrams versus 4 milligrams versus sham in part 2 in a 1:2:2 randomization scheme. The primary endpoint, again, was the mean change in geographic area from baseline to month 12. So we see at baseline that these eyes were pretty well balanced with regards to demographics, geographic atrophy lesion size, and baseline visual acuity. So what were the results? Now, if we look at GATHER1, we see that at 1 year, the results were met. Primary endpoint showed a reduction of 27 to 28% in the geographic atrophy growth rate between the 2 dose arms. This is the 2 milligram dose, and here's the 4 milligram dose as compared to sham. And this benefit across the group increased over time. And you can see the separation here of the curves increasing out to 18 months and then later to 24 months. Now, the problem, again, with this complement 5 inhibition is that choroidal neovascularization also developed in this study. In the fellow eye of these patients, it developed in 3.5%. In the sham arm, it was 2.7%, so about the same. So that's a good control there. Now, in the avacincaptad pegol arm, 1 milligram, it was 4%. In the 2 milligram dose, it was 9%. And in the 4 milligram cohort, it was 9.6%. Now, in GATHER1, it was thought that choroidal neovascularization would hinder the measurement of the geographic atrophy by fundus autofluorescence. So these eyes were discontinued. So we can't tell you more regarding continued treatment in these eyes. But in GATHER2, these eyes were kept in the study. And there was a re-randomization at the end of 1 year in Gather 2 for the Avastin Kepted P-gall eyes to monthly versus every other monthly treatment. And we see in Gather 2 that again, we see the same effect with Avastin Kepted P-gall showing a reduction in the geographic atrophy growth rate as compared to sham, with the purple compared to the gold lines here, similar to what we saw in Gather 1, both clinically significant. In terms of complications in Gather 2 and compared to Gather 1, there were no cases of endophthalmitis, no significant inflammation, and no cases of optic neuropathy. However, we did see, again, cases of choroidal neovascularization. This was 6.7% in the treated arm versus 4% in the sham arm. So again, an increase in the rates of choroidal neovascularization. And these were investigator determined, and then subsequently the reading center also looked at it to look at the rates of non-exudative macular neovascularization, which Phil had talked about earlier also. There was an exploratory analysis done for this drug, and it was looking at what is the visual acuity outcome with Avastin Kepted P-gall versus sham over 12 months. And overall, you can see here for the Avastin Kepted P-gall arms that the risk probability for cumulative visual loss is lower than in the sham group. And in fact, the reduction is felt to be a 56% reduction in the rates of significant visual loss. So that's the story for the complement, and I'm sure you will have a robust discussion if time allows as to which patients to put into the study, informed by the decisions and the biomarkers that Dr. Rosenfeld has discussed with us today, as well as Dr. Fawzi. Let's turn now to one other drug that's an integrin regulating drug, and this is known as rezuteginib or ALG-1001. This is very early on, but this drug attacks mitochondrial dysfunction in intermediate AMD. Recall that eculizumab and other drugs have not been shown to be of benefit previously, but this one appears to show some signaling. It reduces oxidative stress and perhaps can improve cellular function and survival. In patients who are treated with rezuteginib that had intermediate AMD, there were actually improvements of visual acuity of 8 or more letters, 10 letters, and 15 letters in 48%, 32%, and 20% of patients respectively, as compared to 7%, 7%, and 0% in the sham arms. And interestingly, these improvements in best-corrected visual acuity were strongly correlated with improvements in microperimetry and color function. So perhaps this is a drug that's rescuing the mitochondria and that can help in development, prevention of progression of intermediate AMD. Let's turn now to the second half of my talk, which is neovascular AMD. Fortunately, in 2023, we have multiple drugs available. We have bevacizumab, ranibizumab, aflibercept, and faricimab, as well as brolucizumab that we can use to treat neovascular AMD. There are, however, limitations in the current treatment regimens, and we see overall an average gain of anywhere from average 9 to 11 letter gains in the mean change in visual acuity from baseline over 1 year to 2 years as compared to no treatment in which patients lose vision. So this is a far improvement compared to what we had back when I was a fellow or even practicing as an attending. However, if you look on this graph on the right, we see that the real world doesn't really show us these results. In the real world studies outside the US with Aura and Lumiere, as well as within the United States, looking at the IRIS database and the Vestrum databases, we see pretty much that the visual acuity results are not as good. And why is that? Well, it's because there's under treatment. Patients aren't getting the right number of treatments that they need. And we know that the number of treatments is correlated with visual acuity outcome. So can we improve this? Well, we can, perhaps, if we have drugs that require fewer treatments, or perhaps obviate the need for a return to clinic. So I'd like to highlight 2 recently approved treatments, which include the bispecific anti-VEGF and anti-Ang2 drug faricimab, which perhaps can give added durability, and then also the port delivery system, which is currently in recall, but can give a sustained drug delivery over time and obviate the need for multiple visits. I'll also touch upon some experimental treatments, including anti-VEGF-C, anti-VEGF-D drugs, gene therapy, and a tyrosine kinase inhibitor. So first, let's look at a different mechanism of action. Let's look at the angiopoietin system and see how does this affect choroidal neovascularization in AMD. Well, in normal homeostasis, we know that angiopoietin 1 is involved. It's the key player in the angiopoietin type 2 pathway in maintaining healthy vessels. Angiopoietin 1 activates the type 2 receptor, which is expressed by multiple cells types and present in high levels in healthy vessels. And this is what keeps us healthy. This regulates the survival signaling, vessel maturation, and maintains our blood vessels. Unfortunately, under stress conditions and pathologic conditions such as AMD, there's a switch and the level of Ang2 increases. Ang2 is a competitive inhibitor of Ang1, and it will displace the Ang1 from the type 2 receptor and bind to it. Once Ang2 binds to the type 2 receptor, there is a new cascade that occurs, and this inhibits survival signaling and leads to vascular instability. In addition, Ang2 makes the VEGF2 receptor more susceptible to the VEGF-A binding and results in the angiogenic vascular permeability and promotes angiogenesis. And in case you were wondering, Ang2 levels have been measured in disease states, in not only AMD, but retinal vascular occlusions, diabetic retinopathy, and proliferative diabetic retinopathy. And you can see that the mean levels of the Ang2 are increased in these states. And there's no significant difference really in the Ang1 levels. So Ang2 does play a role and is upregulated in these pathways. So it makes sense to consider a drug that inhibits the anti-VEGF-A as well as the Ang2 pathway, and this is known as faricimab. It's the first bispecific antibody that blocks both. It was studied in the phase 3 Tenai and Lucerne studies, and patients with AMD, previously untreated, were randomized to receive faricimab 6 milligrams at 4 loading doses, followed by a personalized treatment interval based on disease activity assessments at weeks 20 and 24. So they were either given drug Q8, Q12, or Q16 based on disease assessments. These patients were then compared to the aflibercept treated eyes that were given 3 loading doses and then were treated Q8 weeks as per label through the end of year 1. And then in year 2, the faricimab treated eyes, all of them regarding Q8, Q12, Q16, were put into a personalized treatment regimen based upon visual acuity assessments, OCT, and the presence or absence of macular hemorrhage, as shown in the lower algorithm here. And these, again, were compared to aflibercept dose Q8 through study end. So what happened in Tenai and Lucerne? If we look here, we see that the average mean visual acuity with the faricimab dosing arms were similar to the aflibercept dosed arm at Q8. That is, regardless of the treatment interval, there was no significant difference in visual acuity, Q8, Q12, Q16 weeks. In terms of the OCT, we saw a reduction in the OCT thickness for the faricimab treated arms at any of the intervals as compared to the aflibercept treated arms. In terms of the number of injections in the PTI phase, however, to get the same result required 3 injections of faricimab overall in this group as compared to 6 given the aflibercept or 6 in the if the drugs were given Q8. But again, in the PTI arm, we saw fewer drugs required. So how long could we give this drug? Like, what was the interval? Well, in the study at week 48, essentially 1 year, we saw that 45% could achieve this result when dosed at Q16 weeks, and 33% could be dosed at Q12. So together, 78%, almost 79% could be dosed at Q12 weeks or longer and still achieve the same visual acuity as with dosing with the aflibercept Q8 and the same reductions in the central subfield thickness. And by the end of 2 years, there was a shift with more eyes being able to be dosed at Q16 week intervals, such that it maintained 78% being able to be dosed at Q12 weeks or longer. In terms of safety, there were no new safety signals seen with equivalent signals between faricimab and aflibercept in the studies. So that is one treatment option we have, perhaps, of increasing the interval for dosing for our patients. Now, what about a sustained drug delivery? Is there such a device approved? The answer is yes. There's the port delivery system known as the PDS, which is a surgically implanted device through the pars plana shown here, and it's refillable in the clinic. And it gives a continuous customized distribution of ranibizumab at 100 milligrams per ml to maintain a therapeutic drug concentration in the eye. Unfortunately, though, this is currently recalled due to a problem with the septum. It was studied in the phase 3 Archway study, in which patients were given this PDS device with the special formulation of ranibizumab, 100 milligrams per ml, and dosed Q6 months and supplemented as needed, and compared to monthly ranibizumab every 4 weeks. Now, in Archway, patients had to have been diagnosed within 9 months. So these were pretty fresh lesions, and they had to have required at least 3 treatments or more within 6 months. So these had to be high frequent needs patients that were enrolled into the study. So what was seen in Archway? Well, interestingly, in this non-inferiority study, the PDS indeed was non-inferior and showed equivalent efficacy to monthly ranibizumab. You can say, well, at 4 weeks, there's a slight drop here in the vision, and that's related to the surgery. But once that comes back up, by 8 weeks, we see essentially similar visual acuity results with no significant difference between monthly ranibizumab and the PDS device. In terms of the OCT, again, we see equivalence with good control of the amount of fluid present. In terms of the number of treatments, 98% required no supplemental ranibizumab injections before the treatment refill at 6 months. So it did meet the durability result. Adverse events, however, with the PDS, of course, would include endophthalmitis. And most of these endophthalmitis cases were felt to be related to conjunctival dehiscence. So perhaps, the conjunctival dehiscence leading to bacteria entering the eye more frequently than would otherwise be with an intravitreal injection is a real concern. And so because of this 1.6% risk of endophthalmitis being much higher than with intravitreal injections, we really have to be very careful when we use this post-approval in terms of how we make the wound. So there's really an emphasis on limiting conjunctival erosions and surgical technique. Unfortunately, there is another adverse event that became obvious in the fall of last year, and that was a septum dislodgement. And here's an image published in JAMA Ophthalmology where you can see what a normal PDS device looks like. There's a flange here with an overmold, and then there's this septum that sits there attached to the overmold. With refills, this septum can be dislodged. Now, it doesn't result in any leak or doesn't have to be removed, but because of this, it has been recalled, and there is some work being done to try to fix this problem. Patients have been surveyed who have received this PDS implant, and this was a sub-study done of patients in the Archway study. And they were asked at week 40, if they were in the PDS, did you prefer the PDS or did you prefer the intravitreal injections that you were previously given? And 93% of the patients in the PDS arm said that they preferred the PDS device compared to 1.3% who preferred the intravitreal injections. And then there were some that didn't have any strong opinion. That's why they don't add up to 100 if you're looking at the numbers. And of these 93%, 73.5% had a very strong preference for the PDS. And they also, even those with ocular serious adverse events, including endophthalmitis, 75% still preferred the PDS. And the reason for that primarily was this: fewer number of treatments, less discomfort, and then required less time for treatment. So it's the time-saving component there and the durability of the drug. So time remains to be seen what will happen as the PDS is reworked to prevent the septum dislodgement. So in the last few minutes, I'd just like to highlight a few other experimental treatments that are being studied, which include the VEGF-C and VEGF-D inhibitor, OPT-302. This is an IgG1 soluble fusion protein that is an IgG-like domains 1 to 3 of the VEGF receptor 3. And it's used in combination with anti-VEGF-A therapies as a sequential injection. And in the phase 2 study, it was shown that this drug at the higher dose resulted in superior visual acuity gains compared to ranibizumab alone. It's currently being studied in combination with ranibizumab in the SHORE phase 3 and in combination with aflibercept in the COAST phase 3 study. So remains to be seen how this will perform out in the future. I'd like now to turn to gene therapy. ADVM is no longer being studied because of problems that we're seeing with the diabetic related drug, but the RGX drug 314 is still in clinical trials. And what this does is it uses a novel AAV8 vector, which contains gene encoding for the anti-VEGF Fab. So if you will, it's almost like creating an anti-VEGF-A factory inside the cell. And this results in continuous production of this anti-Fab into the eye. And this was studied in a phase 1/2 study. And it was shown that in patients who had high needs, all of these dots represent anti-VEGF injections. After the RGX 314 is given, you can see that the number of injections markedly is decreased. So this makes sense, right? If this drug actually works and you can create VEGF factories in the eye and produce your own anti-VEGF, you won't need to inject them over time. So this is going forward in the phase 3 atmosphere and ascent studies to date. And then lastly, there's also tyrosine kinase inhibition with a drug known as vorolanib. Vorolanib is a bioerodible drug release system in a drug release system like a Duracert system. And this is known as EYP 1901. And you can see here this Duracert containing the vorolanib tyrosine kinase inhibitor. And in the phase 1 Davio study, it was looked at primarily for safety because again, it's a phase 1 study. And they enrolled all patients who had previously been treated for neovascular AMD and had a minimum, again, of 3 anti-VEGF treatments within the previous 6 months. And they were given 1 injection of aflibercept followed by EYP 1901. And what they found was that there were no significant safety endpoints, no drug-related systemic endpoints, and the majority of ocular non-serious AEs were mild and expected. What is interesting to me is if we look at the secondary endpoints, again, with the caveat that this is a phase 1, and we see that the median time to supplemental anti-VEGF is 6 months. And in fact, 35% of these eyes remain supplement-free up to 12 months. And there is a 73% reduction in the treatment burden at 12 months. So I think, the future will tell us whether this drug shown in the phase 1, again, showing this marked reduction in the number of treatments, whether this will bear out in the phase 2 and if it goes forward to the phase 3 studies. So in summary, in 2023, we're very fortunate that we have new drugs that are effective for the treatment of geographic atrophy in terms of the anti-complement drug. We have multiple emerging drugs. There are also stem cell and other replacement therapies, which I didn't have time to go into today. With regards to neovascular AMD, we've seen that new mechanisms of action, including anti-Ang2 combined with anti-VEGF and perhaps anti-VEGF-C and anti-VEGF-D in combination and tyrosine kinase inhibitors can also impact the treatment of neovascular AMD in a good way. We've also seen that sustained delivery devices are out there and that gene therapy may also prove to be effective in lowering the treatment burden. So hopefully in the future, we'll see these new treatments that will help us decrease the treatment burden for our patients and improve the visual acuity outcomes. I think the future is bright for AMD, and I thank you for your attention. Are there any questions? Amani, can you give Amani that mic, please?
Jenny, so for the anti-complements, how do you think we should be using them and which patients should we enrolling and starting? Do you think about the extrafoveal or subfoveal? I mean, just putting it all together, what are you going to do?
It's a very difficult question. I've been toying with this myself. We do have a drug for geographic atrophy. So I think because of that risk of choroidal neovascularization, I think that's where we all hesitate, right? If we didn't have that, we wouldn't worry about it. Of course, it's treatable, but when I propose that to my patients, they're like, well, I really don't want to have to get another problem, and I don't want to cause that either. So if it's their only eye, I would say no at this point until I give a better feel. Granted, it's low, it's like 3 to 5%, but that's still higher perhaps than they would have. So I think for a patient, you have to look at it very carefully. If their other eye has choroidal neovascularization, I would be less likely to use it because they're already at higher risk for developing CNV. Okay, then I would look at the biomarkers. Phil and you have shown us very nicely what some of the biomarkers are for choroidal neovascularization occur. So if they have a very high drusen volume, if they have the double layer sign already, if they have hyperreflective foci, and there are these changes that are seen in addition to the GA, then those are also eyes that are high risk for developing CNV as well. So those are eyes that perhaps I would shy away from also. So then if we have the perfect patient that doesn't have that and just has very early geographic atrophy in each eye and is well informed and understands the risk, then I would say that would be a patient that I would be willing to consider this treatment in them and then follow them closely. And I would also do what Phil says is I would get an OCTA and see, is there macular neovascularization that's not yet exudative?
So one of my conceptual problems with this is when we treat our wet AMD patients, they get better, the OCT gets better, they have an incentive to come back because if they miss an appointment, their vision drops. How are we going to sort of get buy-in from those patients to get monthly injections for an undetermined amount of time we can't really measure the improvement and they can't feel the improvement.
That's a great point, Amani. And so your point is there's no functional outcome that they're perceiving. And that is extremely important as well, because you don't want to do any harm. So what I would do is this is where imaging comes in. And this is where you show them what the study results show. You take the baseline fundus autofluorescence, you do it over time, and then you show them if they have two eyes, right? Because that's my ideal patient, that changes between this eye and the other eye. And then you can also do microperimetry. You could do that junctional zone. We do a lot with microperimetry and show the functional deficit. And then also with the other drug, it was shown that the proportion that eventually loses vision, depends how close it is to the fovea. There's also a difference. So I guess another point I would do is that if the patient has the GA that's close to the center, right, but not yet there, that would be a patient that I think would be at higher risk for losing vision as well. But it's a very difficult question. And I think we're going to learn over time how to have this conversation with our patients and how to pick which patient is best. Anita, Dr. Agarwal?
I think Amani told me Phil wants to say something.
Oh, sorry, Phil, I didn't see can we unmute Dr. Rosenfeld?
I'm unmuted.
Okay.
I just hear his opinion.
Yeah.
I was just curious.
He put his picture up.
Yeah, I've been having this discussion with my patients, and for the average retina specialist, this will be a time-consuming process. Be prepared to have a dialogue with your patient. And I've decided I'm only going to treat patients who are symptomatic. And it really may not be relative to where the atrophy is. These patients are symptomatic long before the atrophy gets close to the fovea. And I have one of these, I've got good news and bad news discussions with the patient. The good news is we have a treatment for the first time that can slow disease progression. The bad news is it's not going to improve your vision. So I can make the argument, the sooner you treat, the better. And it's monthly injections, with the understanding that not everyone can get an injection every month. And the every other month arm was pretty good. But I'm going to recommend starting monthly. And I'm really not that worried about the risk of exudation because I know these eyes respond really, really well to anti-VEGF. And they may not need an anti-VEGF injection every time they get a complement inhibitor. So good news, bad news. What I like to say is something is infinitely better than nothing, okay? And we have something. And to me, this is another photodynamic therapy Macugen moment, okay? Neither of those treatments were very good, but it's the first step when you don't have anything to slow disease progression. We're going to get better treatments. We're going to get better ways to deliver the drug. So I embrace the technology, and I have family members. Just during this meeting, I got a couple text messages asking, when is the drug going to be available? At the University of Miami, we have the formulary committee for the university's meeting today. okay, to decide whether to accept it on formulary, which they will, but this is the process. We need to get Medicare coverage. We have to get the drug in our hands. But I have patients lining up for it.
I think that's great, Phil. You know, I've had that conversation with many patients. And again, you're right. It's the one who's extremely informed and who knows what can happen when the GA progresses that are more motivated to get it. And I think, you know, I like what you said about it's another Macular kind of moment or PDT. You know, when we were fellows, we only had for some treatments, like even for DME, focal laser. And we sold it to the patients, right? We're like, you know, you're not going to get better. We're going to prevent you from getting worse. And same with subfoveal CNV. And in those eyes, we dropped vision. Remember that? That was,
I have a question here.
Yes, Anita.
The question is,
And
if you
then
have a
Alice.
patient with a tiny GA in the center and everything else is fine, would you recommend this for that person? Vision say 20/60 in that eye, the other eye is 20/20, and they have a small GA in the center.
I mean, you know, according to the studies, it will slow it down, right? But it's going to take with the approved drug, it's going to take with pegcetacoplan. That's kind of like in the Derby arm, right? Because it's already subfoveal, but it does work. It does slow it down, but you won't see it till 18 months, 24 months. So let's say your patient has a long life expectancy and they're 60, I would treat them. If they're 84 and they have multiple other comorbidities, and you know, I wouldn't want to bring them in every single month because their lifespan might already be limited. So I think it's going to have to be personalized.
So the
Phil?
question is,
You know, I think I just wanted to add fellow eye. Always got
Fellow
to
eye, right.
worry with if the fellow eye is good, that patient's not going to want injections.
right,
It's just a little foveal lesion.
correct. So the question is so in about 18 months, you could see improvement, but that's improvement. I mean, that's not improvement. That's just reduction in the rate of whatever the circumference of the lesion. So once they already are 20/60 and they're going to progress to maybe 20/80 in that 18 months, and then after that, you're going to see a result. So that's really not something to sell to the patient, right? I mean,
Well,
it's
but
not like it's already it's already, I mean,
But
they would
if
have
this is
gone
the
to-
better seeing eye, if this is the better seeing eye, this is the only possibility of salvaging vision that you can offer the patient.
And don't forget also dark adoptometry, right? There may be some in that junctional zone, a difference there that we're going to slow that down. That may affect that for a particular patient.
I think it's going to be for I guess your generation that sold submacular surgery and submacular laser.
All right, Phil, we're being labeled here.
Oh boy.
foveal laser for subfoveal CNV. I think that generation is better at selling things than we are.
But we're not that far apart in age, Amani, you and I. Okay, Alice, go ahead.
Speaking of that generation, so the reality is that this study is showing us 24 months.
Sorry, can you hear her?
I'm sorry.
Okay, louder.
This study is showing us 24-month data, and the gap is widening, and it's important, I think, to remember. You know, when we started doing these injections every month, everybody said, "Oh, nobody would do this. Nobody would come in," and the reality is that we know if you stick to the regimen that keeps the neovascularization quiet, that their vision is retained over 15 years.
Absolutely.
And the slow growth, growth of that lesion reduces their vision. That's why they lose vision. So aggressive treatment, I think, or appropriate treatment, I think, is frequent. And with geographic atrophy, it's the similar finding. Now, I suspect any factor that is one factor that affects the rate of growth of geographic atrophy is probably going to be associated with neovascularization. It may be that all of them are, because you're supporting whatever the substrate is that is reducing the risk of loss of vessels. So I think that's going to be a part of just about any component of, at least for the first many years, of anything that slows the progression of geographic atrophy.
I think it's a really important point that Dr. Lyon brings up, that the curves are still separating. And so Gail, the 3-year extension, is going to tell us what that difference is, and probably going forward too. Dr. Mirza.
Can I just say, can I give a shout out to Alice? another wonderful fellow that I experienced as a resident. Thank you for helping me become the person I am today, Alice. And I look forward to sharing patients with you. And I want to emphasize, it's not just the growth of the lesions, it's the appearance of new lesions. So I think of it like compounded interest. The sooner you start, you're going to accumulate more functional retina that you can preserve in the future. So send me your patients, Alice. I'll be waiting for them.
So I would echo a lot of what is said. Is I call that retina real estate, keeping as much as we can. But I would point out patients often ask me, what's worse, wet macular degeneration or dry macular degeneration? And we used to say all the time, it's wet, right? Because it's really aggressive. I've switched to saying, well, that depends on what you think is worse, right? Until now, we have had no treatment for GA, right? So I do think that this is something that patients are going to want. And I do think that it's going to require much longer conversations, which we tend not to have time for, but we need to make it.
Exactly. I mean, this is one of those where you have a pro con. I'm sure there'll be one at the upcoming sub day, and we'll hear more. So thanks so much, everybody. We're going to take a 15-minute break and then come right back. I'll be giving the next talk, Do Treatment Outcomes Differ Based on Initial Drug Selection for Diabetic Macular Edema? These are my financial disclosures, some of which are relevant for today's talk. In 2023, we're increasingly seeing insurers that require step therapy mandates. And this is because of the cost savings by using drugs such as bevacizumab first. I think in the back of all of our minds, our hesitancy for using step therapy is that we want to be able to choose what we believe is the best drug for our patients. And then we're also worried about the potential worsening in visual outcome during the time to demonstrate suboptimal responses to drugs before switching to another anti-VEGF drug. We do have some guidelines based on the Diabetic Retinopathy Clinical Research Network Protocol T and Protocol AC, which can help guide our initial drug selection. And then you might also wonder, well, how do the newer drugs fit in, such as faricimab and the biosimilars? And I'll attempt to discuss that today. In the US, based on these 2018 Medicare costs, we can see that the cost of bevacizumab is almost 26 times cheaper than aflibercept, and definitely cheaper also than ranibizumab. And this is the genesis for why bevacizumab is being mandated as initial drug therapy in these step treatments. So first, let's look at Protocol T. What did Protocol T teach us? Well, it taught us that the drug selection can be based on baseline visual acuity. So this was a randomized study of 3 drugs, bevacizumab, aflibercept, and ranibizumab. And in this study, as you can see by the graph, all 3 drugs resulted in better visual acuity outcomes at the end of 1 year than at baseline. So all 3 drugs improved visual acuity. However, Protocol T also taught us that the visual outcome was linked to the baseline visual acuity with regards to the drug. That is, if the baseline visual acuity was good, 2032 to 2040, all 3 drugs did well, and the improvements were anywhere from 7.5 letters to 8.3, and these were not clinically significantly different. However, if we then look at the eyes that had 20/50 or worse vision, we see here on the dashed lines that the visual acuity results were, in the solid lines, that the visual acuity results differed based on which drug was used first. So for aflibercept, we see a gain of 18.9 letters at 1 year versus 11.8% for bevacizumab in orange, and then 14.6 letters for ranibizumab shown in the light blue. And these were statistically significant differences. Now, if we look at the results at year 2, and we look at the differences between aflibercept and ranibizumab, we see that this disappeared. There was no significant difference in the mean visual acuity outcome at the end of 2 years. However, we also see that the difference was still significant with regards to bevacizumab. Furthermore, if we look at the OCT or the visual acuity, we see that both aflibercept and ranibizumab did better than bevacizumab when the vision was 20/50 or worse at baseline. Looking at the OCT outcomes, again, we see that based on the initial visual acuity, the OCT outcomes again differed and were better for aflibercept or for ranibizumab, shown in the blue and the orange lines for gains in vision, compared to ranibizumab, shown in the light blue. So to summarize, if the visual acuity for your patient is 20/50 or worse, and you're choosing between these three drugs, you can choose aflibercept initially, and you'll have better vision through 2 years compared with bevacizumab. Or you could choose to use ranibizumab, knowing that if you chose aflibercept or ranibizumab, you're going to have the same visual acuity result at the end of year 2. If, however, you have to use bevacizumab, know that it still results in 68% that have a visual acuity of 20/40 or better at 2 years if their initial vision was 20/50 or worse. So you're still going to get some improvement in vision. So the question now comes in, if you have an eye that's 20/50 or worse, and you start with bevacizumab, and then you find it's not working well, and you switch to aflibercept, is there going to be any visual acuity difference? Are you going to harm your patient? Well, Protocol AC and the DRCR network looked specifically at this. And they looked at patients who are over the age of 18 or older, type 1 or type 2, that had center-involved diabetic macular edema with visual acuity of 20/50 to 23/20. And they could not have had any anti-VEGF treatment within 12 months or any other DME treatment within 4 months. These eyes were randomized to receive aflibercept monotherapy or to receive bevacizumab first. And again, the purpose was to compare the efficacy and outcomes of aflibercept with bevacizumab or aflibercept alone as needed for treatment of center-involved diabetic macular edema. So at baseline, patients were given whatever drug they were assigned to, either the bevacizumab or the aflibercept. 6 monthly injections were given using the usual DRCR algorithm. And then after 6 months, depending upon how they were doing, they could be extended or not. Now, at 12 weeks, however, if they were in the bevacizumab first group and they weren't meeting the criteria for improvement, they could be switched to aflibercept, and then they were maintained on aflibercept after that. So what was the criteria to switch? So for the bevacizumab first eyes, if they had persistent center-involved diabetic macular edema with persistent thickening of the central subfield on the OCT, and they had been given 2 prior bevacizumabs consecutively, and the vision had not improved by 5 or more letters, and the CST had not improved by 10% or more, and the visual acuity was still 20/50 or worse within the first 24 weeks, they were eligible to switch. If it was past the 24 weeks and their vision had improved, the CST had not improved, and they were still thick, then, and they were 20/32 or worse, then they could be switched also to aflibercept. So at baseline, we can see that the eyes were similar with regards to the demographics. Hemoglobin A1C was 8 in each group, and the visual acuity scores were 20/63 median for each group. These were thick eyes, 478 for aflibercept, 496 for bevacizumab, and for the most part, these were phakic eyes. Prior treatments, again, no difference with regards to having had prior anti-VEGF in the distant past, prior focal laser grid, or even prior PRP. So very similar groups. And in terms of follow-up, the follow-up was good in terms of completing the year 2 visit, 90% for the aflibercept, 86% for the bevacizumab. So there were 158 in the aflibercept group and 154 in the bevacizumab group. We see here that the total number of treatments at the end of 2 years was 14.6 injections for aflibercept and 16.1 for bevacizumab. Now, this is interesting. Let's look now only at the bevacizumab eyes, and let's see when they switched and what percent. Okay, so remember, the first time they're eligible to switch is at week 12 or month 3. And we can see at this point, 11% met the criteria for switching from bevacizumab to aflibercept. So they hadn't improved by 5 or more letters. The OCT had not improved by 10%, and the visual acuity was still at 20/50 or worse. By 24 weeks, 39% of the bevacizumab cohort had been switched. And at 1 year, 60% of those originally started bevacizumab were now on aflibercept. At the end of 2 years, a total of 70% of the original cohort were no longer on bevacizumab, but were now on aflibercept. Now, using this algorithm, we see early on at week 12, week 24, and even week 52, that their eyes that were in the aflibercept monotherapy group were still doing better than the bevacizumab group. Makes sense, right? Because these include the eyes that are not responding well to bevacizumab. By the end of year 2, we see that there was no significant difference in visual acuity using this algorithm. In fact, at year 2, we see that equivalent percentages of patients had been 20/20 or better, 20/40 or better, or 20/200 or worse. So we didn't give them any harm at the end of 2 years by using this algorithm. In terms of the change in visual acuity at 2 years, again, similar percentages of eyes improved 15 or more letters, 10 or more letters, or worsened 10 or more letters, or 15 or more letters when treated with the aflibercept or when treated with bevacizumab. Looking at the OCT, as expected, the aflibercept treated eyes did better initially than the bevacizumab treated eyes, right? Because these include the eyes that were not responding in the bevacizumab group that had to be switched over to aflibercept. However, at the end of 2 years, when this algorithm was used, there was no difference in the OCT outcomes in terms of thickness. In terms of complete resolution of the macular edema, this was 60% for the aflibercept monotherapy eyes and 55% for the bevacizumab switch to a aflibercept treated group. Looking at retina-related ocular events, we see no significant signal in terms of complications or requiring PRP, vitrectomy, vitreous hemorrhage, development of TRD, or neovascularization of the iris, or neovascular glaucoma. In terms of ocular adverse events, again, no safety signal seen between the eyes given bevacizumab first or the eyes treated with the aflibercept monotherapy. If we look at the systemic APTC events, including non-fatal MI, non-fatal stroke, or death due to potential vascular or other unknown cause, again, no safety signal was seen. Looking at ocular adverse systemic events, there was a slight imbalance in the percent that required hospitalization in the aflibercept monotherapy at 48% compared to bevacizumab at 32%. And the DRCR group looks at this and says, well, you know, if you compare to the totality of the data, this is probably just a statistical OMA at 0.05 or 0.04. So Protocol AC helps us conclude that if you use this algorithm and you are switching after 3 bevacizumab injections, if you're not seeing a response, you will not get a significant difference in terms of your visual acuity outcome or your OCT outcome over 2 years, comparing aflibercept monotherapy versus bevacizumab first. Bevacizumab first with the switch to aflibercept at 12 weeks or later, therefore, is a safe and effective regimen in these eyes with visual acuity of 20/50 or worse. With the caveat that if you start with this, realize that about 70% of this cohort of bevacizumab first did require a switch to aflibercept, and that the vast majority of these were switched between 3 weeks and 6 months, or 3 months and 6 months, that is 12 weeks and 24 weeks. So that helps guide you in what to expect if you have to use bevacizumab first. Now, what about the newer drugs? What about faricimab? Well, faricimab was not studied in Protocol AC, nor was it studied in Protocol T. So let's look specifically now at this newer approved drug, faricimab, which is the bispecific antibody that I mentioned in my earlier talk, inhibiting Ang2 as well as VEGF-A. In the Yosemite and Rhine studies, which were the phase 3 randomized double masked studies, patients were given one of 2 doses of faricimab, one of 2 regimens of faricimab, I should say, as compared to aflibercept 2 milligrams every 8 weeks on label. So the faricimab arms were faricimab 6 milligrams, 6 loading doses, and then continued out Q8 weeks for the duration of the study. The other arm, which is a more interesting arm to me, is the faricimab personalized treatment interval, in which they were given 4 loading doses, and then based on the response, the patients were treated or not treated as needed based on visual acuity, OCT findings. The primary endpoint was the average visual acuity, averaged between weeks 48, 52, and 56, and patients were followed out to 2 years. So what did we see? Well, if we look at the visual acuity gains here, we see that there was no difference between the faricimab dose Q8 weeks or the faricimab dose and a personalized treatment interval with intervals Q8, Q12, or up to Q16 weeks as compared to aflibercept dosed every 8 weeks. If we look at the change in OCT, we see that the faricimab dosed eyes Q8 weeks, shown in this darker blue, versus the faricimab personalized treatment interval Q8 up to Q16 weeks, had better reductions in the thickness of the retina with decreased mean central subfield thickness as compared to the gray line shown here of aflibercept Q8 weeks. So we got the same visual acuity effect, but a slightly better drying effect. We can look more closely here and see when did these eyes achieve absence, the first time they had no diabetic macular edema present. And let's compare the faricimab dosed arms, Q8 and PTI with aflibercept dosed every 8 weeks. And we can see that the time when 75% cumulative incidence of absence of diabetic macular edema occurred earlier for the faricimab treated eyes than with the aflibercept treated eyes. And you can see that point occurring here at about week 20. This occurs much sooner. Now, also, when we look at the dosing interval, we can see that 62% of these patients at the end of 2 years were able to be dosed every 16 weeks with faricimab, and 78% could be dosed every 12 or more weeks with faricimab treatment. Now, if we look at the mean number of injections, what this translates into is 3 injections for faricimab in year 2 in the PTI arm, as compared to 5 injections if you dose the drug Q8 weeks or if you give aflibercept Q8 weeks. In terms of the safety signal, there was no significant safety signal seen between the faricimab arms and the aflibercept arms. So this helps inform us that if you're trying to pick a drug and you're trying to get the same visual acuity effect, but you're trying to decrease the number of treatments, perhaps you could consider choosing faricimab, again, depending upon if the insurance will allow you. Okay. Now, the question may pop in, well, what about biosimilars? We have reference products, which are the approved FDA branded drugs, if you will, ranibizumab, aflibercept, right? And bevacizumab, not FDA approved, but the branded name is bevacizumab. Now, the biosimilar drugs to these have these other 4 letters here after the name ranibizumab. So it's ranibizumab Nuna, ranibizumab EQRN. These are how biosimilars are named. So sometimes it's similar just to refer to them by their trade name. So ranibizumab Nuna, the biosimilar to ranibizumab, is known as Byuvis, and ranibizumab EQRN is known as Cimerli. Both of these biosimilars are FDA approved. Byuvis in September of 2021 and Cimerli in October of 2022. There are other biosimilars in development, which include SB15 and MYL1701, and these are biosimilars to these aflibercept. There was an INSIGHT phase 3 study that will hopefully lead to approval of MYL1701 for diabetic macular edema. I will point out that these, the ranibizumabs here are approved, these biosimilars for treatment of AMD and DME, as well as choroidal neovascularization from myopia for these drugs. In terms of bevacizumab, there's a bevacizumab VIKG, which is a biosimilar that actually is trying to get the first FDA label approval, and it's going through all of these phase 3 North 1 to 4 studies. So we'll have to wait and see. So you might say, well, what can I expect from a biosimilar? You know, there's been no direct comparison in terms of like a protocol T type study. Instead, these biosimilars are approved on the basis of a clinical trial looking at a sensitivity endpoint. That is, they have to be compared to the reference drug at the time point of the highest change for treatment response. So for all these anti-VEGF drugs, you're going to look at the visual acuity change within the first 8 to 12 weeks. And we're going to see, do those curves overlap each other? That's how the approvals are based. And in fact, for both of the ranibizumab biosimilars, they did meet that endpoint. They did meet their sensitivity endpoint, and that led to approval. So technically, based on this, you can expect no clinically meaningful difference in terms of safety, potency, or purity of these drugs, biosimilars, from the reference products. So you might ask, well, why is it cheaper? Well, it's cheaper because the R&D is less expensive. They only have to compare to that sensitivity endpoint, dramatically lowers the cost for the clinical trials. Most of the costs are for upfront drug development. And so this is why biosimilars are less expensive. So in summary, when we look, at the initial drug, when we try to decide which drug to use, we can probably say early on that your best visual acuity outcomes can be achieved provided you pick a drug that will work. And if it doesn't work, you switch early only if needed for persistent diabetic macular edema, and you use the DRCR-like protocol. So up to 12 weeks. We don't know longer than that. The studies haven't been done. We also can say that clinical trials have shown similarity of biosimilars to reference products. So you can expect these to work similarly to how the reference products works and extrapolate the results from there. Also realize that your initial drug that you use will affect the time to the resolution of the diabetic macular edema. Some drugs will dry the retina faster, as we've shown. Some drugs will require fewer treatments. And some treatment burdens, in terms of the number of treatments, therefore may differ by drug. So I think what you save perhaps upfront in costs might require and translate to more treatments over time, or perhaps to needing a switch over time as well. But we're fortunate that we have all of these options today, and thanks for your attention. Any questions? I know this is another can of worms talk here. Questions? So I'm going to do a poll in the room. Who has used a biosimilar yet in this room for DME? So no one yet has tried a biosimilar. Okay, so it's probably because there are a lot of universities here and we're trying to get them on the approvals to get on the formularies. In this room, who has tried faricimab for DME? Okay, all right, and keep your hands up. Have you noticed an effect when you've used the drug in terms of better drying when you've switched your patients? Okay, so people are still raising their hand. Who has used faricimab as the initial drug treatment from the get-go for a patient with DME? Okay, and so I'm going to pick on you since you raised your hand. So Amani and Beryl, can you give them a mic? When you picked faricimab, why did you pick faricimab for that particular patient? Just out of curiosity. I've done it too, so.
Is the fellow eye of someone who was in the trial, and they've
Okay.
done so well in the altimeter trial, so we just went to faricimab first.
Okay, and Beryl, why did you pick it? Can you get him a mic?
So I think, and I don't want to give away too much from my talk that we're going to do some
Right.
real world data, but I think in patients that have been switched over, we see the drying effect. So the thought is, let's just go ahead and start. And a lot of our chronic DME patients, the ones that you think maybe would be better off with a steroid to begin with, those are ones that we're starting with faricimab.
And did you look at the visual acuity? Did that drive you better, worse visual
Yeah,
acuity?
they're
versus
generally
better?
worse vision. So 20/50 or worse.
And you too, Amani, worse vision?
Yeah.
Yeah, I would agree with that. The ones that are really thick, worse vision, just off the bat, try them on it. And then last question, in this room, who's been affected by step therapy? Raise your hand. Okay, so significant amount of people. And who has been successful in getting a branded drug to overturn the step therapy initially? None. Oh, one. Okay, great. And so I think, when you do talk to your insurers, and let's say you're having trouble and the patient's still not responding, do quote the AC results and just say, if we switch now after 3, if you approve this, we know that we can get similar results at 2 years, but I wouldn't know what to tell you after that. That might help inform your decision, their decision. Amani?
Yeah, I just wondered, you've tried to, so instead of doing the 3, start with the aflibercept and you succeeded? Is that possible? Like instead of going with 3 bevacizumabs and then the aflibercept, have you tried to actually not do the
You mean
step?
with a step therapy, but you'd have to get it approved by the insurance.
So they
I've
can
done
do
it
that?
because the other eye did not respond. And so I said, why do I want to start all over again based on the other eye and, comorbidities, try to get this
Oh, wow.
eye better, this
I
eye
didn't even
treated.
know that was an option.
You have to talk to the medical director to get that, but it does require some time on the phone. And again, I did this because my patient was adamant they didn't want to go that route of start the bevacizumab and then try this drug later on since they had that happen in their opposite eye. But nobody knows, right? Because I could have just tried the bevacizumab in this other eye and it may have worked. So it's just extrapolating. Dr. Skandra, Dimitri, you're going to say something? Oh, you're agreeing?
I also had a couple of patients that would go straight to aflibercept.
Okay, great. We'll move on now to Dr. Deeba Husain, whom I introduced earlier. And Dr. Husain will now speak about steroids. So rationale and outcomes for steroid for diabetic macular edema. Move it over here.
Thank you. So this is, I have no relevant financial disclosures. Diabetic macular edema is an important disease because it causes vision disability in the working age population worldwide. The Wisconsin epidemiological study of diabetic retinopathy has found about between 20 to 25% prevalence of DME in type 1 and type 2 diabetics after 10 years of diagnosis with diabetes. And the DCCT trial has reported about 27% prevalence of diabetic macular edema about 9 years after diabetes. Yao, who estimated global prevalence of DME, has reported a 6.8% of DME among all patients with diabetes, and also estimates that in the United States with non-Hispanic whites are less likely to have diabetic macular edema versus non-Hispanic Blacks in the US. And another important factor to know is that 50% of patients with diabetes who are not treated have a chance of losing 1 or 2 more lines of vision in 2 years. And that is the reason it is important to find an appropriate treatment. And as we know, each person responds differently. So what are the risk factors for diabetic macular edema? Longer duration of diabetes, 10 years is where most people have looked at incidence. And of course, poor control, so higher hemoglobin A1Cs, along with presence of hypertension and hyperlipidemia. And there's also some association with secondary risk factors, such as renal function and thyroid dysfunction to be associated with diabetic macular edema. So to understand why steroids can work in this process, it's important to also understand a little bit about what we know currently of the pathophysiology of this disease. So DME happens in mild to moderate diabetic retinopathy, and it's characterized by presence of intra as well as subretinal fluid causing retinal thickening in the macula. And chronic hyperglycemia is the basic mechanism that leads to accumulation of advanced glycolated end products or AGEs that disrupt the blood-retinal barrier, characterized by endothelial dysfunction and loss of pericytes. This is what leads to the leakage of fluid and thickening within the macula. There's growing evidence that Mueller cells may be the first to be affected in diabetic macular edema, and that activated Mueller cells are capable of recruiting leukocytes, causing the breakdown in the blood-retinal barrier, causing direct glial dysfunction and neuronal cell death. And with progression of disease, these Mueller cells can become apoptotic, which then inhibit tissue repair, as has been shown in several preclinical studies. And then there are molecular mechanisms that lead to diabetic macular edema, VEGF playing an important central implication, and that's why anti-VEGF drugs are the first line at this time for diabetic macular edema. But inflammation overall plays an important role in both the causation and the worsening of diabetic macular edema. And there's expression of pro-inflammatory cytokines, such as intracellular ICAMs, interleukins, IL-6, IL-8, tumor necrosis factor, and cycloogenase 2 that are all upregulated and cause neutrophil and monocyte attraction to this area and vascular permeability deterioration. All these inflammatory factors cause changes in the neurovascular unit. Therefore, changing the homeostasis between the cells within the retina, such as the astrocytes, the ganglion cells, the Mueller cells, endothelial cells, and amacrine cells, leading to vasodilation and leakage of fluid and reduction of vision. So it does make sense that steroids should be beneficial in the treatment of diabetic macular edema because corticosteroids play a very powerful anti-inflammatory and anti-edematous effect by targeting the synthesis of all the pro-inflammatory mediators involved in DMEs that we just talked about, like IL-6, IL-8, MCAM, ICAM, TNF-alpha, VEGF, as well as GF and Ang2. And one of the things that corticosteroids does is reduce the VEGF level. So just like anti-VEGF drugs work, the steroids should work also. So therefore, the rationale of using steroids for DME involves targeting of the inflammatory processes and preventing changes in the retinal glial cells. At this time, as Jenny just talked about, the first line therapy for DME is anti-VEGF treatments, but several clinical trials and studies have shown that 40 to 50% of the patients on anti-VEGF therapy for 6 months, monthly injections, have persistent edema. And therefore, they do need something else besides anti-VEGF therapy for the swelling. And then there are certain patients that have relative contraindications to use of anti-VEGF, such as patients with recent history of cardiovascular events or strokes, pregnancy, some patients that are unable to adhere to the monthly injection regime, and then those diabetics that are undergoing cataract surgery sometimes need a stronger coverage besides just the anti-VEGF. DRCR, a network T protocol, reported that after 2 years, the rate of persistence of DME ranged about 44% in patients with Eylea and 68% in patients with Avastin. And therefore, many of us seek alternative treatments beyond anti-VEGF in an attempt to reduce the central macular thickness and help optimize and improve the vision in our patients. But there's no clear-cut guidelines as to when patients should be either switched or a secondary added steroid treatment should be done. The American Society of Retina Specialists conducted a survey asking retina specialists when they incorporate steroids in their therapy. 38% of the US specialists responded that after 4 to 5 injections of anti-VEGF, if there is no response, they switch. 28% responded that they try anti-VEGF injections for 6 to 9 injections. And a small percentage reported that they sometimes switch after the first injection. There's some interesting work by Monk and colleagues with predictive OCT biomarkers for steroid response. In this work reported in 2022, they have reported that those patients that are poor responders to anti-VEGF therapy are linked with low or normal low levels of anti-VEGF in the serum and aqueous. And in these patients, there are certain OCT predictors that may tell us that they will respond better to steroids. These include presence of large amounts of hyperreflective foci, exudates as we see them clinically, disruption of the outer retinal layers, and other signs of chronic intracystic disease extending into the outer retina, and a low choroidal vascular index. And these patients might benefit from kind of an early switch from anti-VEGF to a steroid therapy. So what are the options available in the US for steroids? Triamcinolone, both the preservative-free in the form of triesence and Kenalog is available at 1 or 4 milligram doses is used as intravitreal injections. Dexamethasone, which is available as an implant, long-acting implant, and dispensed with the injector as Ozuradex, and fluocinolone, which is the Iluvien, again, a further longer 3-year lasting implant that is available as an injector, and I'll talk briefly about each. So triamcinolone, as we said, can be used in 1 or 4 milligram doses, and it can be injected, as quickly as 8 weeks, but sometimes up to 12 to 16 weeks, depending on how long it's lasting in each eye. The Protocol I DRCR network study had shown that there were reductions in central thickness in triamcinolone arm. This was a study which was looking at Lucentis versus triamcinolone comparative study, and they showed that the central macular thickness was very similar in the two with the two medications. And in the group, which was pseudophakic, 273 patient sub-analysis of the pseudophakic eyes showed that the vision improvement was also comparable to Lucentis in that arm. And of course, there are very significant side effects in terms of cataract acceleration in almost all the phakic patients. And about 50% of triamcinolone patients experience clinically relevant increase in eye pressure that needed intervention in terms of medications. The other steroid that is available in the market is a dexamethasone or 0.7 milligram biodegradable implant by the name of Osirodex. It is dispensed, as I have shown in this picture, as with an injector, loaded on an injector for us. The clinical study that was done to look at the efficacy and safety of this was the MEAD study, which phase 3 of which showed that there was improved vision by 15 letters in about 22% of patients as compared to sham at 3 years, and that the macular thickness was also greatly reduced in patients with this dexamethasone implant. So at this point, it's FDA approved for use, and it has been shown to last 3 to 6 months in the eye. The side effects, again, being that 68% of these patients develop cataract, and 41.5% of the patients need anti-glaucoma medications, with about 0.3% of patients needing glaucoma surgery. And the fundus image shows what the implant looks like once it's injected in the eye. Fluocinolone or Iluvien is the other implant. It's 0.19 milligram dose, sustained release, which is effective for 3 years in the eye. The clinical study done to evaluate this for safety and efficacy is the FAME study, which showed 15 letters of vision improvement in about 30% of the eyes. But again, 82% of these patients developed cataract, about 40% needed anti-glaucoma surgery, and about 4.8% needed surgery. So I'm sorry, 40% needed the medications for management of glaucoma, and 5% needed surgery. But those eyes that were challenged ahead of time for steroid and were shown to be non-steroidal responders, actually there was no increase in IOP in that group. So at this time, the FDA approval for DME in patients who have been previously challenged with corticosteroids, there's an approval for this medication for those patients. The biggest problem with steroid use is there's a very significant prevalence of cataracts. Almost everybody, the cataracts increase, and many patients need cataract surgeries. There's a very significant prevalence of IOP rise, reported anywhere between 30 to 40% patients needing medications for control of glaucoma, and up to 4 to 5% of patients needing glaucoma surgery. They're the usual complications of intravitreal injections, and a rare complication of migration of the implant, like shown in this photo, where the implant migrated into the anterior chamber, and those need to be taken out surgically. So talking about outcomes with steroids is controversial because there are not that many studies looking at this, and the studies that are there have significant limitations in terms of efficacy of steroids. Protocol B of the DRCR was one of the first studies to look at 1 and 4 milligrams of triamcinolone versus at that time, the photocoagulation study to look at the effect of response to diabetic macular edema. 689 patients were enrolled in this study, and it was a 3-year outcome. The authors concluded by saying that there was no long-term benefit of intravitreal triamcinolone as compared to focal laser. But the big problem with this study was that only 36% of the patients in this 3-year follow-up had information on the study. So this could have biased the results of this study. Subgroup analysis within this study did show that there was significant improvement in the macular edema, and also that the vision improved in patients that were receiving steroids. The same thing was with Protocol U. This was a study the DRCR network did comparing intravitreal Lucentis alone in patients, and those that did not respond to then add a dexamethasone implant, Osirodex, for persistence of DME. And this study showed that dexamethasone implant could reduce the retinal thickness, but it did not improve the vision. The limitations of this study were that there was no clear-cut definition of persistence of edema. And even if the DME improved, it was still called as persistent, and that may have biased the results of this study. At the subgroup analysis in this study of patients with less than 20/50 vision actually did show that combination of steroid and Lucentis was better than steroids, than Lucentis alone. And at the end, I would like to talk about a Cochrane review that kind of reviewed all the studies done in evaluating DME, looking at the efficacy and safety of steroids versus any other treatment, which included observation, laser, anti-VEGF. 10 trials were included. There were over 4,000 patients included in the trial, and they concluded by saying that intravitreal dexamethasone implant, or Osirodex, improves vision more than sham, but the efficacy of dexamethasone implant versus any anti-VEGF drug, whether it was Avastin or Lucentis, was inconsistent, even though macular thickness reduction was very significant with the steroid use. Again, intravitreal fluocinolone implant was capable of improving the vision more than sham, but it was as similar to a response with laser, but less than anti-VEGF injections. And of course, Cochrane review showed, again, what we had seen before, that there was significant prevalence of cataract, as well as very high rate of increase of IOP needing treatment and surgery. And there is a very large meta-analysis, which is an international review done to look at safety and efficacy among the different intravitreal corticosteroids for DME. This included 19 eligible randomized studies of about 3,000 patients. And this study did show that use of any steroids, whether it's triamcinolone or dex or fluocinolone, is capable of both improving the vision as well as reducing the central macular thickness. And interestingly, the higher the dose of steroid, the better is the response. Higher doses are available in the rest of the world. It's not approved here for us to use, but Europe and Asia, they have available higher doses, and they have shown that higher doses work better, especially for triamcinolone. So I'd like to summarize here by saying that inflammation plays a significant role in the pathogenesis of DME, and therefore it makes sense that steroids can be beneficial for treatment, either alone or in combination therapy. We do know that it has significant side effects in forms of cataract and glaucoma, and therefore the best results in steroids are seen in patients who are pseudophakic and non-steroid responders. Thank you.
Thanks so much, Deeba. That was great. My question to you is, we know that as DME progresses, the cytokine profile shifts, and that with chronic DME, there are more inflammatory cytokines that happen over time, and then the VEGF cytokines drop. So knowing that, how does that inform your use of steroids? You know, would you use it as the initial if somebody's had DME untreated for a long time, or would you always try an anti-VEGF?
Usually I do try always anti-VEGF. It's interesting work to show that there is a small group of people where the VEGF levels are actually low, both in aqueous and in serum. So maybe that would be a group where you could try steroids primarily, but I think in our practice, we all start with anti-VEGF. So.
Is there any way, you know, I know there's like Raman spectroscopy and you can like see what the level of the glucose is like in the aqueous. Is there anything like that? Is there a VEGF meter out there or no?
Not that I'm aware of.
Okay, so I see a bunch of bioengineering grad students in the back. We need a VEGF meter. Any other questions for Dr. Husain? Thanks,
Thanks.
Deeba. Okay, if not, we will move on to our next speaker, who is one of our former residents here, Dr. Viral Shah. So Viral is a clinical assistant professor here at UIC. He was an ophthalmology resident where he excelled, and he was selected as one of our chief residents. And then he went over across to the other side to the University of Chicago, where he got his vitreoretinal training. And he then also got an MBA at Northwestern from the Kellogg School of Management. He's currently a partner at University Retina and Macula Associates, and he's very interested in clinical trials. And as you'll see, I think you're going to share some data with us, perhaps from the Truckee study. So Viral is going to be speaking to us now on evaluating real-world outcomes in AMD and DME. Viral?
Thank you, everybody. And thank you, Jenny, for that nice intro. And thank you for inviting me to talk about some real world outcomes in terms of neovascular AMD and DME. Here are my financial disclosures, some of which are relevant to today's talk. Okay, so here's how the talk lays out. We'll talk a little bit about neovascular AMD and some of the real world data. We'll go to DME and then talk about unmet needs, which Jenny and a lot of the folks have really laid out already with some of what we need to see in our space and what's being done in our space. And then, as Jenny mentioned, we'll talk a little bit about some real world faricimab data, which is known as the Truckee study. So let's just dive right into the neovascular AMD. And I think Jenny brought this up in both of her talks, really, that we do see an unmet need here. We've been fortunate to have anti-VEGF therapies, which have revolutionized our care. And it's really nice to be here talking about this. We've got kind of giants in the field of anti-VEGF therapy online today. We heard great talks from Phil. And so, you know, I like putting this slide up, especially for the trainees in the room who weren't around, you know, to see this kind of transition in what we've kind of been able to do in patient care and patient management. You know, we've known about VEGF. We may not have called it VEGF back in the early days, but we've known its role in different pathologies. But in the last 2 decades, really been fortunate enough to be able to target it in diseases like neovascular AMD and DME. And as a lot of speakers earlier pointed out, we're starting to now look beyond VEGF because while we've been able to see good results, we know that there's still some unmet needs. So that being said, the results we have seen, so these landmark studies that we've seen with VEGF have set the bar really high. And I think as a result, we haven't seen a lot of breakthroughs in 2 decades. And let's talk a little bit about that, right? So we know that in the real world, we don't see those same visual results that we do in clinical trials. So on the left here in purple, you see kind of what we see in our landmark studies, Anchor and Marina being really the game changers for us back in 2005 and 6, showing robust visual gains in our neovascular AMD patients. But what we see in the real world really doesn't come close. So when you look at, I think Lumiere, I think Jenny, you brought this study up, there's lots of real world studies coming up. And now with our ability to mine data sets like the AAO IRIS registry, we were able to look at real world data and see that our average BCVA change doesn't come close to what we see in clinical trials. And we can talk a little bit about why that might be. So we know, and again, from real world data sets, we know that the more treatments we give patients, in general, the more injections they get, the better they do from a vision standpoint. So you can see on the right of this curve here, patients that got 10, 11, 12 injections in the course of that first year, they have better vision gains, as opposed to the patients that don't get that many injections. On the left of that curve, you often see vision loss in those patients. I think Alice brought that up earlier today. So, when you look at this bar graph, what we're really plotting out is what percentage of patients are getting 1 injection, 2 injection. And what you see is the vast majority are getting 6 injections or less in neovascular AMD. So what about DME patients? When we look at the data, it's very similar, right? So when we look at VISTA and VIVID on the left, these are, phase 3 landmark studies looking at aflibercept for DME. And we saw 10.7 letters of vision gain in those studies. What do we see in the real world? Tom Chula has a really nice paper out now where we see 4.3 letters of vision gain in our DME patients that we are injecting after 1 year. So again, not even half of the vision gains that we're seeing in clinical trials. If you look at this data, and I think someone had presented a slide like this earlier, but if you look at AAO iris registry analysis, you can see that almost 50% of patients that are getting anti-VEGF therapy are getting 3 or fewer injections. So when I show you data that's 10.7 letters of vision gain, that's with 8, 9, 10 injections in clinical trials. And so when we see in real world what we're doing, we're really not treating as often as we do in clinical trials, which is likely the reason we're not getting the vision gains that we see in those clinical trials. Again, just another Lumiere is another data set from the real world. And if you look here, 5-year open label prospective study, just collecting data on what we're doing in the real world. And we see at 1 year, we have vision gains in our DME patients of 3.5 letters. And you can see really the dichotomy. So patients that get 4 or fewer injections, we're seeing 0.5 letters of vision gain versus 5 injections or more in these patients, and you're seeing 6.9 letters gain. So within the question is, well, why in the real world are we not treating more often, right? And lots of reasons why. Having trained at UIC, and talking to the fellows and residents, we know that a lot of our patients just can't maintain that level of treatment. So we see a lot of loss to follow-up, and lots of data on this slide, but really the gist is over a quarter of our patients, no matter what, whether they're PDR patients getting laser, getting injections, or they're NPDR patients getting treatment for DME, injections for DME, over a quarter of them are lost to follow-up. And there's been really nice work done in this area looking at the reasons why. So we know that the younger patients in general are lost to follow-up. Why? They have to work. They have other issues going on. They have families to deal with. So we know that those are patients that aren't able to come in as often. And then we know it differs by race. So if you look at the data, you see 19.4% of our white patients are lost to follow-up versus 30% or more when you look at African Americans, Hispanics, Native Americans, Pacific Islanders. And so, why is that important to know? Because we need to educate patients and talk to them about the importance of treatment. Carolyn Baumel had a really nice paper in 2020 looking really into the reasons of non-adherence. And I won't read this list, you can see for yourself. And a lot of it is stuff, it's common sense. We know, we've got to educate our patients. We know that there's difficulty for these patients to come in, whether it's transportation, mobility, or the need to go to work and not having the days off of work to be able to balance these type of treatments out. So that kind of dovetails into this conversation of unmet needs. And Jenny had a great conversation about new treatments that are coming out, because we know anti-VEGFs are great. The bar has been set high, but we know, I think Deeba just mentioned Protocol T. There's still 40, 50, 60% of our patients that still have residual fluid despite injections every 4 weeks. So we've got to talk about, what the unmet needs are and how to address those unmet needs. So when we look at the PAT survey, and if you've taken the PAT survey, great. If you haven't, I'm on the committee that writes the questions. Please take the survey. But these are very important data sets for us, right? We learn from our colleagues. And what we've seen in these surveys is when we ask, what are the big unmet needs? You'll see time and time again, the top 3 reasons. We need to reduce the treatment burden, because I think we recognize we cannot treat patients every month or every other month for a long term. And then long-acting sustained delivery. We mentioned earlier gene delivery, port delivery system, just lots of different ways we're looking to solve that problem. And then new mechanisms of action. And that's really kind of an important one for us because we're starting to see that now, right? We're starting to see that develop in our field now. So, and I won't talk too much about this because there were some really great slides on this earlier. But faricimab is just one example, and this is going to connect kind of to some real world data that I'm going to mention in a minute here. But we know that these diseases are multimodal. They're not just driven by VEGF. While they are, VEGF's a big player in that. We know that there's other factors, and Deeba just mentioned a whole host of inflammatory mediators. Well, one of them is Ang2, and I think we had a really nice kind of explanation of why potentially addressing Ang2 might help us add to what we've been able to do with the anti-VEGFs alone. So faricimab is one example of a molecule that targets both of those pathways. And then just briefly, when we see a new mechanism of action, what we also want, and if you looked at those top 3 unmet needs, one of them was durability. We need stuff that lasts longer than 4 weeks or 8 weeks. And what you're seeing, fortunately, in these data sets, so Tanaya and Lucerne was looking at neovascular AMD, phase 3 studies looking at faricimab and neovascular AMD. We see that 74 to 81% of patients can get 12 weeks or more interval between their injections, with over 60% getting to 16 weeks between treatments. So, the point here is that if we are not able to treat people in the real world as often as we need to, well, then what we need to do to bridge that gap is get treatments that last longer in order to maintain or achieve those results that we're seeing in clinical trials from a vision standpoint. That was AMD data. This is DME data. Again, Jenny did a really nice job presenting this, so I won't harp on this too much. But again, just wanted to point out that these newer treatments, what we're really looking for is not only efficacy, but durability. And we're seeing that again here in DME patients, we're able to get over 60% of patients to 16 weeks between their treatment intervals and still maintain good vision outcomes. All right, so let's talk about the Truckee study. So what is the Truckee study? The Truckee study is basically a consortium of study centers around the country collecting real world data on faricimab. And in Truckee, what we're doing, we're one of the clinical trial sites, not industry sponsored, so an independent kind of study group looking at real world data. This is particularly looking at neovascular AMD and the treatment of neovascular AMD. And what we're looking at is collecting some data points, right? So demographics, prior treatment history, efficacy, which not only includes vision, but also some imaging modalities, central subfield thickness, and looking at fluid status, PEDs, things like that. And then looking at durability, how often are we treating these patients in the real world? And then, of course, very importantly, safety. I mean, if you've been working with these newer treatments, you can see that when they hit the real world, safety is a really big part of what we're looking at now in the real world, because we've seen some of the newer agents not be as safe as we were hoping, or not as safe as what we might have seen in the clinical trial. So what are we seeing so far? So you guys are going to see these data sets come through. If you haven't already in some of the big meetings, you're going to see some of these at the big meetings, ASRS, ARVO, AAO. So this is just kind of a sneak peek as to what you might see in the coming meetings. And this data set's going to continue to grow and evolve as we continue to generate new data and collect more patient information. But so this is the latest kind of information that we have. We have 491 total patients that have been treated with faricimab in this study population. That encompasses over 1,200 intravitreal injections. We have follow-up after 1 injection in 335 patients, and follow-up after 3 faricimab injections in 89 patients. And so I'm going to kind of just dive into that data now. Here's some demographics, right? So mean age, again, this is neovascular AMD, so the mean age here is not a surprise. A lot of these patients, if you see here, are patients that were switched. And I think this is kind of what you see when a new agent becomes available in the real world, is a lot of the early use is in patients that we were switching over. Jenny asked the question of, are we using this in new patients or treatment naive patients? We are. And so at the bottom there, you can see 10% of this data set is treatment naive patients. And that number is growing as we start to collect more data. So just overall efficacy, and I'm gonna cut into this a little bit more and give you just more kind of nuance here in a minute. But overall efficacy, we see that after 1 injection, we're seeing modest vision gains. Now remember, the vast majority, 90% of these patients are patients that were switched from another agent. But we are, despite the fact that a lot of these are previously treated patients, are seeing improvements in anatomic findings. So CST thickness improvements, PED height improvements. And that's what you want to see if we're looking for progress in newer agents, right? So let's break that down a little bit. So let's look at just after 1 injection in treatment naive patients. We're seeing what we expect, more robust vision changes. So after 1 injection, 4.9 letters of vision gain, and more robust CST and PED changes with 84 microns of CST improvement. Again, that's just after 1 injection in treatment naive patients. These are patients that were switched from a different agent, right? So if we look here, again, not a surprise from a vision standpoint. After one injection, we're seeing a modest change in vision, but we are seeing significant improvements in CST thickness and PED height. And I'll dive into the CST question because people will ask, well, is that intraretinal fluid? Is it subretinal fluid? We'll talk about that a little more in a second here. So here's the breakdown. If you look at patients that had intraretinal fluid, subretinal fluid, or a PED finding, if you look at that, the ones that you see in blue here, the bar graphs in blue, are patients that had those findings pre-faricimab. If you look after one injection, you see that bar graph drop down. In other words, fewer patients had intraretinal fluid, subretinal fluid, and PEDs after that one injection. Again, what you want to see when you switch a treatment agent, you want to see anatomic improvement, and that's what we're showing here. Now, this is a group of patients that were switched from aflibercept. I told you the majority of the switch patients, about 60% of the patients, were patients that were switched from aflibercept, which is not a surprise. It's one of the more commonly used on-label medications out there. And we see, again, similar trends in modest vision change, but again, a drying effect from a CST and PED standpoint. Let's look at that same kind of IRF, SRF, and PED breakdown. Again, reassuring here, where we see patients that still had persistent fluid on the aflibercept, we're seeing few of those persistent fluids on faricimab. And that's, again, after just one injection. So we're excited about showing this data after 3 injections, after 6 injections, and as time goes on. And then this is patients after 3 injections of faricimab, all comers. We see 3 letters of vision gain, and then again, anatomic improvements, and we'll break that down here in a second. Again, after 3 injections, seeing nice, drying effect. So more patients that have an absence of intraretinal fluid, subretinal fluid, and PEDs. Efficacy after 3 injections in patients that were switched. So this is kind of, for me, the data point that's the most important, which is these are patients that are getting therapy in our office, but there's still room for improvement. So up until now, a lot of these patients were getting some anti-VEGF at a fairly frequent interval, but still had room for improvement. And you can see that room for improvement here is that patients are still gaining vision after 3 injections in the switch, still seeing improvement in anatomy and PED height. And again, similar trends in these groups that have 3 injections of faricimab. More of them are dry, fewer of them have intraretinal fluid, subretinal fluid, or PEDs. The last kind of important data point is safety, right? So in these 491 patients, in this 1,231 injection group, we've seen 1 case of infectious endophthalmitis, 1 case of mild intraocular inflammation, which resolved after 2 weeks of topical steroid therapy, and 0 cases of retinal vasculitis, 0 cases of retinal artery occlusion. We know that there's been worldwide about 500,000 doses of faricimab given so far, and 0 cases of retinal vasculitis. And again, if you've been part of any of these real-world data sets, you know that that's a really growing of importance as far as safety goes. Just end it with a couple of quick cases here. This is a patient of mine. You can see was on aflibercept in the top left, had kind of persistent subretinal fluid, switched to faricimab 35 days later. So it didn't really change the interval too much, maybe went from 4 weeks to 5 weeks, but you can see a nice drying effect. There is a little bit of subretinal fluid in that top right picture, just a little bit compared to before. So we treat a couple more cycles, and after 2 more injections, so again, this is after the 3rd faricimab, you can see on the bottom, really nice dry OCT and some vision improvement as well. Just one last case here. This is a patient of mine with intraretinal fluid that I was treating fairly regularly for many years with the aflibercept at about a 40-day interval, less than ideal. This is someone that you probably want a shorter interval with, but as you know, it's not always possible in these patients. But you can see after one faricimab, a really nice drying effect on the right and some vision improvement. Here, intraretinal fluid bothers me more than subretinal fluid. So really kind of pleasantly surprised to see this. This is one of our earlier cases of a switch. So just in conclusion, we know, and it's been mentioned a couple of times here, real world outcomes for neovascular AMD and DME just don't match clinical trial outcomes. And so the goal now with clinical trials is really to work towards addressing some of these needs. whether it's with durability, whether it's efficacy, whether it's new mechanisms of action, I mean, and all of the above. And it's really critical to look at real world data because we learn a lot about how we practice. We learn a lot about the disconnect between clinical trials and the real world. And it's helping us modify clinical trials. You know, Jenny went into these DME studies that we're doing with faricimab and modifying it, looking at kind of treat and extend type of protocols that we're doing in the real world so we can really get an idea of how we're helping these patients and bridging that gap that we have right now. Thank you.
Thank you so much, Viral. That was a great talk on how we use this drug in the real world today and the impact that it's actually having. You know, to supplement what you said, when we look at the matched dosing phase in the faricimab studies, you know, for the first 3 where you're getting the faricimab and then you're getting the aflibercept, when we look at the percents that have fluid, there was a greater reduction in the fluid in the faricimab arms compared to the aflibercept arms. That goes nicely with what you were showing here clinically. And we also are seeing, I think, clinically, the PEDs are coming down, which is something that we didn't typically see, right, when we treat these eyes with AMD. Questions for Dr. Sheth? Yes, Demetra, can we give her a mic, please?
An overall question, with these new extended durability studies, we tolerate more fluid and more visual loss than before. So are you still aiming for dryness or you apply this to your real world data?
Yeah, so it's a great question. So the question is, you know, in these clinical trials, we do allow for some fluid, right? If you look at even protocol AC, right, we had to hit a certain threshold before we changed their interval. In my real world practice, don't necessarily tolerate a whole lot of fluid. So in a lot of these studies, they would go 4-week changes in their interval. I mean, I think a lot of us in the real world will go 2 weeks because we want to see just that. Are we allowing for fluid? Are we allowing for vision changes? And if I do see it, I will kind of keep the interval contracted. Yeah.
So do you think it will still be less injections?
I do. So I still think, so your question really is, are we really seeing extended durability in the real world? And I will tell you, yes. The one thing that these real world data sets tell you that the clinical trials don't are these patients that you switch, right? None of the clinical trials involve patients that you switch in the middle of a study. And in those patients, we are seeing with these newer treatments, our ability to dry those patients out at the same interval, but then also at the same time extend them when previously we were not able to extend them. Yes.
You had somebody who was aflibercept maybe every 8 or 9 weeks because there was fluid, but the vision was 20/25 and you were watching them. And then you decided, okay, I'm going to switch them now. Are you giving, do you do the switch to a shorter duration or do you still stay at the 8 or 9 week intervals?
Okay, so this is a little bit more complicated, right? So I've changed the way I do it. So initially, I would just change the one variable, the drug, right? So I wouldn't change the interval. In my diabetics, what I'm finding is I do need to stack those treatments a little closer. And I think it's to get the anti-Ang2 effect. But then I am able, now that we've had this out in the real world for a year, what I'm seeing is I am able to then extend them out further. But it does take a little bit more impact up front, especially in the chronic diabetics. In my AMD patients, I'm just switching, keeping the interval the same, and then seeing if I can extend.
Great. Thanks again, Viral. And we look forward to seeing extended Truckee data that will help inform our management. So our next speaker is Dr. Mansoor Abtahi. And Mansoor came to us after completing his PhD at the Sharif University in Tehran in 2013. Now, you might not know the Sharif University in Iran is the highest ranked technological university in the Republic of Iran. So we're very happy to get him. And he had worked for some time in Iran before coming here. He is currently a postdoctoral research associate in the Biomedical Optics and Functional Imaging Lab at UIC under the direction of Dr. Shencheng Yao. So Dr. Yao was called away for a family emergency and had to go to Asia and recommended that Dr. Abtahi give this talk. So we're very happy to have Mansoor with us today. He's focusing on using deep learning and computerized vision techniques for the segmentation and classification of OCT and OCTA datasets during his PhD. And Mansoor will be speaking to us today on the topic of arterial and venous flow flux analysis in OCTA. Abtahi.
Hello, everyone. I'm Mansoor Aptai. I'm presenting on behalf of Dr. Xingchen Yao about our research on differential artery vein analysis in OCTA. When we are analyzing vessels in OCTA images, we can ask this question: what will be the result if we separate arteries from veins? In our previous studies, we used the spectral information in either fundus images or OCT images to distinguish arteries from veins. And we use this information as a guide to segment arteries and veins in the OCTA images in the macular region. We used differential artery vein analysis on blood vessel caliber and blood vessel tortuosity for DR and sickle cell retinopathy staging. Here we can see that mean blood vessel caliber has a little change during different stages, but by separating arteries from veins, you can see that the caliber of the arteries are decreasing, but the caliber of the veins are increasing. The ratio of the artery vein caliber can increase the sensitivity for DR staging. We did a similar study for sickle cell retinopathy, and our result shows that most of the changes occur in the veins. For the first time, we developed a deep learning model to segment arteries from veins with the OCT and OCTA images as input. The output of this model is artery vein maps or AV maps. The accuracy for segmenting artery and veins in this model was about 86%. In a follow-up study, we compared different configurations with different data inputs, such as OCT only, OCTA only, and early fusion of OCT and OCTA images, and late fusion of OCT and OCTA images. As you can see, the OCT only has the lowest performance as we expected because OCT images has less vessel details than OCTA images. OCTA only, early fusion and late fusion have competitive performance, and late fusion with late fusion architecture with 96% accuracy has a slightly better performance than OCTA only. Here we can conclude that OCTA only architecture is sufficient enough and more simpler than the late fusion architecture to segment arteries and veins. Different retinopathies may affect capillaries first. Because of this, we did artery vein segmentation for capillaries in 3x3 images. For generating ground truths for 3x3 images, we used 6x6 images as a guide. And this is the performance of different configurations for 3x3 data set. And as you can see, the ranking of the different architectures are similar. And we can see that using the 3x3 images, we are able to segment capillaries, and OCTA only architecture in this case is sufficient enough and simpler than late fusion architecture. AV maps are binarized vessel maps. After generating AV maps from OCTA images, we are losing the intensity information of the OCTA images. Because of this, to preserve the intensity information of the OCTA images, we developed a new deep learning model to generate arterial venous area maps. By multiplying OCTA images and AVA maps, we can have OCTA AV maps. OCTA AV maps has all the intensity information in the OCTA images, but in red and blue channels for arteries and veins separately. Using the OCTA AV map, we can calculate all the OCTA-related quantitative features for arteries and veins separately. We also introduced some new quantitative features using AVA map and OCTA AV maps. From AVA map, we have arterial area and venous area. As you can see, between control to mild DR, there is a significant increase in arterial area and a significant decrease for venous area. We also have perfusion intensity density from OCTA AV map. Perfusion intensity density is the mean of the intensity values in a desired area. We define total PID, arterial PID, and venous PID. As you can see, for all of the PIDs, from control to mild DR, PID is decreased. But from control to no DR, total PID has not changed. Arterial PID is slightly decreased, and venous PID is slightly increased. Because of this opposite AV changes, by defining the ratio of the arterial PID and venous PID, we can have a sensitive biomarker to differentiate artery groups from each other. We also developed another quantitative features called normalized blood flow index, using the perfusion map and noise map of the OCTA images. We use some kind of thresholding method, segment perfusion map and noise map from the OCTA images. And we compared these features with blood vessel density and blood flow flux. We calculated this feature for superficial and deep layer of the OCTA images for control nodal and mild DR. And as you can see, only normalized blood flow index of the deep layer can differentiate artery groups from each other. To summarize, differential AV analysis can increase OCTA sensitivity. Deep learning AV segmentation at capillary level is feasible. MF-AVNet shows that OCTA itself can provide sufficient information for robust AV segmentation at microcapillary level resolution. And AVANet enables quantitative AV area and intensity analysis. Normalized blood flow index promises a sensitive biomarker for early DR detection. And thank you for listening. I'm open to any comment or questions.
Thank you so much, Mansoor. Yeah, I think, looking at the arteries and looking at the veins in different stages of diseases is going to be really important to try to detect the earliest onset of disease. And I think right now we're looking a lot at diabetes and looking at diabetes without visible retinopathy and looking at blood flow changes. And I think looking at the artery versus the vein separately is important because, as you showed, they go in opposite directions. So my question to you is, do you think that this AV automatic segmentation is going to be able to be used by other people, open sourced? Is it there yet, or is it going to still take a little bit more work before we can get it to them?
Our neural network is publicly available. For the case of the data set for training the neural network, if any university is interested, we can communicate and give them the data set for training the model.
Thank you. Any other questions for Dr. Mansour? If not, thank you so much. And we'll move on now to Dr. Dimitra Skandra. So I've known Dimitra for some time now, and I first heard her present, I think, during an award lecture for the fellows at the ASRS or Retina Study, one of those. So Dimitra is another person who is a superstar, but this time in Greece. So she came over from Greece, where she had graduated with highest honors from medical school, and then came over and did a research fellowship, initially at Mass Eye and Ear, right? Then you did a postdoc also at Mass Eye and Ear, and then you went to Cornell and did your residency, and then did your fellowship at Harvard, and subsequently joined Northwestern, and also worked at the county hospital for some time from 2013 to 2016, and then got recruited to University of Chicago, where you rapidly rose to associate professor of ophthalmology and hopefully soon professor of ophthalmology. I helped write your letter, so hopefully that will happen. And so Dimitra is a physician scientist. She is the founder and leads the University of Chicago Retinal Microbiota team. She also founded and secured funding for the University of Chicago Retina Big Data Computational team. And one of the results of this work is the discovery that metformin may be protective for AMD. And she does a lot of this research. She involves her residents and her fellows. And I think you're one of the few faculty who've had both a resident win the Beam Fisher competition and also a fellow win the Beam Fisher competition 2021 and 2022. She's also on the University of Chicago Women's Surgeons Honor Roll and a 2022 member of the University of Chicago's NIH NIDDK sponsored Center for Interdisciplinary Study of Inflammatory Intestinal Disorders. So Dimitra is probably one of the world's experts on the gut microbiome. And in the past, she used to work with, I should say, mouse droppings or mouse feces. And now I understand that you don't have to actually use that because they're in pill form now. So I'm happy to hear that. So it's a little bit, not in the lab, but I think for humans, it's in pill form now. So you can improve your gut microbiota, which is extremely important nowadays. So Dimitra, I give you Dr. Skandra, who will speak on metformin and retinal diseases.
Thank you, such a generous introduction, and it's great to be here. So a little change of, I'm very short, so I think you'll see half my head there, sorry for that. Change of gears, so do I use this one? Yeah. We're going to talk about not injections anymore, but how something that is used in the rest of the medicine can be used for retinal diseases. And do I go? All right. I do right click. Okay, all right. I have no relevant disclosure. So metformin has gained a lot of attention in the AG world, like Silicon Valley techs start taking it early. I'm actually even considering myself that they call it the miracle drug. There's a lot of articles in the Times and tech journals. And then actually they're thinking of starting a very big study for prevent aging with metformin. They actually trying to take aging as a new disease definition for the FDA and NIH, and actually going to start a trial called TAME, hopefully soon. They're actually going to start 10,000 patients on metformin and follow them for 10, 15 years and see if they prevent any prospectively aging diseases. And the reason the trigger is such big studies, it's going to be a multi-million-dollar study, is that they found that patients that take metformin, they have less cardiovascular disease, less cancer, they have less neurodegeneration, in particular dementia and cognitive function. And the additional patients, diabetics that take metformin, have the same life expectancy with non-diabetic patients compared to patients that take metformin. And these are mostly based on retrospective data sets. So what about the aging disease of the eye? When you read about how metformin modulates the biology of aging, you actually see a lot of overlap between what we know that how metformin modulates how the pathobiology of AMD. with inflammation, oxidative stress, mitochondrial function, microbiome, SIRT, and all this like oxidative stress biomarkers, lipid metabolism. So the big question is like, does metformin decrease for AMD? I'm going to cover this, and I'm going to focus most of this because this is the most known data about this, both for big data, computational data, and also what preclinical data show. So let's start with big data first. So does metformin use decrease the risk of new onset AMD? So there's been a lot of interest in this topic. So the last 4 to 5 years, a lot of studies that we published from several groups, including ourselves. So I'm going to touch base those that show an effect and those that don't show an effect. So I'm going to start with a study we published a couple of years ago. It's actually the biggest study in the literature that will basically look at patients who have a new diagnosis of AMD without any AMD diagnosis for the years prior and have consecutive drug coverage and also medical coverage to look at other medications and also other diseases and see if those that take metformin have less risk of developing any new AMD. This is mostly using ICD-9 data codes because we finished a cohort study in 2017. And we found that lower to moderate doses of metformin was associated risk, decreased risk for developing a new AMD, both in the whole cohort and also the diabetic only cohort. But the effect was only found in patients that have no diabetic retinopathy. Around the same time, another study came out from the University of South Florida, a much smaller study that looked at 2,000 cases of AMD versus controls. And they also found that metformin was associated with significantly decreased odds ratio of AMD, which was not noted with other diabetic medications. So kind of like arguing that it's not just decreasing, improving the glucose metabolism, but actually there's something about the metformin drug itself. But it was a smaller study. The groups were not very well matched for the diabetes incidence. And also they did not account for smoking, which we know is a well-accounted AMD risk factor that likely we could address in our study. Jay Stewart was actually one of the pioneers. He's one of the first people that started working on this field. And he published around the same time with us a cross-sectional study that was about 3,000 diabetic patients, and then found who has AMD, dry and wet AMD among his patients, and then see if those that taking metformin was less likely to develop AMD. And he found that people with metformin documentation have significantly less likely to decrease AMD, including those with dry AMD only. He only had 122 patients with AMD and only 26 with AMD, so it's hard to say about the subtype effect. And there was no documented effect of other documented mutations. It was a relatively diverse database, but it was only a small number, only 130 patients with AMD. And then the other article that was actually a prospective cohort study from Taiwan, they started not watching patients with new diagnosis of AMD, of diabetes and metformin, and then show if they have less to develop AMD 10, 15 years later. And they found that those that were taking metformin in the prospective cohort study, retrospective cohort study, they have significantly lower risk of AMD. But like in everything in medicine, not everything is the same. So there are actually, in Britain, there's a very big cohort study from the UK that did not find any association between metformin use and the risk of AMD, but that did not analyze any subtypes either. And also a very recent study that just came out of ophthalmology, that they went to the diabetes prevention study that basically took patients to see if metformin can prevent diabetes, and they were taking either metformin or placebo or lifestyle change, and then watched them for 15 years. And then for the subgroup analysis, they did the imaging only at the end of the study. And they saw if people that were in the metformin group had less AMD compared to those that were in the lifestyle or the placebo group. And they did not find those that were in the metformin group to have less AMD. But I really want to point out, most people were discouraged. Actually, I was very personally encouraged for the study, even though it was negative. And I'll tell you 2 reasons. Number one, there was no baseline imaging. So we don't know of these people in the endpoint if they were like balanced at the beginning regarding who has advanced AMD and who does not. Number one. Number two, people were allowed metformin outside the study. So even those of the placebo or the lifestyle arm, they could still take metformin outside. And number three, as you said, the numbers are very small, but even with these very small numbers, you still see almost a trend without the baseline matching. And also the fact that the age is much younger than when we actually get late AMD. The average group when the end of the study was about mid-60s. So most of our patients actually were diagnosed through dry AMD, intermediate AMD, GA, they're much older. So you already see a trend of 0.09 with a small number. So I don't think this is definitive. It was negative. It would be nice to have an effect, but I don't think it's as discouraging as some people thought initially. Because I actually think that it's a trend without baseline match. So, and then there was a meta-analysis that was just published that looked at all the studies out there, all the data, prospective cohort, case control studies, cross-sectional studies. And when they pulled them all together, they found there was an associated reduction of odds in AMD with a pooled odds ratio of 0.8. So as you see, unfortunately, in most of the studies, including our study, all the AMD were put together, like dry and wet, and we don't have any subtype information. So we have really limited data for subtypes of AMD. So that's what formed the decreased risk of AMD subtypes. So what do we have? So Brian Butterbeek, who was actually my senior resident when I was a resident, he looked at dry AMD only cohort, and actually he did not find an effect. And actually maybe those that have current use of metformin may have higher risk to develop dry AMD. And only prior exposure was significantly associated with decreased risk of AMD. And if you look at his claim database with diabetic patients over 55 years old with 2 years of enrollment in the insurance plan. But again, the beginning point was diabetics, and then Watson for 10 years. Again, then about age of 65, which very often these patients are not still diagnosed with their AMD when they're 65. So we're trying to use a different approach. We started when people get AMD and then look back. So we don't lose an effect potentially, but the bias by the time the study is a data set, and they're not at the age that usually patients are in our practice. So we look 2 separate studies to look at new onset of new dry AMD, and also new diagnosis of new wet AMD, basically converted from dry to wet. And we have, and we matched it with the controls of a pool of 30 million for systemic comorbidities. And, we'll make sure they have 2 years of exposure and continuous drug data. So in this data, we found almost 200,000 cases of new dry AMD cases and matched controls, and almost 90,000 of new conversion of wet AMD cases. And then we look back and see if those that were taking metformin, they were more likely to be in the control group versus those that were not on metformin. So what we found was similar to what we found actually with all new AMD cases. And actually this time we went up to 2019, and we found that low to moderate dose of metformin up to 1,500 milligrams per day seemed to have decreased odds ratio developing new dry AMD or new wet AMD compared to non-users. And that effect was mostly prevalent in those without retinopathy. And it was exactly the same thing we found also for new diagnosis of wet AMD alone. Just at a point, again, for the age, the average age on the studies is about 78 to 80 years old of age, what we see in our clinic, which is much older than the average age in other cohort studies that they finish them when they're diagnosed with diabetes, and then 10 years later, we're about 65. So definitely these are a lot of limitations. This is retrospective data. This is identified claims data based on billing. We all know that we're busy. Our billing includes myself and not the most accurate always. And we don't have any ocular data in this most of these big databases. We have no labs, but you can control for other comorbidities to reflect more the systemic status. And unfortunately, because most of the studies, including ourselves, they end around 2019, that we get the SAGI-10 codes, that have really specific codes for GA. We have no information about GA, and actually we're working on that right now. And there's an ongoing prospective multicenter study by JSU or UCSF on GA patients, and hopefully we'll know soon about the results. And I think some of our speakers here are part of the study, so stay tuned. So this is big data. What about if we go an unbiased approach? So very quickly on this, if you select all the pathways involved in AMD and you plug them in a big database that has all the compounds and all the pathways that the drugs work, will metformin pop out as a potential drug. And that's why an unbiased approach. So basically that's what we did. We pulled all the genes, both from GWA studies, but also from animal studies and RNA-seq studies and human tissue studies. And we put all the genes and pathways that were involved in the AMD, and you plug them in the pharmacome, which basically is databases that have the association between the 70,000 compounds and all the genes and pathways they affect. And then there's computational mathematical models and predict compounds that could be candidates or represent these genes and pathways. And we're very happy, interestingly, to find that metformin popped out as a top 20 compounds out of 70,000 compounds for all AMD subtypes, that they will break down wet AMD only or dry AMD only, intermediate AMD, GA, and all the dry AMD. And actually for the wet AMD was #1 compound out of 70,000 compounds. This is a busy slide just to give you an idea. Top right where metformin is, the more to the right, the drug is in this very complex graphs. All the drugs, the top drugs in the top, all the genes and pathways in the bottom. The more to the right, the bigger the circle. That means the more complete is the representation of these genes with this drug category, and metformin is the most top right drug. So I'm a lab rat myself, so I always like to go back in the lab. Okay, this is great, but this is again retrospective data with limitations. Let's go back in animal models or preclinical work and see if there's a signal there as well. And let's start focusing on metformin and CNV, coronary vascularization, which is the landmark of wet AMD. And actually, there's a paper a few years ago that saw that intraperitoneal metformin inhibits ALK-mediated pathological angiogenesis, both in vitro and also the laser-induced model. When they give IP metformin, they have less AMD and seems to be through a lot of angiogenesis pathways and the ALK pathway. But for those that did the literature of metformin, most of the metformin, when you take it, it stays in your gut, and actually the systemic circulation is much lower. So actually, my lab would like to simulate more what's in the real world. So actually, we gave the metformin orally to the mice with oral gavage, so we know for sure that how much they take, and that's equivalent about 1,500 grams a day, 300 milligrams per kilogram for the mice. And we saw that like pre-treating mice orally with metformin for 1 week prior to the laser induction, or even 2 weeks before, and continue until we enucleate the eyes, it suppressed significantly the CNV formation and also microglia filtration, both within the lesion and also in the peri-lesion macroglia, and the macroglia less activated forms. And then we sequenced over 20,000 genes because we're just to see what pathways are really involved. And we analyzed 20,000 genes from the RP and the choroid from these mice. And we saw that metformin suppressed a lot of VGF and inflammation-related pathways, ECAM1, VGF type pathway, among top others, PTGF, and also in transcription analysis, computational, a lot of key transcription factors for angiogenesis and inflammation. But the holy grail, we all agree, is neuroprotection. So what's out there for metformin and neuroprotection? And actually, in vitro, we start with in vitro first. In different models that they basically take human RP cells and they kind of reduce oxidative stress with like H2O2. They found that the cells, when they're treated with metformin, they have to have better cell survival, decrease reactive oxidase, and it seems to be through the phagosome activation and improving mitochondrial function, and MP kinase, as we all know, metformin is an MP kinase activator, seems to be key for this action. But you know, in vitro, almost everything works, so you need to move to more in vivo models. And actually, when they gave sodium butyrate to the mice, this destroys the RP cells. Like you give an injection to the mice, and there's no RP like a day later. So when the mice were treated with metformin, they had like a mass preservation of the RP cell architecture and RP cell occluding and the RP cells. So in vitro and some in vivo models, and I think the key paper came out of from Florida group is a PNAS paper that basically used several models of light-induced retinal degeneration and also RPE toxic degeneration, and they saw that the mice were like subcutaneous metformin. They had significant improvement in ERG function and also cell loss and apoptosis, and it was through MP kinase, and they seem to regulate a lot of the like mitochondrial enzymes that we know they're key for regulation of oxidative stress. So if metformin is neuroprotective, what about retinal dystrophies? I'm surprised that the field of and I was hoping Bob Hyde is here, so maybe we can talk about it. But there's no clinical data out there. Nobody has looked in people that have retinal dystrophies, or they're diabetic, and they get metformin, they have better outcomes. But there's a very good paper from RD model, which kind of like simulates the retinitis pigmentosa, that when the mice got metformin, they had really improvement of the ERG, maintain ERG and cell function and apoptosis. And they did RNA-seq, and they found different crystallines and other like apoptosis-related factors for potential survival. And interesting though, someone would think, okay, since it's not mutation, it's neuroprotection, the mutation doesn't matter. But there is one paper out that says we should potentially be careful about this, because when they did the mouse model with specific mutation for rhodopsin, which is a very common mutation, even though the metformin improved the trafficking of the rhodopsin through the endiculum reticulum, they actually found more toxicity because it was accumulated and it caused more toxicity. So we need to be a little more careful because maybe not all mutations are the same effect, even though theoretically you think this independent of the mutation effect, which makes it very exciting. And actually, there is an ongoing trial right now that people with Stargardt to see if those that start taking metformin have better outcomes and ERG. And I think it's very exciting to see the results of this study. So metformin affects inflammation, affects VEGF pathways and angiogenesis. What about retinovascular disease? And unfortunately, there's not much in there. And I think it's understandable because there's so many comorbidities with diabetes. It's very hard to really say if it's a drug or is a diabetes effect when you look about the burden of diabetes. In the same study that I talked earlier about AMD, the diabetes prevention program, they did not found any less incidence of any complication of diabetes, but there was no powered enough for complications. This was a study to see if we could prevent diabetes itself, not the complications that come months later. So again, it's a potential initiation age problem. But when they look at in a cohort study from basically see if patients on metformin get less diabetic retinopathy, there was low risk of diabetic, non-proliferative diabetic retinopathy and side-threaded diabetic retinopathy among metformin users versus non-metformin users. And it seems to be a dose-dependent relationship. But interesting, similar with what we found, that patients already had diabetic retinopathy. It was not protective anymore. So similar pattern that we saw in AMD studies as well. So it makes it tells you that when the damage, whether it is already damaged, metformin may not be enough to stop the progression anymore. And similar effect from a US study that metformin users have less sight-threatening severe NPDR among diabetic patients. Only one study could find for vein occlusion from Taiwan that maybe metformin users are diabetics, they have less vein occlusion, but I think it's not enough data to really say much. And again, glaucoma is not a retinal disease, but I think in a way it is. So just a very interesting out there because, you know, many of our patients have glaucoma as well. So can you kill two birds with one stone? There's a very good two big data studies that metformin is associated with decreased risk of vision loss from glaucoma. And it seems to be dose-dependent effect. And in diabetic patients, again, separate two JAMA ophthalmology papers that metformin associated with low risk of open-angle glaucoma in diabetic patients. But another small study did not find the effect. So there is an ongoing trial for that, so stay tuned for that as well. There is a prospective study about visual function and metformin in glaucoma patients going on. So hopefully we have some interesting data. So in conclusion, metformin AMD, there are some big data showing that there may be decreased risk of AMD in some subtypes. There is an overlap between the pathobiology of AMD and metformin pathways of action. There is a signal that in a computational way, it may overlap with a lot of genes involved in AMD and some preclinical data that can be anti-angiogenic and neuroprotective. But definitely we need to consider prospective trials, both for the conversion to the wet AMD and maybe intermediate dry AMD with some of the new biomarkers that Dr. Rosenfeld mentioned today. And hopefully that J. Stewart study will show a signal for the GA. Few data for glaucoma, DR, and IRDS. So hopefully we know more, but maybe some role there as well. So I want to thank my team, my funding, and thank you for your attention.
Thank you, Dimitra. That was wonderful. Really comprehensive overview of metformin. Sounds like the new wonder drug. Maybe we should all be on metformin. How was it actually even first thought of? I'm just curious, like why would they even think that it would result in a lower rate of AMD? Like why was it even looked at in the first place?
I think initially it started because there's a lot of literature in the aging world that the people on metformin have less age-related diseases, but nobody's thinking about the eyes in the outside of the eyes world. So at least for me, when I start looking into this, my initial observation is that I notice a lot of my diabetic patients, they don't have AMD. And I'm like, oh, maybe it's something about the drugs. And then I read the literature that is out there. But I think it's just, it seems to be a more broad effect than initially with diabetes.
Yeah, and I was also impressed by that drug analysis where it popped out, you know, and I think I saw Ross reactive oxygen species. And it was pointing to there, so that it says an antioxidant effect of some sort. Yeah, yes. Anita, Dr. Agarwal.
The question is, all these patients are on metformin are obviously much younger than the aging patients. So does that can they correct for the age to see really that this helped in aging?
So I think that's the problem with the cohort study. Yeah, so that's the problem with the cohort studies because the cohort studies are focusing when they're diagnosed with diabetes, which are used in the 50s, then they look for 10 years forward, but this is not really when our patients are. So in our studies, our patients were like 80 years old. So we started reverse. So we started people when they get AMD and look back to see if those that were already on metformin, they were more in the control group rather than the AMD group. So
Can't really say. that
no, we
there's a
need the
connection.
prospective study.
A
I
lot
don't think
more.
it conclude.
And
any
there's so many
conclusion.
other factors that may be playing a role in those.
Yeah, you don't know. We're actually looking in our non-diabetic cohort right now. So there's a lot of other indications for metformin besides diabetes. So they give it for obesity, they give it for polycystic ovaries, they give it for pre-diabetes, which are not really diabetic, and they have less comorbidities. So actually, we're analyzing non-diabetic cohort alone right now to see if the signal is still there and maybe even stronger. That'll give us more indication.
So you said you're taking it, right? So what's the
I haven't
dose?
taken it yet. I'm thinking about it. I'm really contemplating.
Hey, what is the dose? What dose should we be recommending if you start a study, right? What is the dose you would?
I think the data, the animal data and the human data now, I think it needs to be at least a gram a day. In our studies, the 2-gram dose was not protective, but I think that can be if these patients are really bad diabetics and they need so high doses that it's not a metformin effect. So that's why I'm interested to see the non-diabetic data that we're analyzing now. But in the animal data, it's about 15 grams to 2 grams. So I will do a study 1 gram to 2 grams. If I start taking, I will start low to see if I tolerate, I will go up.
Supposedly it's very well tolerated, right?
Yeah.
Metformin, because I know we're part of that metformin study. I think you're in it too, right? But I think so far the data is not showing any positive
I know.
results yet.
I think it was too late.
It's
If you ask my
probably-
opinion, like I think we need to target intermediate AMD patients with these early biomarkers. If the GA is already this big, I don't think it's going to do anything. That's my opinion. So I don't think it's going to work.
So-
But I don't think it doesn't work in general. I think we just need to start early.
So I think you need to talk to Dr. Rosenfeld. I don't know if Phil's still on, and maybe those people with that hyper transmission defect.
I was
Start
thinking,
on there,
actually
right?
put a note, Dr. Rosenfeld about including this biomarker for metformin trial.
Okay. All right. And with that, we'll go to lunch and then come back here in about 45 minutes. Thanks. Thanks all. So our next speaker, please let me know if you can hear online. We good? Okay, so our next speaker will be Dr. Paul Chan, who will be giving his talk virtually. Dr. Chan is the John H. Panton Professor of Ophthalmology and head of our Department of Ophthalmology. He's also director of the Pediatric Retina and ROP Service and co-director of the Vitreoretinal Fellowship. Paul has an interest in global ophthalmology with initiatives focused on surgical and educational training for the diagnosis and management of adult and pediatric retinal disease. He runs multidisciplinary groups of investigators to establish clinical, teaching, and research collaborations in Asia, Latin America, and Africa, and also serves on the Global One Advisory Board. He is also the recipient of the ASRS Senior Honor Award, the Achievement Award, and 2 Secretariat Awards by the Academy. And very apropos, Paul is out of town doing his global ophthalmology work. I believe he's at the PAAO, so he pre-recorded his talk. He will be speaking today on the new international classification of ROP.
Hi, good afternoon. First, I want to thank Dr. Lim for inviting me here to speak to you today. And I'm sorry that I can't be there in person. I'm at another meeting in Buenos Aires, Argentina. I'm going to discuss with you the updated international classification for ROP. This is the 3rd edition. I was the chair of the ROP committee for the International Pediatric Ophthalmology and Strabismus Council, which supported this effort. Here are my financial disclosures. Most relevant to this is that this project received support from the Knights Templar Eye Foundation, and as I mentioned, from IPOS, which is the International Pediatric Ophthalmology and Strabismus Council. I want to acknowledge the incredible group of people that came together from across the world, some converging onto Chicago, and especially Michael Chang, who led these efforts. Amazing group of people, ROP experts, pediatric retina, pediatric ophthalmology, very passionate about this issue, and it was a terrific project. So when we look at the international classification for ROP, going back to 1984 and then to 2021, which is when this update was published, there's been a significant evolution, right? So first, looking at the zone, stage, and plus disease categories. And again, when thinking about why was this done, well, looking back at cryo-ROP and creating good definitions for this condition. And then in 1987, classifying retinal attachments, and then even further in 2005. So when you look at 2005, they defined aggressive posterior ROP, pre-plus disease, and also defining some areas around regression. So it's been well 18 years now, but 16 years between 2005 and 2021 in updating the classification system for ROP. So why now? Why revisit it? And I think for those of us who manage this condition, we understand that anti-VEGF and imaging have really changed a lot of what we do. When looking back at anti-VEGF treatment and when that was first published around 2005, 2006, we've learned a lot. And almost 20 years later, there's still a lot to learn in that space. But again, our treatment paradigms have changed. Imaging, we have a lot more imaging at our disposal. Fluorescein angiography, and now you're seeing new technologies such as OCT and OCTA in the pediatric retina space. But again, learning a lot about ROP through novel imaging techniques. And again, with anti-VEGF, understanding that this has changed how we understand the disease post-treatment, especially. We've also learned that there can be discrepancies in the diagnosis of disease. So even though we have these set definitions of APROP, there can be inconsistencies in diagnosis even among experts. In addition, plus disease, we know that there can be variability in diagnosis between experts, not just experts, but also trainees, right? So there's still a lot to learn, and it's very subjective. Reactivation and recurrence is a significant issue. We've treated mostly with laser in the past, but again, as I mentioned with anti-VEGF, seeing more issues around reactivation, recurrence of disease, persisting avascular retina. So these are the issues at hand: APROP, reactivation, recurrence, plus disease. And so we came together. Again, just some background. This committee was chaired by Michael Chang, who's now the director of the National Eye Institute. At the time of when this started, he was at KC Eye Institute in Oregon. 34 ophthalmologists coming from 6 continents around the world. And you can see here the breakdown, both pediatric retina and also pediatric ophthalmology. So here are the highlights. We got rid of the P in APROP. So APROP was originally described as aggressive posterior ROP, and we all know that it affects the smallest and most vulnerable infants, but we do recognize that this can have some variability in its phenotype, especially in areas with resource limitations. So we are now calling it aggressive ROP. It's not defined strictly by the location of the disease, but more by the tempo and also by the appearance of the vascular abnormalities. Reactivation, this has been a hot topic. As we've looked in the past around reactivation after anti-VEGF treatment, when you look at the literature, there can be a lot of variability in the definition depending on who's publishing. So we wanted to create some uniformity around what reactivation is. I think this is especially important for clinical trials. So if you have reactivation, you have recurrent vascular dilation or tortuosity, extraretinal vessels. You can have extraretinal vessels that may appear as a fibrovascular ridge. Stage 4, Stage 5 classification was also updated. Stage 4 is the same, but Stage 5 is now categorized into 3, Stage 5A, 5B, and 5C. This can have some relevance as we look at surgical outcomes. We all know that Stage 5 detachments are difficult. The prognosis is poor, but if we can get some understanding as to which cases might have better outcomes, we may be able to treat these children more effectively. Persistent avascular retina vascular anomalies. We are learning now that especially in the anti-VEGF era, that we can get areas of persistent avascular retina, as you can see here in these pictures in the peripheral retina. We also know that this can happen in children who've never had treatment and who have regressed disease. So Pete Campbell has shown this, also Wei-Chi Wu, who were part of this group, have shown this in different populations. But again, how do we understand this better and identifying this better, especially with fluorescein angiography? And then what are the implications in the long term? And we all know that ROP is a lifelong condition and can have changes in the peripheral retina, even in older children who have regressed disease. Describing posterior zone 2 and also the notch were very important, understanding that these can potentially have implications for outcomes. You can see here in the picture in the top right that the notch is described, and what we would say is that if there is a notch, that it is in the zone of disease secondary to the notch. So for example, zone 1 secondary to the notch, even though most of the condition is in zone 2. And thanks to Lauren Kolonowski, who works with us here at UIC, for this schematic, which is in the published paper, indicating intermediate zone 2 disease or posterior zone 2. Again, why these definitions? It's not to recommend treatment, but it's really to help us create the definitions so that we can have clinical trials and studies that use the same language. Plus disease historically have been based off of the standard photo, but we also now have learned a lot about plus disease over the past 20 years, especially with quantitative analysis, and also understanding that it's a spectrum of disease. So in the published paper, you'll see the images that represent different degrees of plus disease or plusness. Again, understanding that it's not just plus or no plus, but also a spectrum of disease that is important to understand. In summary, these are the updates. Again, changing AP-ROP to AROP, so aggressive ROP, which means that we're not just identifying this by the location of disease, better defining regression and also reactivation, which is very important, especially after anti-VEGF treatment, further categorizing stage 5 disease into A, B, and C, persisting avascular retina and vascular anomalies, recognizing this and also having better definitions around it. Intermediate zone, posterior zone 2, and the notch were described, and also, as I mentioned, plus disease being a spectrum of disease as opposed to just plus or no plus. So you can use this QR code if you're interested in downloading the paper. It's open access in ophthalmology. And again, thank you, Dr. Lim. Thanks for everything, and I appreciate the opportunity to speak to you all today about the International Classification for ROP 3rd edition. And again, sorry I can't be there with all of you.
Thanks so much, Paul, remotely in Argentina. So our next speaker will be Dr. Anita Agarwal, who is a great friend, and it's my pleasure to introduce her today. Dr. Agarwal is yet another one of our visiting professors who was initially educated outside of the United States, did her ophthalmology, graduated with honors, also in medical school, and then came to the United States, where she initially was a research fellow, and then did her training with luminaries. She worked with Don Gass as well, and then was also at West Virginia, eventually at Vanderbilt, worked with Paul Sternberg, and became faculty there. And she is currently a vitreoretinal consultant at West Coast Retina in San Francisco, and remains an adjunct professor of ophthalmology at the Vanderbilt Eye Institute in Nashville. So Anita, as you might know, is the author of the Gass Atlas, the 5th edition, and she's co-authored numerous landmark research papers, particularly on medical retina as well as genetics of macular degeneration. And for this, she has received numerous named lectureships, including the COS Krill Lecture in Chicago, the 2014 Gass Medalist from the Macular Society, and she is also a Senior Honor and Secretariat Awardee from the Academy, an ASRS Honor Awardee, and the 2015 Optic UK Lecture Medal from the Royal College of Ophthalmologists. So you might see Anita frequently at meetings where she is moderating the interesting cases because she loves interesting medical retina, unusual diseases. So you can email her all of your unknowns. And so without further ado, I introduce to you Dr. Anita Agarwal, who will speak on recognizing the less common macular dystrophies. Anita.
Thank you, Jenny, for that very generous introduction. I'm happy to be here amongst my friends. I thought I'd see everybody else, Paul and Bill, but I guess they're not here physically, but they're somewhere else at a distance. I see Matt, and of course your friends Amani and everybody at Northwestern and Deeba. This moves. It should be a little bit. Okay. How do I make this move? Okay, left. Okay. So today's talk will be on some of the macular dystrophies. There's so much, and there's a lot more that we don't know about, and I don't know a lot about it either. But these days with genetic testing, we're learning a few more things about these. And the idea today is to identify the disorder if you can. And some of them are very common, but there's certain newer imaging features and genetic features that help us. And occasionally there's more than what meets the eye by imaging and genetics. So sometimes we think it's something and it ends up being something else. So let's look at that. This is a very classic appearance of Best dystrophy. The egg yolk appearance was a young child. The important thing is to use autofluorescence imaging. So you can see that the lesion is extremely hyperautofluorescent. And that kind of helps us clue in. Of course, you look at the fellow eye and you find out a family history. The most important thing is that these patients with Best disease have a space under this vitelliform material we call it the Best space. And this space is important because it'll tell us a little bit about some of the later stages of Best disease when you may not have the classic appearance. And if you want to figure out, is this AMD or is this Best disease? Because a lot of patients who have Best disease have pretty good vision. And they may present in their later age, and you may think they have AMD with subretinal fluid. So it's important to look at some of these OCT images. And these are the classic stages of Best disease, the hypopia and the scrambled egg and the atrophic. And the big thing to know is that Best disease patients can get CNVM. And especially if they get CNVM in their later life, you may think that it's AMD. So it's important to kind of keep that in mind and ask about family history. Of course, there's a family history of AMD, but the question to ask is the family history of macular disease in younger relatives in their 40s and 30s. If they say that's the case, then it's Best disease and unlikely to be AMD. And three main features about Best disease is the fact that there's this hyperautofluorescent material, there's the vitelliform space, and the EOG is reduced because the gene is at the and the basolateral basement membrane of the RPE cells where the exchange occurs, and that's where the EOG measurements occur. And this is an example of a later stage of Best disease. As I mentioned, that space remains. So this is not subretinal fluid due to CNVM, but persistence of the Best space. Another example of that, this patient actually is a mother and daughter that I've followed for many, many years. And this is the mother. The right eye developed a CNVM previously and lost the Best space from fibrosis, but the left eye has the persistent space. And the hyperautofluorescence around the edge is useful. And of course, because we had seen both the mother and the daughter, we knew they had Best disease, and subsequently we could do gene testing and confirm it. Here's another type of Best disease that is a little bit atypical, multifocal Best. These are not very common. And now with gene testing, it's quite easy to make the diagnosis. As I mentioned earlier, the VMD2 gene defect, occurs at the pigment epithelial level, so it affects transport and affects the origination of the hyperreflective material. But in addition, in 2008 and onwards, we found out that the VMD2 gene is responsible for certain other diseases, which are kind of syndromic. One of them is autosomal dominant microcorneal rod cone dystrophy staphylomas syndrome. These patients have a defect in the VMD2 gene, and this is autosomal dominant. And the other one was first described here in Chicago, autosomal dominant vitreoretinal choroidopathy or ADWRK. And I'll show you an example quickly about that. The only one that is unusual here is autosomal recessive dystrophinopathy, where it's a recessive disease. It's a much more severe mutation because it's a recessive condition, and we'll talk about that in just a second. Also to remember that the VMD2 gene can also cause pattern dystrophy. Here's an example of ad work. Most of these patients are asymptomatic. They don't have night blindness, but they do have this peripheral, very symmetric ring of peripheral pigmentary changes. Again, a family history is useful. This is ARB, and many a times we will actually see patients with ARB, and we may not think about the diagnosis, but with autofluorescence imaging, it's really useful because you get this multifocal hyperautofluorescent spots. These patients can present as young kids with a narrow angle, hyperopia, the pressures may be elevated, and they have this macular changes, and it's a recessive disease, so it's a little bit more common in countries where there's some amount of consanguinity. And in fact, a lot of patients were presented from India with autosomal recessive dystrophinopathy. And to think about the fact that they have short eye and elevated pressures. Here's the original cases that were when this was first described in 2008. This is courtesy of Camille Boone, this was part of his PhD thesis, and they have this hyperautofluorescent material. They have intraretinal fluid, intraretinal cystic spaces, and thinning of the retina. So the vision goes down, usually in the teenage or 20 years of life, sometimes later on, and one has to consider ARB in these patients. And as I mentioned, it's a null mutation, so the mutation is much more severe than having an abnormal protein or an abnormal product. Unlike the previous autosomal dominant disease, this is another autosomal macular dystrophy. These patients have two phenotypes. One phenotype is a compact drusen type of phenotype, where patients are completely asymptomatic. The second one is a osteostaphylloma type phenotype. In the past, in the textbooks, we had a third phenotype, which was considered the subretinal fibrosis, but I believe that subretinal fibrosis is just a secondary phenomenon from a CNVM. All of these patients, lesions, conditions affect the pigment epithelium, so they can develop CNVM, and you can have that as a complication. So this patient is a young girl who's 13 years old, was seen for a refractive error and was found to have this. We examined the father's sister, and the patient's brother, all of whom had similar findings. You see that the father and the daughter almost have the same size of drusen. The father is in his 40s and the girl was about 13. So this tells you a little bit, at that time we didn't realize this, and it was not published, that many of these patients don't have progression of their lesions. And we discovered that when we were following this family. Can somebody mute? Who was mute? Has their, can you please mute? I think one of the Zoom attendees has they're unmuted, please. Okay, all right. So when we were looking at this mother and daughter who came in with the staphyloma-like picture, the mother told me that she was told she had Best disease, but then she brought her daughter, who was about 18, who's going off to college. And I looked at the daughter and I said, this doesn't look like Best disease. This looks like North Carolina macular dystrophy. I said, may I look at your eyes? And I looked at the mother's eyes. And it's amazing. The girl was about 20/50, but the mother was 20/40. And the mother was at least 22 or 23 years older than the young girl. And I was surprised as to how she could manage to reach 20/40 in both eyes with this kind of staphylomatous lesion. So I sent her off on an OCT to look to see if there was anything that would give me a clue. I was wanting to see how many photoreceptors she had that she could manage to reach 20/40. And to my surprise, her macula was completely staphylomatous and there were no photoreceptors there. So that's when it dawned on me that this is probably an in utero defect, and it's not, they have eccentric fixation. It's not something that's progressive. And so when we got the OCTs back, we looked at it and we discovered that they had these intrachoroidal cavitations. And that was the first time it was recognized, though there was actually a paper from I think Dr. Maud Goldberg's group that had this finding in their series of 5 or 7 patients. Anyway, long story short, we realized that these patients have this defect that's in utero and it doesn't progress. So we think that the RPE, the Brooks membrane, the sclera, all of them form anywhere between 4 to 12 weeks of life. And during that time, the defect manifests in the pigment epithelium. We think that all these structures are affected. If the defect occurs a little bit later, they probably get the drusen-like phenotype, but if it occurs early on, they probably get the staphyloma type phenotype. And as a result of that, these intrachoroidal cavitations are formed because the vitreous probably got trapped as the eye was forming, the sclera on the choroid was forming. similar to what you see in high myopes. That's what was already described in in the late 80s or early 90s from Europe, from France, about having these intracortical cavitations in myopia. So we think that North Carolina macular dystrophy is a congenital defect, occurs in utero, stable and mostly non-progressive unless you develop choroidal neovascularization. And Ken Small, when he was a fellow and he looked at all these patients in North Carolina, he went and visited all these patients, examined them. He actually made a note in his paper that many of them looked like the drawings that were done in the 50s and 60s when these patients were seen with North Carolina macular dystrophy. So we do know that's a non-progressive disease. Unlike those ones, this is a mitochondrial disease. And I put this picture so that this is something that we should all recognize. These patients have these islands of geographic atrophy that spares this foveal center. And as time goes on, the islands expand and it looks like AMD, but this is something that can easily be mistaken for AMD if you didn't think about it. And the key thing is to find these little small dots that are there, if you can find those, but of course they can look like pigment changes in AMD. They don't have drusen, and it depends on the age that you got this patient. This gentleman came in for type 2 diabetes exams every year, and he had these scars, and he was watched as scars with APM-PP. Till I asked him, how's your hearing? He said, oh, I've been wearing a hearing aid since I was 45. And then I realized he had my MIDD with dystrophy. And this was his sister who was older to him, who's followed at WashU. And I asked Dr. Grant, if he wouldn't mind looking at his sister's pictures. He was kind enough to send me copies of the pictures. So the sister has these pigment dots. So these are the early signs of mitochondrial methylated diabetes and deafness with the A3243G mutation. But if you just saw this, you can easily pass it off as AMD. And similar to AMD, they have geographic atrophy, and the rest of the remaining ellipsoid line looks intact till it eventually gets affected. Autofluorescence, as I mentioned, is useful because autofluorescence picks up those little autofluorescent pigment, RPE, things that have probably migrated out of the from the Brooks membrane into the outer retina. Unlike those disease, unlike the previous one, which is a mitochondrial disease, this is another autosomal dominant disease, which again can be mistaken for AMD. But if you see the classic nodular round lesions, then you can tell yourself that this is not AMD, but this is Melotia levantinese or Doyne's honeycomb dystrophy. The one thing about Melotia levantinese is this radial arrangement of the drusen temporally, and they're also supposed to have drusen nasally, like you can see in this example. Here. Okay, I've got it there. So this radial peripheral arrangement is very typical of Melotia levantinese, and if you see that, you won't mistake it. And it's also mentioned that they have drusen nasal to the disc, and these two sisters were positive for the gene. Oh, I was going backwards now. But then this is the older sister. She's older to the previous one. They have a strong family history of, we're told they had macular degeneration, and she has good vision. And both of them have the same mutation, but the sister has a very mild phenotype. And if you saw the sister, you would never make the diagnosis of Melotia levantinese. So it's important to sometimes look at more than one family member. These days with genetic testing is really very easy. So here's a OCT of a patient with Melotia, which the little excrescences on the pigment epithelium that you see on OCT correspond to what you see in histology with these sort of basement membrane excrescences. And we know that the defect is in the extracellular matrix protein, EFEMP1. And unlike all of those ones, pattern dystrophy is sort of a little collection of cases that are put in this condition called pattern dystrophies. Some of them are specific for certain genetic defects, and many of them probably could be acquired things, non-specific things, could be old CSR, because CSR also causes the same yellow flecks. And big overlap is between Stargardt's disease and pattern dystrophy. If patients with Stargardt disease or patients who had yellow flecks had good vision, we call them pattern dystrophy. If they have poor vision, we call them Stargardt disease. But now we know that many patients with ABCA4 mutations have extremely good vision. So genetic testing is useful in some of these patients. And we'll look at some examples. Here's a very classic pattern dystrophy, a solitary vitelliform lesion with central pigmentation in the middle. If you do a fluorescein angiogram, you see that halo of hyperfluorescence around the central pigment. And here's a little rarer version of it, the reticular form and the fundus flavi maculatus form. The one thing to know is that the fluorescein angiogram does not show a dark choroid that you see in Stargardt disease. This is from Don Gass's collection. He called them all different names. He called them hobby horse, butterfly, torpedo, and a rooster. He called this one a rooster. But we now have genetic testing for this, and peripheral RDS is probably the most common mutation that you see in patients with pattern dystrophy. And I'll show you some examples of the variability even within the family with the same mutation in the peripheral RDS patients. This is Stargardt's up here, and this is pattern dystrophy. So the only difference is that, Don Gass used to look at these patients those days we didn't have genetic testing. He would look for choroidal vessels in the macula. If he saw choroidal vessels, he'd say this is pattern dystrophy. And if he saw a vermilion background without where the choroidal vessels were masked, he'd think that was Stargardt's disease. So we'd send them off a fluorescein angiogram, and then we'll look for the dark choroid. And this is autofluorescence, which, initially when autofluorescence came about, people were talking that you could differentiate one from the other by this peripapillary sparing. But I can show you that these two examples show you that there is peripapillary sparing in both pattern dystrophy and Stargardt disease. So you can't use that. You can use a fluorescein angiogram. This does not have a dark choroid, so you know this is pattern dystrophy. However, this patient who has genetically proven pattern dystrophy with RDS PRPH2 mutation does have a dark choroid. So for a Caucasian, she has a dark choroid. It looks relatively less dark because of the fact that she has so many flecks that are hyperfluorescent. And she has 20/20 vision in this eye and has a sister with this. Both of them are positive for the mutation. And this gentleman came to me, and he looks like he has ABCA4 Stargardt disease, but he was 20/20 in the right eye, and he could be refracted to 20/40 with the schisis with a +4 refractive correction. And I was so sure that he has ABCA4 disease. This is before we could do genetic testing. And he has a dark choroid for a Caucasian. Then he tells me his mother has some eye disease. So the mother flies down from New Mexico, and she comes and she has this thing. And I looked at the gas atlas. This was before we had genetic testing. And he had two patients of this, and he had called them non-X-linked choroideremia because they were both in women. And that was in Gass's atlas, 4th edition. So that's what I called her. And then we sent off both their blood, the son, the mother, and the mother's father came down from Chicago to IG, and it took them 3.5 years. After 3.5 years, they gave me the results that this was PRPH2. So both the mother and the son have the same genetic defect. The son has a dark choroid, mother has an extremely visible choroid. She has extensive atrophy. And the son has a thick choroid, the mother has a thin choroid. She also has these outer retinal tubules, so the same mutation. And surprisingly, the grandfather who had something that looked like this did not have the mutation. So I don't know if they couldn't get it, but this genetic testing was done about 2012. So I'm not sure if they couldn't get it from the grandfather, but all the three people's blood samples were sent to IG. So in summary, peripheral RDS can have variable phenotypes, even in the same family. Many of them have good central vision, but sometimes central vision is affected. And they resemble Stargardt's in something. There's something between Stargardt's and peripheral RDS, so we have to figure it out. Unlike that, this is a patient who I saw this patient with Don Gass. Actually, I saw the next patient with Don Gass. This I saw with Paul Sternberg. So the lady she was about 48 years of age. She'd already lost vision in her left eye, and she was Paul's patient. She had this CNVM that was extrafoveal. At that time, we did not have anti-VEGF injections, so she got laser. This was, I think, in about 2005. And she came in about a year later with a new CNVM when I saw her. But for my surprise, I noted that she had extensive pseudodrusen going all the way to the periphery. She had a strong family history of AMD. Many of them had lost vision from disciform scars. And at that time, I didn't think much about it. And I went back and looked at this patient that Don Gass and I followed for a long time. In fact, I even did PDT on this lady. She was 48 years of age. She had lost vision in the fellow eye 10 years prior. And at that time, we only had PDT and laser. So we tried all of that on her. note that she also has extensive drusen going all the way. And finally, I think in about 2013 or 14, I decided that these two may have source B. So I sent off their blood for genetic testing, and they both came back with the same mutation, the two different families, and they had the same mutation with TIM3 and ended up having source Bs. So we see patients with AMD that looks a little bit too much for their age. We should think about looking for TIM3. So the OCT shows that the drusen are quite similar to what you see in AMD. And so always think about TIM3 and source B. They're in our midst, and sometimes we don't pay attention to the age, so it's important to look at that. And most surprisingly was this 53-year-old gentleman who I saw in 2018, and he was told he had RP, or he told he had AMD, and there's a family history of early onset, a little bit of nyctalopia. He had nyctalopia in his 50s, and this is what he looked like. So I thought he had I didn't think he had late onset retinomacular dystrophy because all his lesions were in the macula. This was his right eye, this was his left eye, and he did complain of difficulty with night vision. Lower scene was okay, everything was limited mostly to the posterior pole, nothing much in the periphery. GA again corresponds to those atrophic changes. Here's his OCT. You can pass this off as AMD, GA, but this kind of this irregular RPE with some stuff under the RPE. And it took, again, 2 years to get results from iGene. This was sent in 2018 when we did not have my retina tracker. And after 2 years, I got the results back, and he has a mutation in the TIM3. So he has a type of source B that only presented with GA and not with neovascularization. Of course, the rest of his family who are affected are in Argentina, so I don't have a chance to examine them. But he did, even though they said it's a VUS, because it's a gain of cysteine, it is a pathogenic mutation that can cause abnormal accumulation of material under the pigment epithelium. So sometimes we'll be surprised, and these surprises kind of teach us a lot of things. So this was a young lady who's in her 40s who actually was ticketed twice because they said she ran through the red light, and she lost vision over a year. She went from 20/20 to about 2100. Fundus is normal, fluorescein angiogram is normal. Her OCT shows central scotoma, so she was not faking. Her color vision was not bad. I think she read 9 out of 11, but the key finding was really in her OCT. If you look at the OCT, the central foveal OCT ellipsoid line is all fragmented. This is the right eye. Here's the left eye. The left eye had a little bit of dragging of her peripheral vessels, so her retina is a little bit dragged, but that's why the contour is a little bit unusual. But she does have this fragmented ellipsoid line in the center. But if you look at the ellipsoid line above, right, the cut on the superior part of the macula, cut close to the edge of the inferior part of the macula, the ellipsoid line is pretty well and preserved. So this patient has occult macular dystrophy. At that time, we could not do genetic testing, but now we confirm that she does have a defect in the RP1L1 gene. Their full field ERGs are normal, but they have an abnormal multifocal ERG. So it's really important to kind of establish the diagnosis by getting a multifocal ERG. And if you suspect this based on the OCT findings, you can definitely get genetic testing, which is available. And this was first described by Miyake. He found the mutation, the RP1L1 gene. And as I said, it was difficult to get this testing done when I first saw that patient, but now it's pretty straightforward and you can get gene testing. So in summary, we reviewed features of some uncommon macular dystrophies. Many of them have overlapping features with other dystrophies or degenerative diseases, and variable phenotypes within the same family can kind of confuse us. Gene testing is teaching us much, but still a lot of unanswered questions, and there are a lot of new questions that we have to figure out. Thank you.
Thanks so much, Anita. That was fantastic. I think I have to watch it again. So I'm glad we're recording it. I have a question. Where do you get your genetic testing, and how often do you test?
When I was at Vanderbilt, before we had my retina tracker and we didn't have the panel testing, all the testing that I did was targeted testing. So I kind of my best guess as to what it was, and I would send for it. The only time that I had a little overlap was between ABCA4 and PRPH2. So we sent it to Baylor College of Medicine those days. The other one is GeneDx. But those were private places that we had to go through the insurance. Luckily, Vanderbilt had one person dedicated to send, it's called the send out lab. So I'd just call her and tell her, but she'd get me all these forms I had to fill up. But now with My Retina Tracker, it's straightforward. I mean, we are part of it, so it's free. The only disadvantage is that they will only test one person in the family. If you have autosomal dominant disease and you want a sibling or a parent or somebody to be tested, they have to pay for it, which we have a little fund. So we pay from that fund, $150. They only charge $150 for just targeted testing for the same mutation. If you have one family member with that mutation, they'll just charge $150. Otherwise, My Retina Tracker does for free. Now I just learned that Invitae, which is part of Spark Therapeutics, as combined with My Retina Tracker through Foundation Fighting Blindness. So you could, again, Invitae would do more than one, but they test a little different panels. Sometimes you don't get a result in one. I have done both. I've gotten the other one. Occasionally, I don't get in either because the panels only do certain mutations and certain genes. If like fever, they have fever genes in the Invitae panel, but most of the time it doesn't come back positive. If you really suspect it, you have to send it through the insurance to GeneDx and then came back positive. So sometimes you have to go beyond it. But I tell you, My Retina Tracker, I don't know how long they'll keep it for free. It has opened up our eyes and it's taught us a lot of new things, and hopefully it'll be benefit to lots of patients.
Any other questions? Dr. Skandra, can you give her the mic, please?
Yeah, I think if there's any doubt, if they have, that's why it's important to not just put them all in. In fact, at Johanna Seddon at, when I was doing AMD genetics, Johanna was doing her AMD genetics at in Boston, and Johanna Daniela Ferreira presented data from her study. How many of them were actually PRPH2? There were a significant number, not a lot, but of course, significant ones that were put into AMD genetics, but they were all pattern dystrophy. So I think if there's doubt, you have to. And especially, late presentation of Best disease. I have seen an older lady who came in when, those days we couldn't do testing. And I asked her, she said, oh yeah, a lot of people in my family, but I said, what ages were there? Oh, they were in the 20s, 30s, 40s. So that's the clue. So it's important. Whoever came with them, her son was with her. I told him, can I look in your eye? So it's always important to look at the family members who came with them. And many of these, all those North Carolina macular dystrophies, the first time I made the diagnosis was the mother was there. And I looked at the mother. The mother was complaining that they were divorced, that it came from the father's side. He has poor vision because he wore glasses. And I looked in her eye and I saw she had the same small drusen. So now I had to tell her that actually she has it. Anyway, so it's, I think it's important to kind of ask more than, I think, especially if you're putting them in clinical trials. Right. Okay, thank you.
Fantastic. Thanks again, Anita. And I will put a plug in for my book on case studies in medical retina. I know, Anita, you have some cases in there, Amani and some other people in the room as well. So be on the lookout for that book that hopefully should come out later this month. I'm editing it with Bill Mueller. It's a brand new book. Okay, next speaker will be Dr. Amani Fawzi. So Amani will be talking about macular telangiectasia diagnosis and management. Please come back to the podium, Amani.
Thank you, Jenny. And I thought this talk title was topical because of the updates to the MACTEL trial. So I thought you invited me to talk about the trial. So my first half of the talk will be about the MACTEL trial, and the second half maybe about our research. All right, so we were participating in the MACTEL trial, the phase 3 trial. So I'll tell you a little bit about that. And that just finished, and they sort of had the top line results in November. And so basically they met their primary endpoint in the two parts of the trial. So there was a protocol A and a protocol B, sort of FDA mandated, two protocols separate, and both of them met their primary endpoint, which is slowing down the rate of growth of the ellipsoid zone in this disease. So now the company, Neurotech is taking the CNTF into the FDA to see if they can get approval. And it's basically an implant that's put in the pars plana, sewed in, and it's got a trapped cells inside that implant, releases ciliary neurotrophic factor, and then that supposedly helps keep the photoreceptors alive. the implant was safe, durable, and well tolerated. And that's all we know at this point because the study is with the FDA, hopefully. And so I'll go back and tell you a little bit more about the MACTEL project. It started in 2005. I think Dr. Gass was there. I don't know if Anita was there. Yeah, so the backstory is that it started.
Somebody who owns a lot of departmental stores in Australia has this disease, and he asked Alex, who was a fellow in Sydney, and Alex could reach Don Gass, and Don Gass was sick, but he went up with media to Baltimore to kind of start the kind of be there at the first initial whatever talks and planning.
So that was the beginning, 2005, and it started out as a natural history, but the ultimate goal was to find a cure for the disease that the Lowy Foundation sort of son has. So they started out with natural history, imaging, genetics, and then on the side, they started some laboratory studies, which Martin Friedlander's running. And they also did some histopathology studies in donor eyes and developed the donor eye program. And Emily Chu has been key in this project now, and I'm grateful for her slides that she shared with me about the study. So currently, the natural history observational study is still ongoing, and we're still recruiting for that. So if you have any patients, please send them. And they're probably up to more than 1,900 now, and that's the basis for the natural history studies. And the importance of the natural history studies is basically defining the endpoint for the clinical trial. So that was the starting point. And here's sort of a classic example of MACTEL. You have that pigment migration along the vessel and the right angle venule and a hyper reflective spots and the sort of grayish tinge to the retina. From the MACTEL study is probably when we learned that loss of hypoautofluorescence in the fovea and loss of macular pigment was an early finding in this entity. So if you have that alone, it may clue you in when the patient even doesn't have any of the other signs. So loss of the macular pigment and loss of the autofluorescence or hyperautofluorescence in the fovea is a very early sign. Blue light reflectance showed you the entire sort of extent of this hyper reflectivity and the sheen in the macula that extends outside the fovea and the vasculature. And then, of course, the telangiectasia, which is a key feature of this entity, and it extends temporally mostly, but also nasally. So there's a lot of telangiectasia that we see in this disease. And of course, there's some leakage, but the key feature here is the cystic changes are not exudative. So they're more degenerative than exudative. And that's one differentiating thing. And of course, you can develop secondary subretinal neovascularization. And at this point, if they have bleeding or leakage into the retina and exudation, this is treatable with anti-VEGF. At this point, that was the only treatable part of this disease. And that's when the MACTEL trial came in with the implant studies. And phase 2 read out back in 2018, and it was positive. And they showed that in those 67 sort of phase 2 trial patients, the implant slowed down the progression of enlargement of the ellipsoid zone loss. And so to get this through the FDA, they really had to prove that ellipsoid zone loss can be used as a surrogate marker for disease progression, because this is an entity where the vision stays 20/20 very, very late. So how do you get approval? You can't use vision because vision stays good for a very long time. So they had to show that visual function and ellipsoid zone loss correlated very tightly. And that was the starting point. That's when the FDA approved that ellipsoid zone loss as a primary endpoint. And that was a key step to move this forward into phase 3 trials. So CNTF is a neuroprotective cytokine produced largely by Miller cells and neurons, expressed in the retina under stress conditions. And this was shown in animal models, and it's shown to protect photoreceptors in rodent retinal degeneration models. And Neurotech developed this orphan drug designation to get it through the FDA for this application. And so the implant is sort of has a metal ring that's sewed into the sclera, much easier than a Retisert implant for those who have done it, because it's smaller and round. So it shouldn't be a big deal to sort of, if this gets approved. And in the implant is a trapped, cells are trapped in there, and they're just producing CNTF and neurotrophic factors. Nothing escapes except the small molecules. So only the CNTF is getting released, but the cells stay trapped inside the implant. So when you're putting it, you have to be very careful not to touch or break that membrane. And so this was sort of the beginning for the phase 3. So phase 1 was a safety study. Phase 2 was the sort of the starting point, 67 eyes that showed the primary endpoint was successful. That took them to phase 3, which we participated in. And thank you to everyone online and in the room who referred patients for that study. And two identical FDA mandated protocols, A and B, to recruit 112 participants. Only one eye per patient was enrolled to receive the implant. And it was multi-center international study. But to go back to the pertinent eligibility criteria, in case this becomes approved, one wonders if the same eligibility criteria will stay through. So it's important to realize that this is not for everyone with MacTel. It was a very tight inclusion criteria to get patients to be eligible for the study. So they had to have an ellipsoid zone loss that was between 0.16 and 2 millimeter in size. And most of our patients failed because the loss was too small. They were too early. A lot of our patients, we tried to get them in the study, their vision was too good and the ellipsoid zone loss was too small. So that basically means, so here's an example of lesion too small, which is the majority of patients we tried to get into the study, because when you measure this one is 0.02, not enough. Here's the lesion that's just the right size, 0.16 millimeters. So that's the right size, which is about the thickness of the fovea, if you want to sort of guesstimate it. But we basically had to do a baseline study, send the pictures to the reading center at Duke, they'd measure it and they come back, say eligible or ineligible, thumbs up or some thumbs down before we could get them in. The second sort of important criteria was hyperreflectivity. They couldn't have a lot of hyperreflective foci. So unlike the hyperreflective foci from this morning, these are very different hyperreflective foci, and they were exclusion criteria. And there was a very good reason for that. So when they looked at the natural history study and the phase 2 study, patients with that hyperreflective appearance really didn't do as well, and they had a worse natural history. Here's sort of an example of that. So here's a patient from phase 2 that was enrolled, and they had that hyperreflective appearance. And this is sort of their baseline ellipsoid zone break. And this is what happened over 24 months. This was, boom, enlargement of the ellipsoid zone. So these patients did not benefit from the implant at all because their natural history was so bad. And so here's sort of an example of what happens with OCT hyperreflectivity. No benefit from the implant versus those without the hyperreflectivity, a very good benefit from the implant. So that was sort of the beginning for the phase 3. So none of the patients that were enrolled had this hyperreflective lesion. So when we first started working on MacTel with OCTA, we were talking to Emily and she wanted us to look at our data to see what this hyperreflectivity means. We had suspicion that this is probably blood vessels in the wrong place, surrounded by pigment. And so we looked at all our eyes with hyperreflectivity. And we looked at the OCTA to see what was going on in these hyperreflective lesions. So in eyes with intraretinal sort of abnormal blood vessels, where the sort of the blood vessels dived beyond the outer plexiform layer, 7 out of 10 had hyperreflectivity. So these lesions really correlate with blood vessels going in the wrong place in these eyes. Here's an example of patients where the lesions went even deeper in the outer retina, and they were all new blood vessels. So hyperreflectivity in this disease really means blood vessels in the wrong place, whether outer retinal or deep retinal vascularization. So now I'll go to the research part of the talk, now that I've told you about MacTel. So we're waiting for FDA to give the thumbs up or thumbs down for this implant. But this is sort of one of my favorite papers about this disease. So when we started using OCTA, I told my students we need to find something interesting to do. And this paper sort of popped in our sort of search. And it's really a lovely study done by the group in Lariboisier, Godric and Tadayoni. And they looked at basically eyes where the outer hyperreflective material or the these abnormal blood vessels went into the outer retina, and there was complete overlap between the ISOS loss or the ellipsoid zone loss and these blood vessels. So that was sort of a complete overlap of vascular changes and photoreceptor changes in the end stage of the disease. But then they had some eyes in their group that had the outer sort of photoreceptor loss, but no new vessels. So we asked ourselves, what is going on in those early stages? So in the end stage, there's complete overlap between photoreceptors and vasculature, but what's going on in the earlier stages? So we basically had the OCTAs on these eyes, and as Phil earlier told us, we could take slabs in the outer retina from the OCTAs. They'll give us the structure. So we can do a slab in that area here and look at the ellipsoid zone loss. And then we know that there's telangiectasia, and in MacTel, the telangiectasia is in the deep vessels. So we can outline the telangiectasias manually. And then look at, is there any overlap between those two lesions? So telangiectasia manually, outer photoreceptor loss, sort of automated, and then look for overlap and pseudo color the overlap in orange. And so there's a wide range of overlap as we've seen. So when you look at very early stages where there's very little photoreceptor loss in green here, there's hardly any overlap. The lesions are adjacent. But then when you look at sort of mid stages of the disease, then they start to overlap the telangiectasia and the photoreceptor loss. And then these end stages that Godric described, there's complete overlap. So what's really happening here is that as the lesion size on photoreceptors grows, the overlap becomes more complete. But what about the earlier stages when there's no lesions on the photoreceptors, right? What are these lesions correlating with? So this is the new grading system that Emily Chu and the group, the natural history group, has developed. It's basically looking at OCT as the grading criteria, and they sort of escalating from ellipsoid zone break that's non-central to central to pigment to neovascularization as the sort of end stage. And there's 6 stages in this classification. So we wanted to look at what's happening here before the ellipsoid zone becomes central. And our hypothesis was that it's the interdigitation zone. So the sort of the earlier loss of the tops of the photoreceptors and the RPE cell overlap. So Janice Ong, who just matched to Harvard with Deeba, be on the lookout for her. She's a superstar. She took this project, and we basically, instead of ellipsoid zone loss, we wanted to look at the interdigitation zone. So loss as green and attenuation as yellow. And then again, looking at the telangiectasias, and she developed this algorithm to look at them in 3D and look at overlap again. Here's the telangiectasias on top of the ellipsoid interdigitation zone loss. And to make a long story short, the overlap basically in the earlier stages is with this interdigitation zone loss. So when the telangiectasias are early, when the disease first appears, the overlap is between the telangiectasia and the interdigitation zone loss. And as the disease progresses, photoreceptor loss becomes more and more progressive until you get that complete overlap with ellipsoid zone loss and outer retinal neovascularization. And this paper is already published. Didn't want to take too much time. But the takeaways are basically as the disease progresses, you start with interdigitation zone attenuation, then loss, then photoreceptor loss with ellipsoid zone, and the telangiectasia keeps growing and the overlap becomes complete with death of the photoreceptors. Telangiectasia and photoreceptors are sort of interrelated as the disease progresses, and hyperreflective foci are a bad sign. They correlate with these vessels that are sort of an end stage in the disease. And we're looking forward to results of the sort of FDA judging of this, and our patients are watching. They have a Facebook group and they're all waiting for this. So they'll be coming out and coming to you soon to get the implant, hopefully with approval. And thank you to Jenny and to the audience.
Thank you, Amani, for sharing us these results and giving us your research. That was amazing, amazing work. Are there any questions for Dr. Fawzi? Yes, Anita, please give her a mic. Dr. Agarwal is asking, what do you think is the first inciting step?
Yeah, that's a great question. So, you know, I don't know. I mean, you have to believe that the Miller cells, somehow dysfunctional Miller cells, because they're the one thing that connects photoreceptors and blood vessels. So maybe the defect starts in the Miller cells and the well, I don't know how to visualize sphingolipids, right? Sphingolipids, the mutation is in not every patient, and I think they're looking at sphingolipids as a supplement right now, but I don't know where that's going to go. But what we know about sort of the end stage is that Miller cells are completely wiped out. So there has to be something happening in the Miller cells, because that's the link between photoreceptors and blood vessels in my mind. So, so what is that thing? I don't know. I don't know.
Outer retina, does that make any difference?
So yeah, that's you asking the same question that Vasara asked me. You guys.
Well,
must
did
be-
you read the 1984 paper by Don Gass where he drew this by hand?
No.
1984, he figured all this out.
Yeah.
For all those people who want to talk about Mactel, you have to read his first paper, because this is all described by Don Gass, and he figured out those photoreceptors were going, the blood vessels are growing into the outer retina, and the RPE cells are
What did he
migrating
think
out.
it was?
He thought it was Mueller cells.
Okay,
Yeah,
good.
yeah.
I got glad I got
Yeah.
it right. So, but yeah, so the classification system, what Voss was saying is that he was sort of he's a little miffed at the classification system because it doesn't talk about the cavitary lesions, whether inner or outer. It only thinks about photoreceptor loss and hyperreflective lesions. So he's taken a point with that, and I mean, I guess that's a valid point, because these cavitary lesions mean degeneration, loss of tissue, right? And the new classification system doesn't consider it. Yeah, it's definitely not leakage, right? Yeah, no, I think Don is always right.
Great, thanks. Amani, what do you think those crystals are? Maybe you said to it and I missed it. Where are the
No, I
crystals?
didn't
You didn't say it.
I didn't say anything, and I didn't think about them, but I'm wondering if they're sort of the end plate, the Miller cell end plates that are dying or degenerating because they sort of have that appearance. What did Don say, Anita?
Give her a mic.
Okay, good.
Okay, so end plates of the Mueller cells per Don Gass. Great. Thank you so much again. And our next speaker is my colleague, Dr. Felix Chow. Felix is an associate professor of ophthalmology here at the UIC. He graduated from Duke University with a bachelor's in science and biomedical engineering in 1996. And then he worked for Motorola as an engineer, and then finally saw the light and decided to go to medical school and become an ophthalmologist. So he went to Iowa, went to medical school, got a Gold Humanism Award in AOA there as well, was a Duke resident, and then we were fortunate to get him as our fellow, and then fortunate to keep him as our faculty. So you've been here since 2010, so you can figure out how old he is now, and then some because of the years before. So Felix is very interested in imaging studies. He's done animal models of ROP. He's analyzed SD-OCT changes in sickle cell with me and some of our patients. And he's also been involved in providing care for the Boston K-Pro patients, very difficult patients, and also participates in a lot of the clinical trials here at UIC. He's a pediatric retina specialist, also taking care of ROP and retinoblastoma, as well as adult retina. So today I've asked Felix to speak about fundus autofluorescence and its clinical utility. Felix?
Thanks so much, Jenny, for the chance to speak. This will be a kind of a big picture review of the use of autofluorescence in clinical cases. So first, by background, the fundus fluorophores receive excitation light photons and then reach higher energy states. The electrons in these fluorophore molecules then return to lower energy states, and that's what leads to the emission of photons at a lower energy, longer wavelength. These fluorophores are pretty complex with many atoms, so there can be many electronic transitions with broad wavelengths of emission and excitation. So it's important to not overlap them for best imaging. Here's a diagram showing the spectra of typical fluorescence. Usually we're exciting with green and blue light. Lipofuscin, which we'll talk about, has an emission spectra a little lower in the yellow and orange range. And there's also autofluorescence in the near infrared range. Again, lipofuscin is that primary substance that's imaged by short wavelength autofluorescence. Its end formation is with A2E that eventually leads to the lipofuscin accumulation. This was first identified during the pre-injection of fluorescein and was also seen with ICG. So this is how lipofuscin forms in the visual cycle. All transretinal binds to phosphatidyl ethanolamine that forms A2E, other bisretinoids, and then lipofuscin. And as this accumulates, this is what causes the autofluorescence. There are many wavelengths that can be used. In the short wavelength autofluorescence, it's primarily green and blue. Again, lipofuscin is the primary agent along with other bisretinoids, and these have an emission peak in the yellow-orange range. Typically, there's a dark centered region in the fovea, dark vessels, dark disc with a bright background. The foveal pigments, lutein and zeaxanthine, kind of mask some of the underlying autofluorescence. Near infrared autofluorescence uses a wavelength for excitation around 820. This also has a dark disc and retinal vessels, but it actually has more fluorescence at the fovea by higher melanin and luteal pigments. So what can cause hyperautofluorescence? Well, there are many reasons. Primarily, it's from the buildup of autofluorescent compounds like lipofuscin, but there can also be unmasking of autofluorescent compounds from depleted photoreceptors or luteal pigments. Porphyrins from subacute and chronic retinal hemorrhage can also cause increased signal. Other features like barosclera, calcification, drusen, these can all increase lipofuscin and autofluorescence. There can also be decreased autofluorescence or hypoautofluorescence from the loss of autofluorescent compounds like lipofuscin and loss of RPE or blockage from overlying structures. Interestingly, there may be interference from rhodopsin itself, which has filter excitation and emission properties. And this fundus autofluorescence 30-degree image right after this picture was taken, the wider field picture was taken, and you can see how bleaching the photoreceptor rhodopsin led to increased natural autofluorescence from the RPE underneath. So definitely it's an interplay of photoreceptors and the RPE underneath. Note the crystalline lens and cornea can also have emitted light after excitation, and that can throw more noise into our signal. Systems out there employ different strategies. The scanning laser ophthalmoscope uses a flying laser spot that goes over a raster pattern using tunable mirrors. The backscattered light is then reassembled into images. This has a high frame rate, high sensitivity, and needs only a small pupil. However, this lacks true color imaging and can have artifacts. Confocal systems are helpful because they can amplify light from a desired plane, such as the retina, and suppress other light, and that can minimize noise from other structures. Fundus cameras, in contrast, use a high energy flash through a wideband excitation filter and mirrors and apertures to reach the retina. The signal coming back has lower autofluorescence, and there can be more noise from other features. It's also non-confocal. This slide shows many of the different commercial systems out there, many employing confocal SLO, some employing fundus camera technology. Again, the excitation wavelengths shorter for higher energy emission wavelengths, or longer with lower energy with different advantages. So here are different images. Each system has its own proper interpretation. Here's a blue SLO spectralis autofluorescent image. Again, foveal hypoautofluorescence in a larger hypoautofluorescent zone nearby. This is an artifact from vitreous floater. The green SLO from Optos here shows a less hypoautofluorescent center at the fovea and a more visible nerve. Similarly, the green fundus camera autofluorescence here by Topcon has less autofluorescence less hypoautofluorescence at the fovea and nerve. Here's a red free image just for reference. Here's a near infrared image, again with hyper-autofluorescence at the foveal region from luteal pigments and melanin, but also dark hypoautofluorescence at the nerve and vessels. The Cless system can use blue excitation for this type of autofluorescent image, or green excitation for this autofluorescent image in the Cless system. So different types of technologies, different types of images. How do we apply these in clinical scenarios? Well, in AMD, age-related macular degeneration, we know the RPE Bruch's membrane have very significant changes, and autofluorescence really helps us with diagnosis and prognosis. Early lesions are more readily detected by autofluorescence than on color imaging. You can see focal hypo and hyper autofluorescence early as speckled, patchy, and other descriptors for patterns. When there's hypoautofluorescence, that's often from atrophic or degenerating RPE with less lipofuscin. When there's hyperautofluorescence, it's usually hyperplastic or dysfunctional RPE from lipofuscin accumulation. There's variable patterns with drusen. Hyperautofluorescent drusen can have altered, hyperautofluorescent around drusen can be from altered RPE metabolism, and vitelliform lesions also have increased autofluorescence. Reticular pseudodrusen or subretinal drusenoid deposits, these are at risk for progression to advanced AMD. These are readily seen on near infrared and blue shortwave autofluorescence as a network of dots. And geographic atrophy in AMD, there's this profound loss of RPE and fluorophores with well-demarcated hypoautofluorescent regions. There can be junctional zone areas of increased hyperautofluorescence that usually points to the progression of autofluorescence for the geographic atrophy. So here are some examples of subretinal drusenoid deposits, very visible on near infrared reflectance and blue autofluorescence here in spectralis, these subretinal dots, these drusenoid deposits, not as seen on green autofluorescence here. Here's an example of geographic atrophy. Early, there are these smaller focal geographic atrophy regions with patches of hyperautofluorescence surrounding. With progression, those areas of hypoautofluorescent GA areas increase into those expanding borders of hyperautofluorescence. And as progression continues, again, the hypoautofluorescent regions get bigger, but there is still this hyperautofluorescent edge. So this pattern will show up time and again. In neovascular AMD, there are also different patterns based on the RPE integrity, whether there's hemorrhage or exudation, and the type of a CNV. There can be in type 2 subretinal CNV a decreased autofluorescence from blocking. This is associated with leaking on FA. And I call type 1 sub-RPE CNV, there's often hypoautofluorescent spots thought to be from focal RPE atrophy. And there can be rings of hyperfluorescence around CNV suggesting increased activity with the RPE. This can point to more subretinal fluid, need for more injections, and more ellipsoid zone disruption. Worse outcomes in vision with more autofluorescent alterations, as would make sense. RPE tears have a very dramatic hyperautofluorescent folded RPE with an adjacent hypoautofluorescence where the tear has occurred, but this may later recover as the RPE recovers. So here's a vivid example of how there is hypoautofluorescence in classic CNV associated with the leaking CNV. In this image B, there's also a halo of hyperautofluorescence surrounding the hypoautofluorescent CNV, again, probably from RPE activity. Here in this occult CNV with the CNV under the RPE has a more modeled hypoautofluorescent pattern thought from RPE focal losses. And here's a very dramatic image of RPE tear, this large RPE atrophic area now with hypoautofluorescence, and the redundant folded RPE with hyperautofluorescence from more lipofuscin and folding. All right, in diabetic retinopathy, this autofluorescence is a little bit more investigative. In diabetic macular edema, we have identified more visible spaces of fluid from CME and from pooling with autofluorescence. Autofluorescence may allow for longitudinal monitoring of the outer retina and RPE integrity. In central serous chorioretinopathy, in the acute phase, there may be really normal autofluorescent signal, but with time, more changes are seen in autofluorescent. Over years, hypo and hyperautofluorescent areas can occur from declining photoreceptor outer segments in RPE. When it becomes homogeneous with hypoautofluorescence, usually it's with total loss of the RPE in the associated site. There could also be variable granular changes or even a gravitational guttering pattern. Acute patterns have also been tracked over time, going from blocked autofluorescence to mottled autofluorescence to hyperautofluorescence to mixed, and then a descending track. As the duration of disease is longer, there's more of these features, more loss of autofluorescence. PEDs can also have apertures seen as focal areas of RPE loss. So here's a vivid example of mottled hyper and hypoautofluorescence in these more center areas, again with a variable RPE and outer retinal atrophy. Interestingly, there's a shrouding halo of hyperautofluorescence thought to be from outer retinal thinning with more intact functional RPE with lipofuscin. And here's that gravitational pattern, probably from guttering fluid. This is pretty diagnostic for central serous. Retinal dystrophies, as beautifully described earlier, can also have autofluorescent patterns with a degeneration of photoreceptors and RPE with or without lipofuscin accumulation. There can be very distinctive patterns. People have even used neural networks and autofluorescent patterns to diagnose many different dystrophies with pretty good accuracy. As genotypes become the paradigm for making the diagnosis, autofluorescence is becoming important for disease progression monitoring and looking at clinical trial outcomes. In Stargardt's is the most common juvenile retinal dystrophy. There can be variable macular atrophy and yellow-white deep lesions. These flecks can have hyperautofluorescence from RPE lipofuscin increases. There can then reach a maximized generalized increase in autofluorescence as a peak value, and then it can decline with dark flecks and atrophy. In a lot of literature, there is this peripapillary sparing of autofluorescence characteristic of Stargardt's, but as Anita pointed out, that may not be so true anymore. As things progress and there's lower macular autofluorescence, this is associated with more atrophy and rod and cone dysfunction. And as the lesions become more hypoautofluorescent, the type and severity increase. So here's some examples of dense hypoautofluorescence here right in the foveal center with RPE atrophy, but there are these perimacular hyperautofluorescent flecks with lipofuscin buildup. The hypoautofluorescent flecks were thought because of RPE atrophy. And again, at least in this photo, there's peripapillary sparing. Best disease and vitelliform maculopathies are also associated with variable patterns of hyper and hypoautofluorescence. Here's one study showing the pre-vitelliform state. It's essentially normal. And that vitelliform yolk state, the material that develops is more hyper autofluorescent. As that becomes more disruptive as a pseudohypopion, the material itself remains hyper autofluorescent, but that space of fluid is more hypoautofluorescent. As it becomes vitelloruptive, it's more variable. When it becomes very atrophic, involving the RPE, then there's a large geographic area of hypoautofluorescence. Pattern dystrophies also again relate to this abnormal lipofuscin accumulation with the RPE damage in the posterior pole. Many are caused by this PRPH2 mutation. Many different features of hyper and hypoautofluorescence. A lot of them, though, with the same principle. Here's a butterfly pattern type dystrophy. Here you can see hyperautofluorescence right within the lesions themselves. And then in adjacent areas, there's hypoautofluorescence thought from RPE atrophy. Central areolar choroidal dystrophy is a dysfunction of the photoreceptors with visual acuity decline at age 30 to 60. It has a much more well-demarcated outer retinal RPE choriocapillary atrophy as compared to the geographic atrophy of AMD. In AMD, the autofluorescent changes go more beyond the macula and they're less well demarcated than in central areolar. This diagram here shows the stages of central areolar by autofluorescence. In stage 1, there can be a hyperautofluorescent parafoveal hypopigmented lesion. In stage 2, there's a more speckled hyper and hypoautofluorescent change that's expanding as the hyperautofluorescent areas coalesce and there's more atrophy of the RPE. There can be more hypoautofluorescence that leads to stage 3. And then as that hypoautofluorescent geographic atrophy area expands, it's looking more like this, but there can still be this hyperautofluorescent ring. Again, very well demarcated as opposed to a more variable AMDGA picture. Retinitis pigmentosa and rod cone dystrophies have characteristic rod and cone photoreceptor degenerations. These can also have variable ring-like patterns that kind of go with the ring scotoma of retinitis pigmentosa. In the parafoveal region, there can be a hyperautofluorescent ring. This actually corresponds to increased photoreceptor phagocytosis and lipofuscin accumulation. It may constrict with disease progression. There's more retinal sensitivity and microperimetry within this area, and it's more impaired outside that area. Outside, there can be more peripheral ring patterns of autofluorescence as well, and we'll go over some of those here. In this macular wider field autofluorescent image, there's a macular hyperautofluorescent ring. This is the area that has better microperimetry sensitivity. But however, in the periphery, there's a second hyperautofluorescent ring that delineates the more hypoautofluorescent regions. Here's a more dramatic example of dense peripheral nummular hypoautofluorescence in the periphery with dramatic RPE atrophy. There's actually no autofluorescent ring in the macula in this picture. Here's another dramatic case of hypoautofluorescent modeling going to the mid periphery where there's a more mild hypoautofluorescent change. And here there's an asymmetric macular hyperautofluorescent ring showing decline occurring in the macular region. And here's a more speckled peripheral hypoautofluorescent pattern with a more preserved macular region. Another condition is pigmented perivenous retinochoroidal atrophy. On pseudocolor wide field, you can see bone spicules and pigment clumping and CR atrophy around the veins. Interestingly, on green wide field autofluorescence, there's more obvious hypoautofluorescent changes, and also in this case, a hyperautofluorescent macular ring. So better highlighting of pathology there. So retinal dystrophies in general, the autofluorescence helps us understand disease progression over time with increased hypoautofluorescent spots and reduction in size of this macular autofluorescent ring. This may become more important as clinical trials determine if treatments are working. Retinal toxicities can also have distinctive autofluorescent patterns. In hydroxychloroquine, these can be more centrally involved in non-Asians. In Asians, they can be more peripheral. These changes are detectable by autofluorescence, but the Academy still recommends OCT and visual field as primary tests. So here's some images of progression, initially with this classical bullseye pattern of modeled hypoautofluorescence surrounding the fovea. As these areas of RPE and outer retinal decline continue, there's more modeling in central involved hypoautofluorescence, and eventually a more geographic atrophy-like region of autofluorescence. Here, though, with also a surrounding hyperautofluorescent ring showing where the RPE is going through more metabolic changes and likely will decline further. So Pentosan is also associated with retinal toxicities. This has highly variable patterns as well. It's still a bit uncertain what the threshold dose is. Here's one of the key papers just highlighting the broad spectrum of what can occur. Just this modeled hyper and hypo autofluorescent pattern that can expand, growing, involving more of the macula, and eventually these hypo autofluorescent geographic spots can also be seen. The furoxamine mesylate is an iron chelating compound for iron overload conditions. It can also have very distinctive macular and peripheral pigmentary changes. These are possibly starting off as minimal, but they can also have these radiating spoke-like changes of hyper and hypoautofluorescence, and also can lead to more advanced hypoautofluorescent losses and geographic atrophy patterns. Interestingly, other conditions can have autofluorescent patterns, including pseudoxanthoma elasticum. The many features that we see clinically in pseudoxanthoma elasticum can be seen with hypoautofluorescent subretinal deposits or hyperautofluorescent deposits with the comet lesions and dystrusion. The angioid streaks typically are autofluorescent with some progressive RPE atrophy. Here are some vivid examples of this angioid streak pattern with hypo and hyper autofluorescence. When the macula is more diseased, sometimes the wide field images can point to the diagnosis. In macular telangiectasia, as nicely reviewed by Amani, you can also have these idiopathic bilateral macular changes, juxtafoveal, and this is associated with retinal atrophy, crystals, microaneurysms, and CNV. There are variable patterns that are associated on autofluorescence. Here you can see mixed decreased autofluorescence by these pigment depositions. Here in B, there's a more variable hyper and hypoautofluorescent pattern correlating with the cortical neovascularization. In C here, we've got blunted parafoveal venules, that's shown in the asterisk. And interestingly, here in D, there are ectatic capillaries temporal to the foveal center. These are better seen actually on autofluorescence compared to the fundus image. So potential role there for autofluorescence for this condition. And finally, retinoschisis and retinal detachment. These also have potentially helpful features on autofluorescence to make the diagnosis. Here's in this first row, rhegmatogenous detachment with a tear here superiorly. The posterior margin of the detachment is associated with hyperautofluorescence. The zone involved with the detachment in subretinal fluid is hypoautofluorescent, and the tear is hyperautofluorescent by window defects. With more chronicity, the subretinal fluid region becomes more isoautofluorescent, but that posterior margin still keeps its hyperautofluorescent appearance. In retinoschisis, in contrast, the schisis cavities are more isoautofluorescent, and the posterior margin is more hyperautofluorescent. If, however, this progresses to a schisis-associated detachment, there can be an associated hyperautofluorescent pattern associated with the posterior margin, kind of reminiscent of the retinal detachment features. So with retinoschisis, if you see a hyperautofluorescent region near and beyond the posterior margin of the schisis cavity, be on the lookout for retinal detachment with that schisis. So in summary, the clinical use of autofluorescence is expanding greatly. It's a non-invasive technique to look at the metabolic changes in the photoreceptors and RPE layers. Peripheral and macular changes of autofluorescence are helping with clinical decision-making. There are many platforms with different technologies that have slightly different interpretations, and they're increasingly being used for diagnosis, assessing disease progression, and monitoring response to treatment. Thank you.
Thanks very much, Felix. I'm wondering, is there any way to quantitate the hyperautofluorescence? Because I think like in Plaquenil, they're saying that the hyperautofluorescence that occurs actually beats out the OCT. So you see that before the OCT change. Is there a way to quantitate it?
Yeah, there is actually another technology called quantitative autofluorescence. It's a different strategy where it's actually summating the amount of autofluorescence over time. And yes, there are some autofluorescent technologies that seem to detect the plaquenil associated changes more readily even than OCT. However, our academy is still advocating the OCT and perimetry, or sorry, visual fields at this time.
Great, thank you. Any questions for Dr. Chow? Additional questions? No? If not, thank you so much, Felix. Our next speaker will be Dr. Anita Agarwal, who will be speaking on inflammation in the macula. What is that lesion? This highlights another area that Dr. Agarwal is very interested in, and that is in inflammatory retinal macular diseases. Anita.
I think we're running behind, so we're going to try to keep this going fast. And no disclosures. The idea of this talk is again to identify the disorder. Most of you know these disorders. I'm speaking to the core, and we'll just look at a few new imaging features and perhaps a little bit of new understanding. This was a 54-year-old gentleman who had progressive decrease in vision in the right eye for 18 months, and he was asymptomatic in the left eye. Obviously, the right eye has a very obvious lesion, but the left one also has a lesion that's very subtle. And I think if you were on the Anuzzi rounds this week, you saw one of this, and you were on the Anuzzi rounds a couple of months ago, there was one of this. So this is being more easily recognized. And the fluorescein angiogram shows that the lesion is completely in the choroid because the fluorescein is silent, whereas the ICG shows blocked or dark hypofluorescence corresponding to the lesion because the lesion's in the choroid, so that helps us. And late ICG shows not only the lesion, but also the small little satellite lesions, which helps kind of come to the diagnosis. The fluorescein angiogram is normal, except for maybe a little bit of changes in the overlying RP here. I had to save time, I didn't use the right eye's image. But on autofluorescence, you can tell that the pigment epithelium is broken down where that area of yellow, intense yellow was, and that's why the patient was symptomatic. So the lesion obviously began in the choroid, but as over time has affected the outer retina and the RPE, causing him to become symptomatic in his right eye. The money is in the OCT. OCT with enhanced depth imaging that Rick Spade taught us about helps us look at the choroid. And here you can see that this choroid has a space occupying lesion. But the important thing to note is that the overlying choriocapillaries in the left eye that's asymptomatic is intact. So the lesion begins in the choroid and probably spreads. It's a granuloma. And the right eye has been present for a longer time, and there's outer retinal atrophy and RPE changes, which caused him to become symptomatic. And the diagnosis was sarcoid. He also has a history of some prior lung issues and had he was diagnosed with ocular pulmonary histoplasmosis, but I think the diagnosis was revised when he developed this in his eye, and the pulmonologist had a chance to look at him. The important good thing about sarcoid is it's highly responsive to steroids. So within 6 weeks, the lesion shrank significantly, and now all the choroidal vessels are visible. So I wouldn't say that this lesion is a vascular event or a vascular kind of no perfusion in the choroid, but it's just that the choroidal vessels are all being sort of compressed by the granuloma. And once the granuloma subsided, the choroidal vessels are again visible. To summarize, this is similar to what we already saw. And as long as it's in the choroid, the overlying pigment epithelium is intact. Unlike what we saw there, this patient has multiple lesions. She actually has, she's a little bit young for this disease and has more lesions in the macula than one would expect. She was only in her early 40s, and she's been symptomatic with floaters and photopsias for the last year and a half. More typical is a patient with this lesion, where the lesions are much more prominent in the nasal retina or near the arcades and not so much in the macula. They're oval, they're in the choroid. And most of them, kind of the lesions come on subtly, and they have progressive increase in floaters. And because the lesions in the choroid, the fluorescein angiogram again is normal, and the fundus autofluorescence imaging does not show us any abnormalities. The fluorescein, as I mentioned, is normal. And the choroid OCT imaging of the choroid is really important because it shows you a very boggy, thick choroid, much thicker than the retinal thickness. And you don't really see any granulomas here corresponding to these lesions. I tried very hard to find them, and we got high spectralis OCT, to look at all these patients with birdshot to see if we could find something in the choroid to kind of find those creamy lesions, but I couldn't. And the one thing is that the choroid, the OCT is useful in looking at their response to treatment. As you can see in this example, this patient was diagnosed with birdshot and had somewhat large choroidal vessels. He doesn't have that much of a thick choroid as a previous patient who was a myope. And then once you treated the patient on methotrexate and you had stable disease, the choroidal thickness came down, all the Pachy vessels started looking a little smaller. So we know that the inflammations in the choroid and there are secondary dilated choroidal vessels from patients with birdshot choroidopathy. And as we mentioned, it's a disease primarily of the choroid, but if it's ignored or nobody makes a diagnosis, then the retina is secondarily involved. These patients can get photoreceptor changes, so they develop photopsias. Eventually, if it's not diagnosed or not treated adequately, they can get outer retinal thinning and they can look like secondary retinitis pigmentosa. Unlike the last two patients who had bilateral disease, this patient has unilateral disease, fog in the vision in the right eye for about a week or less than a week, and she's a known myope, and I was followed for a myopic schisis for many years that had progressively worsened, but her vision was still at 20/25, and she developed these creamy lesions in the right eye. Often these patients, you may not see them when you're looking at the slit lamp with a small slit. Sometimes you miss those lesions, but you send them off for fundus photographs, and nowadays with autofluorescence imaging, you can catch the lesion. The left eye is normal, except for high myopia. And wide field imaging shows that the lesions are not just in the macula, but also in the nasal retina. And sometimes they go up to the periphery and often they don't go up to the periphery. And when you order a OCT, I always want to get a spectralis because I want to go look at the entire lesion. I don't like just 5 cuts in the macula that they do with Zeiss. So if you look at the areas where the lesions are involved, you can see that there are flares in the outer retina corresponding to those creamy lesions. Sometimes these flares are not very visible, and sometimes they're just in the fovea, and you kind of can get a little spike or like a little flare that we call that's limited to central fovea. And it's important to really find these lesions and get OCT through those lesions so you don't miss the diagnosis. Autofluorescence imaging, as I said, is very useful, and you can use this to ask your photographer to make OCT cuts in one of where these lesions are prominent. The fluorescein angiogram is useful. These days, we don't need the fluorescein angiogram to make the diagnosis. In the olden days, when we did not have autofluorescence and OCT, we definitely relied on the fluorescein angiogram. And these lesions are made up of small little dot-like lesions, so they kind of have this wreath-like appearance. And sometimes the wreath is visible, sometimes not that visible, but the important thing is the lesion is hyperfluorescent. And the good thing is that it gets better without any treatment. Sometimes the lesions are subtle, as in this patient, you see a couple of them in the macula, then you can look at the periphery, there's a little bit, but red-free imaging is useful. This was before we had autofluorescence imaging. Red-free was a useful tool to make the diagnosis of mutes. And now with autofluorescence imaging, you can skip the red-free. Unlike the previous one that had unilateral creamy lesions, this one has bilateral creamy lesions. The vision is profoundly down to 2100 and 2200 and count fingers between the two eyes. This patient's a young adult who was 19 years of age. The fluorescein angiogram shows hypofluorescence of the lesions. The ICG is also hypofluorescent. But unlike the previous example that we saw of the sarcoid, this patient shows increasing hyperfluorescence on the fluorescein angiogram. However, the OCT remains dark. And a few of these lesions are intensely hyperfluorescent, and that's because they probably have a small pocket of subretinal fluid overlying this. And when Don Gass described this condition, he did not think that there was subretinal fluid, but now in OCT, in some eyes, we do see a tiny bit of subretinal fluid in the apex of these lesions. And there was a lot of understanding, a lot of discussion about this disease. This was one of the first diseases that Don Gass described in 1968. He described a few before that, but this was probably his landmark disease. And my interest in his diseases came out from this condition because we saw patients with this, and we thought they were all recurrent APMPP at that time, and now we know they're all TB serpiginous. So I'll show you those patients. That was part of my thesis when I did my residency in India. And that's how I learned about Don Gass. My chief introduced me to Don Gass through this disease. And at those days, we thought they were all APMPP, and we were wondering why Gass said they don't have recurrences, but we kept seeing recurrences in India because we put them on steroids. And once we taper the steroids, they come back. And anyway, eventually, after 25 years or 20 years, we realized those were all TB serpiginous and not really acute posterior multifocal placoid pigment epitheliopathy. When Gass, started his career in 1963 in Baskin Palmer, fluorescein angiography was new. So he really was using the fluorescein angiography as a tool, along with his knowledge of pathology, to figure out pathogenesis of many diseases. And then this one, he showed us that the hypofluorescence is initially is the lesion is hypofluorescent, and later on it becomes hyperfluorescent. So his theory was that the lesion is in the pigment epithelium. The pigment epithelium gets thickened and it blocks the choroidal fluorescence in the early phases. But the diseased pigment epithelium gets fluorescein that comes through the choriocapillaries. It seeps through the diseased pigment epithelium and gets hyperfluorescent. At the same time, or a little bit later, Alan Bird and the people in Europe said, oh no, this is from, because these guys had a lobular shape, they thought it was occlusion of the choriocapillaries and the precapillary arteriole that supplied the choroidal lobule. There was some inflammatory things happening around the lumen of that, or around the wall of it, and caused the occlusion of the choriocapillaries, and hence these patients develop these lesions. But Gass's argument was, if that was the case, if the fluorescein was seeping through the surrounding choriocapillaries that were intact and had fluorescein in them, the fluorescein should come in through the sides of the lesion and get hyperfluorescent, and not evenly get hyperfluorescent like we see in this example. Anyway, if Don Gass was here, he would have been really happy and a little bit surprised to see that the OCT shows us that this disease is far more than just the pigment epithelium. It affects not only the pigment epithelium, it affects the overlying outer nuclear layer and the photoreceptor outer segments. It also affects the choriocapillaries and perhaps the superficial choroid. So you can see that those creamy lesions correspond to this opacification of the outer nuclear layer, this kind of ground glass appearance of the choroid and the choriocapillaries. And so everything's involved, not just the pigment epithelium, but the photoreceptors and the choriocapillaries. And the autofluorescence is really interesting. When you see these patients in the active disease, when you catch them really early and you do autofluorescence imaging, you will see that the center part of the lesion is hypoautofluorescent, and that corresponds to where it's blocked by this patch of opacification of the outer nuclear layer. And the pigment epithelium around here is probably hyper sort of metabolic, and so that is hyperautofluorescent. So this you catch only in the early phase of the APMPP. You catch them a week or two later when that outer nuclear layer opacification has gone away. They look like TB serpiginous that I'll show you an example of following on. You can see this appearance in other diseases too. If you catch in TB serpiginous when it's acute fame, you can see this, but this is extremely typical of APMPP. And when it resolves, the outer nuclear layer opacification goes away. There's some little patchy ellipsoid loss, and the choroid and the choriocapillaries sort of come back to reasonable baseline. Unlike what we saw there, this patient has TB serpiginous. You can see some of these lesions are older, and they keep getting these multifocal lesions that can either come up independently or come at the edge of a previous lesion. And you do a fluorescein angiogram, they look exactly like APMPP. The lesion is hypofluorescent in the early frames, and then the ICG is hypofluorescent. And the late frame, the fluorescein shows hyperfluorescence. So it sort of looks like APMPP, but if you do an OCT, the pathology is mostly in the choroid. The secondary changes in the pigment epithelium and the outer retina, but you don't have that opacification. Of course, I do have one example that I didn't put in here because I didn't want to confuse us all. Sometimes if they have it in the central macula and you catch them really, really early, you can see that even in TB serpiginous, but I think that goes away fast. So TB serpiginous is mostly, the involvement is in the choroid. So you can see diffuse thickening of the choroid that you did not see in APMPP, then secondary changes in the outer retina and the pigment epithelium. Again, here, sometimes it could be a diffuse thickening of the choroid, sometimes it could be a nodular thickening of the choroid, and we think that the granuloma is in the choroid. And as it heals, you lose outer segments and the outer nuclear layer is lost and things kind of sink down, as Amani showed us in her patients with type 2 juxtafoveal telangiectasia. And autofluorescence is interesting. The autofluorescence shows hyperautofluorescence in the center where the pigment epithelium is diseased and it's sort of picking up things and eating up probably some of the photoreceptor outer segments. So this is a little different from the autofluorescence that you see in patients with AMD. And autofluorescence is a useful tool to see the response to treatment. Remember, TB serpiginous needs antituberculous drugs for almost 12 months. So you can use that as a tool to monitor these patients and see response to treatment. Active lesions to show hyperfluorescence, hyperautofluorescence. And as the lesion goes with time, the autofluorescence keeps on decreasing. And then that's a good reasonable measure to tell that these lesions are probably inactive now. And here's to compare the autofluorescence and the OCT findings between TB serpiginous and APM-PPE. We already talked about it, but it really tells us the story. Unlike the previous example of APM-PPE, this was given to me courtesy of Lee Jampol. These patients have large lesions, not just in the macula, but they go beyond the macula, sometimes all the way to the periphery in both eyes. And these patients are usually treated with corticosteroids to help suppress inflammation quick enough to get back vision. And the fluorescein looks exactly like APM-PPE. The lesions are hypofluorescent in the early frames, and they stain late or get hypofluorescent. And the OCT features are similar to APM-PPE. The only difference is that the lesions are far more extensive, and they may get new crops of lesions, like we'll see in this example. This patient was put on oral steroids and had these lesions that were somewhat subacute after 5 days of initial presentation. Here's the fellow eye. 5 days later, the lesions look a little bit less active. And this is another week or 10 days later. There's about 2.5 weeks after the patient was started on prednisone, he developed new lesions. So this is relentless placoid or some sort of a progressive APM-PPE. We don't know why these patients get more lesions afterwards. And here's another example where the fellow eye also shows some new active lesions, but they behave exactly, they look exactly like APM-PPE on imaging. And once you treat them, they can have some variable recovery of the photoreceptors and some residual loss. Unlike all of those that we saw, those patients usually are younger, young adults, 20s, 30s. These patients that I'm going to show you next are kind of a little bit later, in the 50s or later. And this patient developed with this creamy lesion in the macula, also had evidence of subretinal hemorrhage. And the left eye had a similar creamy lesion, just limited to the macula. It's not as intense and as juicy as the patients with APM-PPE, and it does not have older lesions like you saw in TB serpiginous. So this one is bilateral, affects the macula, and the fluorescein angiogram doesn't show anything like APM-PPE. There may be a little bit of hyperfluorescent darts, mostly because the pigment epithelium's probably been irritated in those areas. We think the lesion is in the choriocapillaries and the RPE, just under the RPE, and I'll show you that on the OCT. And here's evidence of choroidal neovascularization. This patient, otherwise the lesion is relatively sort of quiet. Sometimes there's a little bit of hyperfluorescence from the pigment epithelium. The subretinal fluid going down here from the CNVM, which is why this area is dark. And the ICG remains dark both early and late. And the OCT is really useful. Here's choroidal neovascularization, but you can see the sort of diffuse ground glass appearance of the choriocapillaries, and the pigment epithelium overlying that is somewhat thickened, but it's not really broken down. So this patient actually had decreased vision in the right eye, mainly from the subretinal fluid, because the left eye had the lesion. He was 20/20, and the left eye, the creamy area was not involving the central fovea. So this is the left eye. This eye was 20/20, but you can see this ground glass appearance of the choriocapillaries corresponding to the creamy lesions, and presence of choroidal neovascularization with subretinal fluid. So this patient received oral steroids and anti-VEGF treatment in the right eye, but oral steroids for both eyes. The OCTA was able to confirm that there was a CNVM there. And autofluorescence is, again, because it's in the choriocapillaries and not much of the pigment epithelium, the autofluorescence is relatively quiet, except where the pigment epithelium is irritated. And here's a little bit hypoautofluorescent from the subretinal fluid. And this patient was treated, and surprisingly, he got back 20/20 vision in this eye. He was treated with anti-VEGF and he was treated with steroids. He did not have a recurrence as long as I followed him for more than 2 years after this. And so I did not have to keep him on immunosuppressives or steroids. The left eye was fine. And this was first described in 2005. There were about 5 or 6 patients. One of them was followed by Dr. Jampol and Dr. Gass. The man would go back and forth from Chicago to Nashville, and I had a chance to see him and take care of him for a little bit. And these patients had multiple CNVM. We knew they were not serpiginous, so they kept calling them, this is sort of atypical serpiginous at that time. Finally, they gave it a name called persistent placoid maculopathy. And their series of patients, so most of them had multiple recurrent CNVM, and it was difficult to treat. This is pre-anti-VEGF era, but now we know that the spectrum has evolved over time. Not all patients develop CNVM, like we saw this one. The left eye did not have CNVM, and some of them recovered vision and the outer retinal elements came back. Many of them developed progressive atrophy, and the role of steroids and long-term immunosuppression is not clear. I think Dr. Jampol believes there's no need to treat these patients with steroids or immunosuppressors, but all of us kind of treat them at least with steroids and anti-VEGF. to tell you how this next example is a contrast to the previous patient that I showed. This patient came in, he was about 59 years of age, and he had bilateral creamy lesions. It was a few days old because he had already seen another retina specialist. I had subretinal hemorrhage in both eyes and choroidal neovascularization. Here's his OCT, which again shows the ground glass changes. He has subretinal fluid from a CNVM in both eyes. I don't know why this one's twisted on the thing, but anyway, both eyes had CNVM. So I did treat him with steroids and anti-VEGF agents. Unfortunately, his vision only ended up being 2200 in one eye, 2100 in the other eye. I actually sent him up to see Lee Jampol because I was worried that he wasn't getting any better. And Lee agreed with the diagnosis, but said that was where it was going to be. So some patients with persistent placoid don't get good vision, but some can. Unlike all of those that we saw, this patient has serous detachments with creamy lesions. And this is very classic, and we all know what this is. This is Vogt-Koenig-Harada disease. And the OCT, I know we would see these patients with bacillary detachment, didn't think much about it, but now since Bailey has written about it, everybody's writing about bacillary detachment. We'd seen all these patients with bacillary detachment for many years and didn't think much about it. I thought it was subretinal fibrin at that time. Anyway, the lesion is in the choroid, but you get secondary serous detachments and lots of inflammatory material that pours out into the subretinal space, causing this sort of a cobwebby appearance. And you can use your RPE map here to see the undulations of the choroid causing chororetinal folds. And if you treat them, the good thing about VKH is you make the diagnosis and treat them with high dose, adequate amount of steroids and treat them long enough with steroids and immunosuppressives, you can cure them. And this is within a few weeks of steroids, the choroidal lesions are completely resolved and the patient's OCTs return to normal. If you do OCTA imaging, you can see these things called flow voids. I don't believe these are areas where the choroid is occluded or choriocapillaries is occluded. It's probably some inflammatory stuff that's in the choroid that causes some of the choroidal vessels to not have flow through them just because they're all congested with stuff and cells around them. And because you can't expect somebody to get back 20/20 vision within 3 or 4 weeks of treating them with steroids when they have VKH if they really had choroidal ischemia. So I wouldn't use the word choroidal ischemia. I think flow voids are just kind of areas where the choroidal vessels are compressed from inflammatory cells around it. And that's what was on this slide. Sorry, I already spoke it. The last disease that we have is this one, which where the macular lesion, if you could see it, you can make the diagnosis. Sometimes the lesion is very subtle, the vision is poor, and the OCT is extremely useful because you've lost photoreceptor outer segments and the vision is pretty bad. And you do a fluorescein angiogram, you can see the hyperfluorescence occasionally well defined as in this example, but most of the time it's ground glass in appearance. But the lack of photoreceptor outer segments is dramatic. And you treat them, make the diagnosis and treat them for syphilis with penicillin, everything comes back within about 4 to 6 weeks and they can get back 20/20 vision. Here's, this was given to me by Emmett Cunningham. Here's another example where the receptors were lost and they came back within a few weeks and they can get back good vision. Some patients with syphilis can also have this unusual findings. Most patients with syphilis have optic neuritis or retinal vasculitis and this placoid lesion, but occasionally the lesions can be in the inner retina, sometimes even pushing into the vitreous cavity like this. This was first described in San Francisco by Emmett Cunningham, Rich McDonald, my partners, and this is seen in men having sex with men. These are superficial retinal infiltrates. Here's another example, a severe example of that. This patient had placoid lesions along with these infiltrates and had vascular changes too. So all of those are sort of classic features of some of these diseases. Hopefully imaging is taught us something else, and now we understand them a little bit better. Thank you.
Thank you, Anita. That was quite a treat to see all those amazing images. You know, I noticed you showed a lot of OCT, a lot of fundus autofluorescence, but not much OCTA. So you don't really need it as much for these inflammatory conditions, right?
Yeah.
Okay. Any questions for Dr. Agarwal? None? Okay, then we'll move on. Thanks so much again to Dr. Dimitra Skandra, who will speak on the gut microbiome and retinal conditions.
All right, quite change of gears. So I hope to keep you awake, even though we're not talking about clinical cases. Okay, so no relevant disclosure. So just an overview. I don't know why. Can I go back? Oh, yeah, I got it. Sorry. Oh. All right. So the overview is basically we're going to go through some general stuff of microbiome, the axis, and then I'm going to focus AMD, ROP, and diabetic retinopathy. So gut microbiota, which are the trillion of microbes that live in our gut, actually now we call it microbial organ. And actually it's the way our lifestyle and environment is talking to our body. So it's affected by diet, by medications, antibiotics, our genetics, the environment that we live. And those little microbes that talk to each other, that talk with our body, and we still don't know exactly how they affect our health and different disease states. And one of the theories is like the secrete of little metabolites that both locally and also by traveling away in the body can affect our health and disease status. These interactions are very complex. So the microbiota affect the host, but also the host genetics affect the microbiota function and the immune system and diet and metabolites. So these are very complex and bidirectional interactions. In order to study these interactions, you need very kind of delicate systems. You can isolate the factors that can affect this relationship. So you can really isolate who causes what, and if it's a causal relationship or it's just a correlation. You can change the microbiome with diet. You can give them antibiotics. You can have the germ-free mice either without any microbes at all, or then colonize them with specific populations. Or you can do fecal material transplants, FMT, and really kind of like specify what microbiota you have in your system. Germ-free, that these mice have no microbes, is considered the gold standard because you can really manipulate and isolate the effect of the microbiome alone without medications or diet. So the first evidence, that microbiome affects our health came from other parts of the body, mostly the GI system and obesity, but then more diseases are coming into play. And in particular, a lot of evidence about CNS diseases and psychiatric disorders in the central nervous system. The first evidence that actually there is a effect of the microbiota on the eye came 10 years ago. There was a first paper from Finland that showed that mice that are regular mice, but they grow up in a sterile environment, or environment is like not as dirty as the regular mice because they're really filthy in the cages. They have different lipid and protein composition in the retina. But that's about it. We didn't know any idea about what's going on. So this field is kind of new. It's only like the last decade, and in particular, the retina, the last 5-6 years, that we really start talking about the relation between the microbiota and the eye diseases. So we know there's a gut-brain axis, and they use these germ-free mice and specialized models to really show that what's going on in the gut affects what's going on in the brain. What about the gut-retina axis? We really have proven it exists, and what's the status of it at baseline? What are the pathways that are affected by the gut-retina axis? And actually, what's the role in the retinal diseases? So very complex disease, need a lot of specialized people. So we put together this team at UChicago to address this. And the first question I want to answer is that the usual way in the science, like you do a knockout mice. Well, the knockout mouse for microbiome is the germ-free mice. So if the mice have no microbiome, how is the retina genes and pathways that affect it? So we sequenced all the retinal genes in mice that have no microbiota, and we found that several important genes for oxidative stress, mitochondrial function, and MP kinase pathway, and energy preservation, in addition to some angiogenesis pathways were involved. And many of these pathways we know they've been involved in the diseases. But RP and choroid are crucial for retinal health, and they have a different kind of cellular composition. So the next question is from the same mice that we check the retina, if you check the choroid, if the pathways of the genes are affected the same. And we're very surprised that actually the gut RP choroidal axis is much more prominent and very different than the gut retina axis. So when you sequence like, again, over 20,000 genes, we have 660 highly differentially expressed genes with very stringent criteria of like FTR and log of C of at least 2, which means like at least 10 times more expression, and VGF, complement factor, NLP3, PTGF, MMP19 among the top 10 genes. And when we did the pathway analysis, very significant angiogenesis, inflammation, cytokine receptor activity, adhesion and chemotaxis pathways were involved. And we know that, and we published this, and then we noticed there was a lot of overlap between the gut retina RP choroidal axis and AMD pathobiology. So could the gut microbiome be the missing link in AMD? We know it has genetics, there's a lifestyle diet, but maybe the microbiome, maybe the way these communicate with each other. So the first very good study about this, kind of like showing it not just what patients have, but kind of understanding what's going on, it came actually from Sapieja's group in Montreal, that he took mice that were given an extremely high-fat diet, like 80% high-fat diet, and this mice basically gave them obesity, kind of like pre-diabetes, and then he'd use laser, he'd use CNV, and he found these mice have like much bigger CNV, they have more microglia, but also have different microbiotic composition. They have like less bacteroides and more firmicutes, so there's two main phyla in the microbiome. And then when he gave antibiotics to the mice, this composition changed and the CNV size went down. So that was kind of the first evidence that, you know, by changing the microbiota with the antibiotics, or maybe it's the antibiotics effect itself, we don't know, but it changed the phenotype. And then he speculated and he showed that these mice, when they had different microbiota from the high-fat diet, they have more inflammation biomarkers, both in the serum and the choroid. And the speculation is this like increased gut permeability in the leaky gut. Many of these inflammatory mediators are spilling out to the blood, and then they make it to the choroid and promote the CNV features. The second very important study came out actually from Alan Rowan's group in Tufts that look at dry AMD features and high glycemic index diet. So they gave the mice super high sugar and glucose diet, like 80% like a glycemic index, and they put these regular mice for this diet for 2 years. And these mice start developing dry AMD features, start developing drusen node deposits, drusen-like deposits, and atrophy, and photoreceptor degeneration, and RP degeneration. And then when they look at the microbiome from these mice, they found that they had less bacteroides, more firmicutes, and more Clostridia, which is a class within the firmicutes. Is that a signal I should stop talking? All right. And then they have different stereometabolomic profiles. And they found different untargeted metabolomics in the serum, like serotonin, trimethylmelin, tyrosine, tryptophan, and some other metabolites. And interestingly, when they switched the mice from the high glycemic diet, the very high sugar diet, to the low sugar diet, they rescued the phenotype. So these mice that were already on a high sugar diet for a year, when they switched them back to the regular low glycemic index diet, they developed more normal phenotype, and the microbiome came down normal. So that's mice. Mice are not human. So the first, some of the first studies about microbiome and AMD patients came out from University of Bern, Martin Zickernagel and Sebastian Wolf group, and they look at a small number of like neovascular AMD patients and healthy controls. And it was a small pilot study, 12 patients each. And they saw even in the small study that indeed these neovascular AMD patients compared to healthy controls seem to have different microbiota composition. Overall, there was like a little more firmicutes, a little more Clostridia, and they're trying to kind of dig down a little bit more. But interestingly, they look at the complement system. So the complement system, I want to talk more about the patients. Basically, they look at mice that have no complement, and they saw these mice have different microbiome. To see what they saw about how genetics of the host can affect microbiota composition in other systems happens also in the eyes as well, and factors that matter in the eye. And actually, mice that have no complement 3, they have also more firmicutes and more Clostridia and less bacteroides. And kind of like that showed that these like mice also have different synthesis changes in the microbiome composition pathways, kind of like showing a little bit of understanding how the genetic predisposition can affect AMD. And then again, as I said, there's the patients that saw some different composition. I'm not going to focus too much on the species because it doesn't really matter. I think when you have small studies with small patients that is so variable, saying that there was like a little bit more of this species or the other, I don't think is really that important as we know yet. So they found some bacteroid species that were more enriched in healthy patients, and some more firmicute species that are enriched in the AMD patients, and there's a lot of amino acid and lipid synthesis pathways affected in AMD patients within the microbes. So it's not only that the microbes are different, it's that the genes and the pathways within the microbes are different as well. And the follow-up study, then I think that was a very interesting study. They associated the microbiota composition with the complement and the genetic risk for AMD. So they found that Clostridia, again, that's a class that you maybe hear a lot, within the firmicute seems to correlate with complement factor H mutations in patients with AMD. Again, kind of supporting the animal data that complement factor can correlate with microbiota composition and maybe affect the phenotype for AMD. So we have some first evidence, but we still have a lot of unanswered questions. Actually, a lot of the dietitian studies have shown that the studies used in previous animal studies is very far off what our patients eat. And actually, a true Western diet is not 80% fat, and it also has both high fat and high sugar. And there is animal diets actually kind of reflect more real the American Western style diet. And so there's what about this diet that is both in fat and glycemic index that is more reflect the real life, the real world Western style diet. Is there a relation between the microbiota composition and AMD features? A correlation is really a causation effect. And can we, most importantly, can we really change the features of AMD by changing the microbiota composition with fecal material transplant as a potential new therapeutic strategy that is very hot right now in other fields and actually is in form of pills, as Dr. Lim mentioned. So, the first question went back to the mice, and we basically created this, we bought this diet that is really true Western diet, has both high fat and high sugar. And we found that this Western style diet that is high fat and high sucrose enriched exacerbates CNV features. After 8 weeks of diet, they have 50% more CNV, more microglia, both within the lesion and also outside the lesion. And indeed, the composition of the microbiota changes. They have more firmicutes, they have more Clostridia, but also we know that they have less of some of the helpful microbiota like Bifidobacterium that we know it's a very important probiotic and a very important anti-inflammatory microbe in our gut. So again, just because something is more there doesn't mean it really drives the disease process. So for that, you really have to start doing some more delicate experiments. So what if in these mice or in these conditions, you just take the microbiome out and you deplete the microbiome, either in a germ-free stouts, like never born with microbiome, or you just take it out right before you induce a disease with high spectrum antibiotics that take it out and just modify the microbiome, they completely wipe out the microbiome. And the answer is great, but in order to do that, you have the model that can actually maintain what you put in the mice, so you take out of the mice there. So the most common model of AMD is a CNV model that many of the studies are using, but it's very hard to do these laser mice with a germ-free. So we develop a special model that you can actually, we modify the way the laser is. We can laser these mice within the sterile hood and keep them sterile up to 2 weeks, confirmed with PCR after the laser. So now we can change the microbiota and we have a disease model that can really keep the microbiota there without changing it by the laser procedure. So microbiome depletion composition, microbiome depletion, either with germ-free or with antibiotics, suppress CNV and IBI microglia almost exactly the same. So either they have a head microbiome or you just take it out before the laser, CNV goes down. And interestingly, it also ameliorates the high fat induced CNV exacerbation. So when these mice are on high fat diet for 8 weeks, either they have a head microbiome or you take it out right before the laser, when they have no microbiome, they have basically the same CNV features, size, and microglia infiltration as if they're a normal diet. So basically kind of like first evidence that microbiome mediates the high fat diet induced CNV exacerbation and the CNV formation. And then the real, I think, proof that this is a driving aspect is that when you put the microbiome back, can you reconstitute the phenotype? So basically we took mice that are germ-free, and then we put back microbiome from regular conventional mice on a regular diet. And then another group with reconstitute microbiome from mice that were high fat diet for 8 weeks and have dysbiosis. And we find that we reconstitute the phenotype that mice that receive microbiota with FMT from mice that had bad microbiota and dysbiosis, they have like almost double CNV and more microglia compared to mice that receive the microbiota that was like from regular diet, conventional. The CNV was exactly the same as the mice that were high fat diet for 8 weeks. So far we focused more about what makes things worse and how can make the disease worse, but can we use this to advantage? So if we give microbiota that have more of the good guys and less of the bad guys, can you actually suppress the disease? And we talk about how it seems like the bad microbiota players are within the firmicutes or the Clostridia, and the good guys that go away when we give bad diet is the bifidobacterium, and it's another microbe called Akkermansia. So in these experiments, we took mice that were depleted either germ-free or antibiotics, and we colonized them we gave them FMT from microbiota that had less Clostridia, more bifidobacterium, and less firmicutes. And we're very, very happy to see that actually that suppressed the CNV formation almost by 50%, and also decreased the microglia within the lesion and around the lesion. Both in the germ-free mice that got FMT and also the mice that were conventionally raised and we just like did FMT as it would be a patient. At the same time we're doing these experiments, a very interesting paper came out that fecal material transplant from young mice, when they were given to aged mice, they suppressed inflammation biomarkers in the brain and the retina. So really opening the space that we should start thinking and talking about FMT and AMD. So what about now the other spectrum? So now we're talking about aged patients. What about really, really young babies, another vascular disease and VEGF-driven? So there's a lot of evidence the last 10 years, again, that microbiota affect preterm health, and it's associated with a lot of complications, prematurity. And actually, there was a lecture by Erica Cloud, that's in Chicago, is a very famous neonatologist, and she was saying how the gut microbiota affect brain development through the IGF pathway, and then when they colonize germ-free mice from microbiota from very premature babies, they stop gaining weight as if they were like premature mice. So this question came like maybe the microbiome could be a missing link in the Atrophy as well, that we know IGF pathway and VEGF pathway is also key. So we put together the team that focus more on the microbiome, and actually we published the first study in the literature that actually early microbiome profile, 28 weeks before even the baby, we know which babies are going to have ROP in very high-risk babies, seem to have different composition than the babies that were also high risk but never developed any ROP. And they were enriched particularly in enterobacteria that within the firmicutes category. And they were specifically enriched the babies that were protected with a lot of metabolic pathways and amino acid and lipid biosynthesis that we know that driven by the microbiota. So again, that was a small study, it was a pilot study, but because it was a prospective study, I think gives you that maybe something there. Luckily, following us, a couple of more studies kind of confirm our findings. There is a group from Australia that they found that basically at admission, infants with ROP have a different microbiota composition as well, and they have different diversity, so different kind of like variety of microbes, and particularly had enrichment in Staphylococcus that we also know it's a pro-inflammatory microbe. Again, small numbers, these are rare diseases. And there's a big study that from China that is a lot of limitations regarding, we don't have a lot of information exactly what they gave to these babies, but apparently they look at like a lot of databases, they give probiotics to premature babies, and those that receive probiotics, they seem to have less ROP and less risk of ROP. But in the paper, read the paper, every single sentence, they don't say what probiotics they gave. So I'm a little, at the back of my mind, I don't know if I trust this data, but it's out there that maybe there's something there, and maybe a way for intervention. Like, if we really find what are the good guys and maybe enrich some microbe of these like very high risk, very sick babies, maybe we could really improve their outcomes. For me, that was a very interesting study, and I think that's the best kind of like causation potentially study. That basically TUDCA, which is one of the bile acids only produced for microbes, and they gave TUDCA, which is a microbial-derived bile acid, to an ROP mouse model, and that's suppressed CNV. And we've seen similar results in our animal data. I don't want to, for the interest of time, but TUDCA is one of these things that like really go down when you give that bad diet, and when you give FMT with enriched microbes, they're enriched also in TUDCA. So we see this effect also in the AMD animal models and in my patients as well, that has got the results. So UDCA and these bile acids actually may be one of the metabolites that suppress angiogenesis, and maybe how that microbiome can regulate ROP. And interestingly, more evidence came out how significantly lower systemic levels of IGF is in germ-free mice, and the mice experiencing undernutrition, and mice treated with vancomycin. So maybe that's why we have these babies that are with a lot of antibiotics. They get more ROP. We know it's an risk factor. And these effects were rescued when they gave fecal material transplant and probiotics for gram-positive bacteria in these mouse models. So again, we don't know yet, there's still more work to be done, but it seems like the small chain fatty acids and bile acids are microbial derived, maybe how microbiome can affect ROP development and possible other retinal diseases. And last, I'm going to talk about diabetic retinopathy and retinal arterial occlusion. I think it's the weakest of all the diseases about evidence we have. I think the evidence so far is more like correlation. We really have no evidence about causation. Basically, they've shown that in 3 different studies, a couple from India and 1 from China, that patients with diabetic retinopathy, they have different microbiota composition than patients that have diabetes without retinopathy. We don't know if this is retinopathy. This is just observational studies. I would take them with a lot of caution. They different microbes that they found less lactobacillus and actinobacteria in the DR patients, different results from a different side of the world. Again, these are very, microbes are very affected by where you live. So when you have a study that is, one is in India and the other is in China, very different environments, very different diet. We don't know if it's an effect of the diet, of the environment, of the disease itself. And they did a kind of like a Mendelian randomization study, and they found that the specific microbes that they may are more associated with the risk for diabetic retinopathy, like Ruminococcea, that are part of the Clostridia, and some of the low diabetic retinopathy family taxa. Again, unless we do causation, we really don't know if this is correlation or not, and there's no really good animal studies to show causation. They also found that patients that have diabetic retinopathy, they have different diversity of microbes. I would say that the 2 good studies about diabetic retinopathy is this one that actually looked at metabolites, and they found that the patient with diabetic retinopathy with proliferative features, they're enriched metabolic pathways for arachidonic acid and also purine metabolism, that we know it's one of the pathways affected the gut-retina axis. And the specific classes of metabolites that are microbial derived, or they found in the fecal species that seem to associate with the risk of PDR. But again, this is just correlation. There is, I think that's the best study we have out there. It's from mice, it's from Maria Grant in the University of Alabama in Birmingham. But basically, so intermittent fasting kind of like suppresses diabetic retinopathy features in diabetic mice, but altering the gut microbiota, and she showed that this is modulated through, again, bile acid metabolites that reach the retina, and they correlate with the receptors. But again, no causation was proven, it was just showing the effects. So similar CRAO, they found that patients with CRAO, they have different microbiota composition than healthy controls. But the interesting point here is that they found increased levels of TMAO, that is a known metabolite associated with atherosclerosis and patients with stroke. So it seems that the microbes that produce metabolites seems to be enriched in patients that get CRAO. That kind of like it's in the conjunction with literature about stroke and atherosclerosis. So we're just starting to catch the surface here. There's definitely more to come. I think that we still have some work to do from a proof of concept, experiments and data, and to show causation and correlation. And we need to understand better about the how. I mean, we see now that probably there is a yes, but we still don't know how exactly this happens and affects the retina. And that way we can really design some interventions. So I want to thank you for your attention, and I'm ready for any questions.
Thanks so much, Dimitri. That's really fascinating. It kind of shows that you are what you eat. And it kind of also substantiates the work of Joanna Seddon and ARED, showing us that these healthy vegetables and green leafy things and less red meat can affect the eye. And perhaps, you know, it's this microbiota, right? The microbes. And so my question to you is, do you recommend probiotics for some of these patients? I know you'd probably have to take it for years and years and years because the effect accumulates, but what about probiotics?
Actually, I don't, because we don't know which strains actually are good and which are bad. Any of the probiotics in the market have not really been tested. We don't even know if they stick. There's actually some papers showing now that whatever you eat, you poop. What I tell my patients is eat well, and actually I send my patients to nutritionists. I think the best way to make sure they have a healthy microbiome is to eat well for now.
You know, I just find this very fascinating because, you know, even looking at like obesity studies, they showed that people who are obese have certain types of bacteria in their feces. And that if you were to take the bacteria in their feces and they put it into rabbits, those rabbits get chubby. Yep. Versus if you take feces from people who are not fat and put them in the rabbits, they don't get chubby. So I think, yeah, there's a lot of stuff going on. Yeah, a lot of stuff to be learned from looking at, as you say, poop. So, I think it's fascinating research. Thank you for sharing with us. Any other questions for Dr. Skandra? No? Okay. If not, we're going to be moving on to another guest speaker. And this time it is Dr. Matt Macomber. So Matt received his college degree from Princeton University, and then did his MD, PhD as an MSTP at Johns Hopkins University in the neurosciences. Then I was fortunate. I got to know Matt when he was a resident at Johns Hopkins and I was a fellow. And then Matt was chosen to be the chief resident at Wilmer. He also did a retina fellowship at Duke working under Macular and was a Heed fellow and a Ron Michaels fellow. In 1996, that's quite a while ago, Matt, he joined the Illinois Retina Associates in Rush University Medical Center, where he rose to become professor of ophthalmology and now is the research director for the Department of Ophthalmology. Matt's been involved in a lot of leadership positions, having served as a leadership development program person on the AAO Council, and I served with him at the council there, so I got to work with Matt there. He was also on the AAO Board of Trustees, the ASRS Board, and is past president of the COS. He currently serves on the board of ISPB. He's received the AAO Senior Achievement Award, the ASRS Senior Honor Award, and 2 Secretariat Awards. And currently, Matt chairs the DRCR Retina Network NEI multi-center randomized trial comparing immediate versus deferred surgery for symptomatic epiretinal membranes. So it's really a pleasure to welcome you here to the podium, Matt. And Matt, Dr. McCumber will be speaking on management of posterior vitreous detachment, something that we all see every day in our clinics. Matt?
Well, thank you so much, Jenny. A very nice introduction. I hope I do better than the last poop discussion. So I'm going to talk about the management of posterior vitreous detachment. I really want to compliment Pollock Patel and John Minkowski are some of our residents. Pollock is one of our PGY4s going into retina. She'll be joining the Retina Institute in St. Louis, and terrific study. Which do I push here? Enter. Enter. Oh, use the mouse. Okay, got it. And in fact, this study was published this month in Ophthalmology Retina with a very nice editorial by Bill Smitty. I want to thank the ISPB for some support for statistical analysis. So posterior vitreous detachment, one of the most common things we see in ophthalmology is also as retina specialists, frequently an urgent diagnosis. Symptoms we're all familiar, flashes, floaters, secondary to process of vitreous syneresis, ultimately involving peeling of the posterior hyaloid from the retina. There's vitreous traction, which can result in retinal tear, retinal detachment. And the management is dilated fundus examination with visualization to the aura. We recommend scleral depression to look for retinal tear, retinal detachment. It's initially recommended on symptom onset. However, subsequent follow-up may vary. Now, the way this project evolved, John Minkowski, whose father, some may know, is there also John Minkowski, who practices outside Baltimore, is the name on the Minkowski-Guyton potential acuity meter. And John really went into cornea, and he really did not want to see these PVD patients for months after they developed their PVD. He was wondering if we really have to do that. It seemed to him like it really wasn't necessary. So I said, well, let's study it further. There were some studies out there, but really no large study looking at when is the appropriate time to follow these patients. So initially we worked in the patients in our eye center of Rush University. And then, we said, this is really not adequate. We should really look at a larger database. So we went to the Vestrum Health database, which is really quite good for this research. So we examined the incidence of complications following posterior vitreous detachment through an extended follow-up. The design was a retrospective observational study with the Vestrum database, which is a database of 65 retina practices from across the country, 316 providers. This was the study design. Started out with 787,000 patients that had PVD in the database. And we wanted to narrow that down to patients that had recent PVD. So first we looked at text query for flashes, floaters. We wanted to make sure that symptoms were not there for more than 4 weeks. We, of course, eliminated some things that could complicate it, diabetic retinopathy, for instance. The key thing when looking at these large databases, these registries, is that you want to make sure you're looking at the population you're interested in, because frequently you include patients that really should not be there. And so you have to look at things in all different ways. We not only looked at, for instance, patients that had flashes floaters and had the ICD-9 code for PVD, but we went all the way down and we looked at text to make sure that they had the word new or acute. So these, we were pretty confident. It dropped the numbers down considerably, but we had a population of almost 10,000 eyes that we're pretty sure had recent PVD. So we looked at complications and there was a hierarchy. We didn't want to count things more than once. So we counted retinal detachment, and then we counted retinal breaks that didn't have detachment, and then we counted isolated vitreous hemorrhage, so those eyes that had just hemorrhage, but no retinal tear or retinal detachment. We looked at presentation within 1 month, 1 to 3 months, 3 to 6 months, and at 6 months. And we looked at a number of different specific factors that I'll go through in this presentation. So first, here's the demographics. So the number of eyes was 9,635, and 25% of those developed one of those complications. And I'll go into more detail about that. If you look, there was a number of phakic and pseudophakic eyes, and actually this was brought up by the journal. How come that doesn't add up to the original number? Well, that's because not every chart documents if the patient is phakic or pseudophakic. So this is something that can enter into the work and makes it a little more challenging. If we look, 64% documented extended retinopathy. Extended ophthalmoscopy. Was that good? Is that bad? I don't know. Not too bad, I guess. Most eyes received laser photocoagulation when they had retinal tear, for instance, with 3% cryopexy. So that seems about right. So here's the complication rate of those of eyes that had a complication. In that 25%, 13% of eyes had isolated vitreous hemorrhage, 16% of eyes developed retinal break or detachment, and 4% of eyes developed retinal detachment following PVD. If we look at the distribution of the complications, we see that most were at presentation. However, within that first 16 months, there was 8% had of eyes of the eyes that had complications had vitreous hemorrhage. I'm sorry, 8% of all the vitreous hemorrhages were picked up late within the 6 months. When we looked at retinal break, 3% of eyes had retinal. This is not. 3% of eyes overall developed retinal break in the postoperative 6 months, and 1% had developed, of all eyes, developed retinal detachment late, so not on presentation. So of those eyes that had retinal breaks, 20% were late, and of those eyes that had retinal detachment, 25% were late. So it looks like you do need to follow these patients out. Now, if we look at some of the factors and how they relate to these complications, for retinal breaks and retinal detachment, there was a higher number of males that had these complications than females. And other studies have actually found that the opposite. And so why is this the case? Well, one thing we could not look in our database was at the extent of myopia. And myopia is more common in males than females. You can't look at that, obviously, because in pseudofakes, retina specialists certainly typically would not document if that patient had a history of myopia in every case. So that may explain why there's different rates, but still, this may be a real thing too. We really couldn't look at it further. And that was both at presentation and in follow-up. The males had more retinal breaks and detachment. Now we've looked at phakic versus pseudophakic. This is interesting. Phakic eyes had a higher rate of retinal break at presentation. However, if you look at the pseudofakes, they had a higher rate of retinal detachment late. So you have to think that if you have a pseudophakic patient, this is one factor where you should look at these patients for longer because they can develop retinal detachment late. Now, the times in pseudophakia, we looked at this, and in fact, what we found was that recently pseudophakic eyes, so within 3 months or so, experienced a higher rate of any complication or vitreous hemorrhage. However, there was no significant difference between retinal breaks or retinal detachment based on early or later after cataract surgery. We look at peripheral retinal degeneration. Certainly, this was a risk factor for retinal break, and this was a risk factor for retinal detachment, both at presentation and in follow-up. If we look at the fellow eye, retinal breaks in the fellow eye, this was also associated with higher rates of retinal pathology in the index eye, and it was even more so if the fellow eye had retinal detachment. So these are all factors which lead you to recommend more careful follow-up when these patients have PVD. Vitreous hemorrhage. The question is, if a patient has vitreous hemorrhage, what is the rate of retinal tear, retinal attachment? How closely should you look for those things in patients that have vitreous hemorrhage as well as PVD? And overall, it's 42% of eyes that had vitreous hemorrhage had retinal break. Most of these, again, are in presentation, but a sizable number were late. And 10% overall, when there was vitreous hemorrhage after PVD, 10% had retinal detachment. And most of these, again, were early at presentation, but about 1 in 10 were late. So this study, the largest sample size ever actually reported, looking at posterior vitreous detachment and late complications. There were multiple retinal practices across a broad demographic. Limitations, retrospective study, an aggregate database. Of course, we all know our colleagues, and we know some of them don't document as well as others. Everybody here, of course, documents extremely well, particularly Jenny. I've seen her work. Quality, consistency, accuracy of documentation varies. The PVD were by symptoms, ICD coding. We had no OCT confirmation. OCT is not a perfect predictor, of course, but it can help in diagnosing PVD or partial PVD. There may be selection bias because these are patients that present symptomatic. Of course, we couldn't look at patients that were asymptomatic because we couldn't determine when the PVD may have started. However, it's still meaningful because these rates probably are higher complications because we didn't look at the patients that were asymptomatic. So in summary, risk factors, we did find male sex, although that's confounded by the extent of myopia, which we could not look at. Certainly, vitreous hemorrhage increases risk of retinal tear, retinal detachment. Pseudophakia within 3 months increased all complications. History of peripheral retinal degeneration or history of retinal break or retinal attachment in the fellow eye. So what's the recommendation? Well, certainly these patients should be followed, and we recommend the usual 4 to 6 weeks for another examination. However, if there are these risk factors, such as peripheral retinal degeneration, retinal break, retinal attachment in the opposite eye, pseudophakia, vitreous hemorrhage, these eyes probably deserve another examination, probably more further out, to detect late retinal tear and retinal attachment. Bill Smitty had his editorial, and the title was "Be Vigilant," and I can only echo that. Thank you. Alice.
Let's get her a mic, please. Thanks so much, Matt, for that really helpful work that you've done. It's really wonderful. Dr. Lyon, Alice?
Yeah, so no, I think this is a really important paper, and I think there are a couple of points I wanted to ask. So we all know that the first scleral depression that a patient has, we don't get a perfect view. And so sometimes they just have to have them come back so I can get a better view, right? So they have a sense of what's going on. So how did you decide based on looking at something if you knew scleral depression had been done? How was that determined?
Oh, that's by coding. If the extended ophthalmoscopy was coded.
So you're only supposed to code. Okay, so then there was a posterior and an acute posterior vitreous detachment.
That's right. So it was at most of those are probably at presentation. That's a new finding. So it's certainly reasonable to document extended ophthalmoscopy.
So that's a surrogate, but I mean.
Right. I mean, we could have looked at text looking at documentation of scleral depression, but probably most people wouldn't list that.
Right, so how do people document scleral? Do you write down, I did scleral depression with a 20 diopter lens and 360-degree depression? How do you write that?
How do I do it?
No, how is that? How do most people document that?
Well, we're supposed to have a drawing, right? At least we do that routinely if there's extended ophthalmoscopy. Many insurers require that. And, you know, good practice would be to document the way it was done, whether it was scleral depression or 90 diopter examination of the posterior pole, because now extended ophthalmoscopy is divided into, you know, looking at the posterior pole versus looking at the scleral depression. Good practice.
Anita?
You know, that's a good point. We didn't look at that if I were. Oh, I'm sorry. The question was, Anita asked if we looked at pseudophakia and age. We could have done that. We can go back to the data set and look at that. That's a good thought. I don't think we looked at that.
Because, you know, my biggest nightmare is all these 50 to 60-year-old myopes who've had pseudophakia in the last 2 years. They all come with large tears or detachments, and they have lattice, and, you know, the cataract guys are kind of pushing for cataract surgery. And
That's right. One thing I didn't mention in the demographics, the mean age, I think, was 62 for PVD. So that's right in the age when cataract surgery would be most concerned.
They were like 20, 25 before the cataract surgery. So basically they do it for refractive purposes and not for visually significant cataracts.
Yeah.
And nobody told them that you may have detachment and go blind.
Yeah. Matt, I have a quick question. This was a Vestrum database, so these are retina practices.
Yes.
So the 25% seems high, probably, right, for all PVDs, right? It's really not 1 out of 4 that have a PVD that get a tear. And these, right, so symptomatic and then selection bias is referred to retina person, right?
Excellent point. Yeah, I didn't mention that, but I thought it may have been on limitation slide. Maybe I didn't mention it. These are retina practices. So yeah, the patients were asymptomatic, never got to the retina specialist. So yes, so the overall rate probably is less because many of these are asymptomatic.
Great. Thank you so much. That was excellent, Matt. And now you're going to give us our second talk, right? So back to back. So Dr. McCumber's second talk is myopia-related macular diseases, treatment and outcomes. A square one. Where is it? You, Patrice, we're trying to see Dr. McCumber's second talk. I'm not seeing it here. It's not that one. Sorry, hang on 1 second.
It was in the link.
It was in the link.
Did she get the link?
Is it
This is
still
the
need
very
to be
big
downloaded?
thing. Did you get the link?
Why don't you look for it?
Or I have it on my computer.
You want to grab it?
Yeah.
Okay. Can we play a video while we're waiting for that? You just have it on a chip.
I can put it on.
Okay. Can we connect Dr. McCumber's computer? Is that going to be problematic? You want to give him this? Sure. Okay. So let's have Dr. Bott give her talk next. Where is Dr. Bott's talk? Is it here? Where is it? No. What happens to? I don't see it. Is it this one? I'm not sure. So we're going to go ahead and have Dr. Bott load her talk. Please load it up here. Okay, and you have to move it from here over here. Thank you. Exit. Okay, again, let me pull it for you here. You can complete it, this one. You got it. So our next speaker is Dr. Pooja Bhatt, who is Assistant Professor of Ophthalmology and Co-Director of the Uveitis Service here at the University of Illinois. She's also the Director of the Medical Student Education and the Associate Residency Program Director. And Pooja's primary focus is on the diagnosis and management of infectious and non-infectious uveitis, as well as other ocular inflammatory conditions. Now, in keeping with a lot of the other speakers, guest speakers this morning, but she's one of our own, Dr. Bhatt also received her MD with honors in India and then came to the United States. She was a fellow with Steve Foster and pre-residency, and then did her residency, followed by a fellowship with Debbie Goldstein at Northwestern. And she conducts research currently on various systemic immunosuppressive agents for ocular inflammation and on herpetic eye diseases, especially herpetic scleritis. In addition, you might not know this, but Dr. Bhatt also is pathology trained. So if you have any path questions, please ask Pooja. Dr. Bhatt, my pleasure to have you speak today on when to use PCR and next-gen sequencing.
Thank you, Dr. Lim. I actually got promoted last year, but thanks so much for having me. My topic for this afternoon is when to use PCR and next-gen sequencing. I have no financial disclosures. So PCR technology has been around since the early 1980s. And quickly, I just wanted to go over just the process. And it essentially involves amplifying a known sequence of DNA or RNA. The RNA samples are transformed into a copy DNA via reverse transcriptase PCR. And the amplified fragments are then used to detect the presence or absence of the DNA product in a given sample. And really, the testing is conducted by combining the DNA sample, 2 DNA primers, DNA polymerase, and DNTPs in a tube. The tube is then inserted in a thermal cycler, and it undergoes denaturation, annealing, elongation. And these steps are repeated multiple times to exponentially increase the DNA product. And the result is these 2 double-stranded DNA segments identical to the region between the DNA primers. And a diagnosis is made if the DNA from the sample corresponds to the known pathogen, or if a given marker for non-infectious disease is identified in the PCR sample. So essentially, PCR is a highly sensitive and specific, reliable and rapid, allows for rapid and reliable detection of pathogens from a small amount of sample. And all sorts of eye fluids can be used: aqueous, vitreous, tissue, cornea or conj, and tear samples. With the standard single target PCR, only a few target DNAs can be detected. And so many PCR assays are necessary if suspected pathogen is unknown, or there are many pathogens that are suspected as candidates. And so it may be difficult to perform many assays on small volume ocular samples. And to sort of overcome that, many types of PCRs are available: nested, multiplex, real-time, broad range real-time, comprehensive, and multiplex strip PCR. And this is one of the papers, initial sort of for ocular diagnoses. This was initial paper that looked at a comprehensive PCR system that used a combination PCRs for diagnoses of infectious uveitis and endophthalmitis. And it really looked at 500 ocular samples and analyzed them stepwise. The first step was a multiplex PCR and a real-time PCR that looked at herpetic viruses and toxoplasma. And then the step 2 was a broad range real-time PCR that looked at bacterial and fungal DNA. And really, they have found high specificity and sensitivities. And so the authors concluded that just a comprehensive PCR system is a good study to look for infectious uveitis and also to sort of perhaps screen samples for infections. A further optimization of this testing was done. Essentially, the optimization was to reduce the time for testing, as well as increase the number of pathogens that can be tested. And so this group from Japan did a pilot study looking at a simple and easy to use strip PCR test. The results were validated and compared with qPCR testing. And essentially it was able to evaluate 24 different pathogens at the same time. And the sensitivity was comparable to qPCR. So really a nice quick test. And the same group actually further modified this testing. And the reason for the modification was they were looking to develop a point of care PCR test that could be done in the clinic or in the OR. And so essentially the strip PCR test needed a DNA extraction step that was time consuming and that consumed a lot of volume of the sample as well. And so the group came up with a new direct DNA amplification reagent cocktail that they added to the strip PCR. This test detects fewer pathogens. However, it really can detect, or the test results are quick. It's about 1.5 minutes. Again, it can be used in the clinic or while the patient is in the OR. A smaller sample size can be used, and really this direct strip PCR was able to be stored for about 12 months. It's available in Japan. I don't think it's available here. So where do we send our PCR tests? Here at UIC, our lab will send it to ViraCore, but you can send it to WashU. There's ARUP, so many commercial laboratories that will do PCR. I think the limiting feature is the sample size, so really important to get enough sample. I will collect aqueous at the slit lamp, so at least 0.1 ml. It just requires another person to pull the plunger back so that I can watch the AC as it detergesses and you can see the corneal fold, so you can ensure there's enough sample. There are some disadvantages to PCR testing. It's not unbiased, so you need a differential diagnosis and you need to think of the organisms that you want tested for. There has to be primers available for those organisms. And really in resource limited settings, there's high risk of contamination. There may be access issues with thermal cycler machines. So not sort of generalizable worldwide. And so one of the newer techniques is metagenomic next-gen deep sequencing. And the principle is essentially nucleic acid sequencing. The sequences are generated in parallel at the same time, and there's rapid sequencing of the entire organism genome. So essentially a mixture of microbial and host-derived nucleic acid is sequenced in a clinical specimen, and the sequencing reads are mapped to a library of reference genomes for taxonomic classification and pathogen identification. And when we do metagenomic deep sequencing or MDS, it essentially involves next-gen sequencing of all material within a specimen. It determines the microbial composition of the specimen and then further elucidates which pathogen could be possibly causing the infection within the eye. And the protocols for MDS use both RNA and DNA sequencing libraries. So there have been really few proof of concept studies in the literature thus far. Most of the work has been from Proctor, and they've looked at essentially infections and infectious ocular fluids. And here's what they have two studies. One of them, again, comparing MDS to PCR, and this is a small group where they looked at 6 patients with uveitis, 3 with infectious uveitis, 1 with immune, 1 with idiopathic, and 1 patient had no uveitis. The 3 out of 3 infectious uveitis patients corroborated with the MDS. The immune and no uveitis samples had no agents identified by MDS. However, the 1 patient with idiopathic uveitis was found to have rubella via MDS, and then the researchers went back and tested the patient's aqueous sample with reverse transcriptase PCR and confirmed the rubella. So in this sort of small cohort, the MDS perhaps was a bit more sensitive. And they also looked at vitreous samples. So they looked at 31 pathogen positive vitreous samples and 36 negative vitreous samples by PCR and compared with MDS. And again, results were similar. In fact, there were 28 of 36 negative samples remain negative by MDS, but MDS was able to sort of report a positive result in about 8 PCR negative samples, which out of 6 of them, they were able to corroborate with PCR, but there were 2 samples that were positive with MDS for infections, but the PCR couldn't corroborate that. There are other applications of MDS for corneal disease, cornea and external disease, and also other branches of medicine. It's been looked at essentially infectious, again, meningitis, encephalitis. So what are some of the advantages? It's a sensitive, unbiased, and comprehensive test for a causative organism. So unlike the PCR, it's not limited by the pathogen numbers that can be tested for at once. And it's not biased because we're not working off of a working differential or available primers. The small volume samples can detect many organisms. It's potentially more sensitive than PCR, at least that's really very small, few proof of concept studies have shown that. Sequencing can give us additional information, so not just about the organism, but perhaps alleles that confer drug resistance, and we could potentially know if the organism is drug resistant at the onset. And it could be a new diagnostic approach where samples that are negative for PCR could be sent for MDS, and we could look for the causative pathogen that way. But there are several drawbacks of this technology. There's overwhelming amount of pathogen information, so a long list of pathogens. There could be background contaminants with sort of results that may be sort of unusual. And the researchers have tried to sort of decrease the amount of contamination with variable success. These assays are not formally assessed, or there's no formal assessment or validity. And then what is the diagnostic threshold? How many reads constitute a positive result? We're not sure about that. And then the sequencing platforms are different. The proof of concept studies from Proctor looked at the Illumina platform, but there are nanopore platforms, different sequencers that are used, and the pricing is different based on the read length, number of reads, processing times. And so the cost could be prohibitive. So where to send for MDS? Are we doing this here? Well, I would say you could, if there's a tough case, you could send to Dr. Doan at Proctor. I have sent it to WashU. Our lab at UIC will send specimens. It does a combination of broad range PCR and NGS. I think I have sent patients with perhaps delayed onset endophthalmitis to them, but not variable success. I know Dr. Chow has sent some there as well. And really commercially available NGS is only, I found ARUP that does that, but not on ocular samples. This is for tumors and leukemias. So in summary, I would say PCR testing is here to stay. They're reliable, validated protocols. It's faster than culture. And really the strip PCR or direct strip PCR may enable point of care testing. Metagenomic deep sequencing is an exciting new technology. It may help in difficult to diagnose cases, may help identify new organisms causing a disease, but it needs validation and further study. Thank you. Thank you.
Thanks so much, Pooja. That was great and very helpful for those of us who see unusual patients and have to figure out what is the bug. Are there questions for Dr. Bhatt? So Pooja, I think a lot of times, sometimes people think PCR is like the magic answer. You don't know what it is. You just said a PCR, but you have to have some idea, right, to direct the lab. And you want to just comment, well,
Sure, and
Matt loads up.
Sure, and I would say really, sometimes your clinical diagnosis, even if you have a high clinical suspicion, especially for herpes and the PCR is negative, you would still want to treat because it's never 100%. You could use it as a guiding tool. Thank you.
Okay, so Dr. McCumber will now speak on myopia-related macular diseases, treatment, and outcomes. Matt?
Thanks, Jenny. Jenny asked me to speak on this topic. And I want to thank another one of our residents, Prithvi Bambika, who's a PGY3 and applying for retina fellowship. So keep in mind, he's excellent. And I have some green in here for St. Patrick's Day. Right. Yeah, I know, I'm McCumber and I didn't wear anything green. So myopic degeneration, the cause is multifactorial. There's been 22 autosomal and 3 X chromosome loci that have been associated. So it's quite complex. Of course, we know there's environmental factors as well. A lot of near work promotes progression of myopia. Myopic degeneration involves atrophic, neovascular, and tractional components. Right now, the atrophy component of this, as the posterior pole enlarges and the tissue thins, we have nothing for this. We now have pegacetacoplan for atrophy and AMD, but we still need something for atrophy in myopic degeneration. There's a photographic classification of this atrophy, which is listed here. But I'm going to talk about things that we actually can do something about. myopic choroidal neovascularization, myopic traction maculopathy, and a little bit by myopic macular hole. Is Dr. Lederman speaking as well about recorded? Got it. Okay, so he'll talk more about macular hole. So for myopic degeneration in general, the clinical presentation, it may be symptomatic, such as decreased visual acuity, metamorphopsia, or asymptomatic. The fundus findings we're well familiar with. There's that atrophy, as I mentioned, both in the macula and the posterior staphyloma, peripapillary atrophy, and we can see lacquer cracks. But it's often difficult to assess the macular pathology just on examination alone. You could be fooled if there's a posterior staphyloma and miss that there may be, schisis of the retina itself. And this is where OCT really has become very, very valuable. Let's first talk about myopic choroidal neovascularization. The presentation might be subtle and would require OCT or fluorescein angiography to confirm. We have to look along lacquer cracks, as that's a risk factor that the CNV, of course, can grow through that area. Foster-Fuchs retinal spot is elevation of the retinal pigment epithelium and is indicative of previous regressed CNV. So we know this happens not infrequently. And the treatment options I'll discuss include laser photocoagulation, photodynamic therapy, and anti-VEGF injections. So first, I'll have one patient of mine, patient LG, underwent laser photocoagulation in 1999. Jenny mentioned I started here in 1996. That's how long back it goes. And we had, the residents and don't appreciate how little we had. All we had was photocoagulation at that time. So this was a patient who had juxtafoveal choroidal neovascularization from CNV, and I laser treated it. This was actually in 2012. So this was the last image I have, because before that it was EMR and it was film, and who knows what happened to that. But what you can see here is the patient had 20/40 for quite a good vision for quite a long time. However, this can enlarge over time. And by 2015, you can see how much bigger it is. And then recently I saw him and his vision has dropped to 2400 because that atrophy went right through the fovea. Fortunately, we have nothing we can do about that. So we rarely use photocoagulation anymore if it's in the macula. Anti-VEGF agents for myopic CNV is now what we use predominantly. The Radiance trial came out in 2014. Compare that to photodynamic therapy, which had come on earlier. It proved that ranibizumab injections were superior to photodynamic therapy. So ranibizumab eyes had plus 10.6 letters, ETDRS letters, compared to 2.2 ETDRS letters at 3 months. On average, the treatment was effective with a mean of 3.5 injections based on disease activity in the first year. And about 63% of eyes did not require ranibizumab injections at 6 to 12 months. So usually I tell patients they need about 3 injections, and often we can follow it after that. So here's a patient who came in last year. She's a pediatric resident over at Rush, 33 years old, minus 12.5 diopters, Visian phakic intraocular lenses. Vision was 20/30 in both eyes, and here's her right eye and her left eye, and she was asymptomatic. That was on July 12th last year. And then just 10 days later, she came in complaining of blurred vision, still 20/30 in both eyes, but you can see there is now this area of hyperreflectivity, deep, which is choroidal neovascularization. Fluorescein angiography, it's very hard to see, but there is a little bit of area here of staining and then ultimately leakage, which is the CNV. She was treated with ranibizumab, 0.5 milligrams. It is FDA approved for myopic CNV. She remained 20/30 throughout. Here's baseline with that deep hyperreflective material, CNV, and you see it melting away at 1 month and then essentially gone at 2 months. And that's all she needed. We've been observing since then. But not all patients come in with lesions that are that small. This is Prithvi actually brought to me just a few weeks ago. 40-year-old woman, minus 10 diopters, presented with 4-week history of blurred vision. Vision had dropped to 20/50 in that eye, and an optos scan, but you can see the hemorrhage in the CNV and the macula. Here's it is on fluorescein angiography. So she started receiving anti-VEGF injections as well. Now let's talk about myopic traction maculopathy. So there's a spectrum of changes that can appear in highly myopic eyes. It's estimated that 9 to 34% of highly myopic eyes have, with posterior staphyloma, have some degree of myopic traction maculopathy. It includes foveoschisis, as we can see here, maculoschisis, you see it extends out, retinoschisis even further, retinal foveal detachment, which you can have here, outer lamellar hole, full thickness hole, and even hole with some localized detachment. Secondary to pre-retinal and subretinal factors and forces. There's a differential. So if the patient is myopic, it doesn't necessarily have to be myopic. We can see schisis as well in all of these conditions: X-linked retinoschisis, S-cone, optic nerve pit, sniffer, and peripapillary pachychoroid syndrome. With myopic traction maculopathy, the pathogenesis, there can be vitreomacular traction, incomplete PVD. There can be ERM, residual cortical vitreous. But as well as you have this deformation of the scleral wall and the posterior staphyloma. And so the inherent forces in the retina, particularly the ILM, I found, prevents the retina from laying on that posterior staphyloma. And then you can get the splitting of the retina involving and developing into that traction maculopathy. Reduced retinal blood flow due to choroidal and RP atrophy is hypothesized as well to play a role, decreasing intraretinal adhesions. So there are a number of different ways to manage this: parsimony vitrectomy, eye advocate ILM peeling. Some people have done it without. Then there's macular buckling or combined vitrectomy macular buckling, or you can observe in milder cases. Preoperative factors that determine outcome: preoperative visual acuity, duration of symptoms, axial length, presence of the foveal detachment, staphyloma height, and preoperative choroidal thickening. So the approach I usually use is parsimony vitrectomy with ILM peeling. It relieves that tangential traction causing the macular schisis and foveal detachment. It's important to peel all the way out to the edge, near the edge of the staphyloma. I find that's what allows it to work the best. Since there is risk of macular hole, I often put in air tamponade. Usually I don't use anything more than that. You can also do a foveal sparing approach, which might preserve some visual function, although generally I don't do that. It might also decrease the risk of macular hole development. So here's one case, patient JL, 63-year-old Asian patient. Most of these patients are Asian. Minus 14.5 diopters, noted blur vision, left eye for 2 to 3 weeks. Here's the right eye and the left. You can see this macular schisis with some subretinal fluid, vision's 20/50 minus. Patient underwent vitrectomy with ILM peeling. And sometimes what you see, the right eye remains the same, of course. Sometimes you see the subretinal fluid worsen initially, but as long as you don't see a break, just watch these patients. This is 3 months out. Patient vision dropped a little bit to 20/70 at 3 months, but then by 1 year, that fluid went away. Vision has improved to 20/25. Patient typically the patients will develop cataract, so then they get cataract by 1 year, and then here's 2 years out and the patient's 20/20. So certainly this can work in these patients. Here's another patient, EY, 62-year-old Asian female, minus 15 diopters, progressive loss of vision in the right eye. You see the macular schisis. There is some ERM as well. That's the opposite eye. 20/80 in that right eye. 1 month post-op, you see as well, the schisis is gone in the macula, still some out peripherally, vision improved to 20/30 plus. It's a better eye. So anatomic improvement, studies have shown that about 79% of cases have improvement in the foveal thickness by 3 months. The inner retina schisis resolves more quickly. The outer schisis is about 82% in 3 months. Foveal detachment resolves in most cases, 90% in 6 months. And then the extrafoveal retinoschisis resolves in most cases, but not always. Macular buckling. Does anyone here do macular buckling? Not done in this country very much. Some people advocate it overseas. It avoids cataract. Most cases have evaluated it for macular hole-related retinal detachment in these highly myopic eyes. One study compared vitrectomy with macular buckling for macular hole-related retinal detachment and showed 93% success rate with vitrectomy. I'm sorry, 93% rate for macular buckling and only 73 to 83% for vitrectomy. These are some of the options that have been described in the literature. You can also do vitrectomy and macular buckling, although one study published showed no difference in visual improvement. There was a faster reattachment rate with the combined approach. Also may prevent atrophying macular holes. I have never done this approach. I think it's technically difficult. So let's go on to talk about myopic macular hole as well. Won't dwell on this too long as Yannick will be talking about macular hole. The management is similar to myopic macular schisis, vitrectomy, gas, sometimes silicone tamponade. I've done that sometimes if it's the better seeing eye. Some patients have very bad myopic degeneration in the other eye, and you don't want to leave the patient without ability to see well in the eye you're operating on. Strongly consider ILM peeling again. For larger holes, you consider ILM flap or amniotic membrane or even a retinal patch graft has been described. So here's a patient, TG, September of 2022, presented 71 years old, degenerative lamellar hole here, but vision was good, 20/40 in the left eye. This is the right eye, 20/20. I just was observing him. However, December, he presented with blurred vision and he developed a full thickness myopic macular hole, relatively small. I used the usual approach as I would use in most patients with macular hole. Vision was 20/150. And this is in just 1 month post-op. Used SF6, oftentimes in these myopic eyes, SF6 lasts long enough. They're often phakic too, so the gas lasts a little longer because the eye is so big. I use 23 to 25%, and here he's 2800, but there's still a little fluid. He's still improving. Here's one other case. This is a 51-year-old man, high myopic left eye more than the right. We see here a macular hole with a myopic detachment, actually, 2400 in this eye. Here's actually the drawing, just a plug that retinal drawings are still of value. I still do them, as Jenny and I were both taught. This patient underwent vitrectomy scleral buckle. I used PFO and peeled the ILM under the PFO. Laser, and this case I use C3FA tamponade because of the inferior fluid. And this is his post-op appearance, 2 months out, 2100. He looked great, did really well. So that was a good approach. You have to use a little more force, drag the ILM a little more horizontally to peel under PFO, but it worked very nicely in this case where there was retinal detachment as well. I'm happy to take questions. Thank you.
Thanks so much, Matt. That was a wonderful review of all the myopic macular complications. I think we have a question. If you give the mic, please. Dr. Agarwal, Anita.
I had one of these on Monday. So when you use PFO, do you have peripheral break also, or the macular hole detachment?
That was the macular hole detachment, right? So there was most of these cases have peripheral breaks. They usually don't detach like that from just the macular hole.
Right. So my patient did not have a peripheral break. So what I had to do was do an fluid exchange, flatten the retina as best as I could, and then put Brilliant Blue and try to peel ILM. So not in the air, just on the fluid. I just did a retinotomy and I sucked fluid, and it didn't completely flatten, but reasonably enough. But it took a lot of effort to cut. So finally, with the flex loop, it was hard to cut. I finally used a 25 gauge bent needle and kind of punctured in a couple. Then I could.
Yeah, I have not used PFO very much for peeling ILM, and I was impressed how well it worked.
Great talk. Do you find that you have to use certain gauges or certain specific instruments to help reach a lot of these very myopic eyes? Because I feel like sometimes that's the trouble, and we've had to special order instruments. What do you recommend?
That used to be the case. I would say, what, about 10 years ago? Oh, sorry, about 10 years. The question was, do we have to use special instruments for these highly myopic eyes? I think it was maybe 10 years ago or so, maybe longer, when the instruments really were too short. And then Alcon lengthened their vitrectomy probe. I use a Tano scraper generally. Flex Loop 2, I think they're all long enough to reach the posterior pole. Yeah, I haven't had a problem. Sometimes we used to, in the old days, used to have to indent the sclera to get there, which wasn't ideal, but now I don't have to do that.
Thanks so much, Matt. So our next speakers will be virtual, and it's Dr. Yannick Liederman, who is one of my valued colleagues. Yannick is an associate professor of ophthalmology and director of the Vitreoretinal Microsurgery Laboratory, and he's also co-director of our Retina Fellowship. Yannick is an MD PhD who did his ophthalmology residency, and then this was followed by a fellowship at Mass Eye in Retina. He joined us here at the Eye Infirmary in 2010 and has an interest in surgery and AI-assisted surgery. So he will be giving two talks. One will be on surgery and one will be on myopic holes. So ideally, we'd like to show the myopic hole to follow Dr. McCumber's talk.
Good afternoon. It's a privilege to be able to be a part of the Retina Symposium, and I'd particularly like to thank my partner and colleague, Dr. Jennifer Lim, for allowing me to take part. I'm going to speak to you this afternoon about advances in surgery for macular hole. The mainstay for macular hole surgery in uncomplicated cases remains pars plana vitrectomy with internal limiting membrane peeling. Most surgeons do use a visualization agent, and in the United States, that still predominantly remains ICG. However, there has been a trend, particularly with improved commercial availability of Brilliant Blue G, to adopt that here in the US, and that is the mainstay outside of the United States. Advances in surgery for macular hole are predominantly for either complicated cases or failed repair. So following pars plana vitrectomy with internal limiting membrane peeling, 2010 saw the advent of ILM flap-based techniques, and we'll be speaking about those, followed by transplantation of the ILM, the next significant change in technique for complex cases involved autologous retinal autografts, and several years later, the use of human amniotic membrane graft has been described, and a number of these adjuvant techniques are in use for complex cases. Ephraim Mandelkorn in Toronto and colleagues described macular hole hydrodissection. This, in principle, is similar to a technique employed in plastic surgery, whereby the edges of a skin flap are undermined in order to facilitate bringing the edges together. And so, in principle, we can use this technique in macular hole surgery by undermining the retina proximal to the hole and separating the adhesion from the RPE, in theory, allowing the edges of the hole to more easily come together. So here's a surgery from Dr. Mandelkorn. Again, the mainstay of this technique is ILM peeling. Here he's using proportional reflux hydrodissection through the macular hole to undermine the retina. A number of colleagues and I use a different approach whereby we inject BSS using a subretinal 41 gauge injection cannula, and this may avoid some trauma to the RPE immediately underlying where the fovea will be. And you can see that aspiration is just recently performed as well at the edges of the hole, has the potential to traumatize the photoreceptors there. The results in terms of closure rates, particularly when you look at stage 4 and large holes, are quite good, as well as improvements in postoperative vision. And you can see for complex cases, such as case number 4 in the column on the left with a flat open configuration, does show a closed hole, although with some postoperative foveal atrophy. Next, we'll move on to internal limiting membrane flap techniques. These were initially described by Sofia Mikalewska and Jerzy Naraki in Poland. Initially, a temporal as well as nasal flap was created, and the ILM was either stuffed into the hole or, as shown here in the schematic, folded to overlie the hole in order to facilitate postoperative closure. And the next modification of this technique involved sparing of the nasal ILM in order to reduce potential damage to the papilomacular bundle and RNFL there. So there have been a number of different ILM-based flap techniques, including semicircular inverted flaps, multi-layer flaps, pedicles, the Texas taco. What all of these share in common is attempting to make the technique of stably creating a flap more reliable for surgeons, as well as, most importantly, ensuring that that flap remains in place over the hole during the postoperative period in order to facilitate closure. In terms of outcomes, we really have a paucity of studies that were randomized comparing conventional surgery with relatively wide ILM peeling with these flap techniques, but nonetheless, in a number of these studies, the closure rate is quite good, and the visual improvements seem to be certainly commensurate with conventional hole closure when we can close a macular hole. Next, let's move on to autologous grafts, first described by Tamar Mahmoud and presented at the veil vitrectomy meeting in 2016. So here's a case that I had of a macular hole that underwent conventional surgery with phagovitrectomy as well as ICG visualization to facilitate the ILM peel, long-acting gas tamponade, and face-down positioning. And you can see that postoperatively the hole remained open with a flat open configuration, resolution of cystoid change at the margin of the holes, and we really don't see any epiretinal residual tissue or proliferation. So all of these are negative prognostic indicators for the success of a subsequent surgery. So this is a case in which I performed an autologous flap. We're using PFO to protect the hole. We can confirm that there's been a wide peel. And here we're using diathermy to delineate the graft in the mid-periphery. These are automated vertical MPC scissors to create a full thickness retinal graft. I've experimented with the sizing of it a fair bit, and you can see here it's oversized such that the diathermized retina, which is presumably not going to be viable, is outside the margins of the hole. We leave it under PFO for 2 weeks, and 14 days later, here's removal of the PFO with the graft still in place. So at postoperative week 1, the OCT on the lower left, we can see that the graft is in place, and that oversized graft is at the margins of the hole and even spilling out over the hole into the epiretina proximal to it. What's interesting is that after about 6 months, you can see that that residual graft, particularly on the epiretinal space, presumably has atrophied, and you can see there's very nice alignment of the outer retinal reflective layers. However, one reason why we may have modest or no improvements in vision is when we look at the outer retina, you can see that there is an outer nuclear layer here where the fovea should be, as expected when we're taking non-macular or non-foveal retina and using that to create a graft. So while some of these patients do make some modest gains in vision, and in spite of the fact that the hole looks very nicely closed, it's difficult to see how patients could regain foveal vision. Other techniques include anterior and posterior capsule flaps. This is in order to create a scaffold on the inner retina, presumably to allow these holes to close. And finally, we'll move on to amniotic membrane grafts. This was described by Stan Rizzo and his junior colleague as a method, again, to facilitate hole closure. So here's a case from Dr. Mandelkorn in Toronto. He's using reflex hydrodissection to undermine the edges of the hole. And a technique that is fairly commonly used involves taking the amniotic membrane graft and then inserting it into the hole. Here it's being unfolded, again overlapping the margins of the hole, but it's actually being inserted into the subretinal space. One of the limitations with this technique is that that graft is going to permanently remain in the subretinal space, and as you can see on the postoperative OCT on the right, while there is hole closure with very thin retina there, vision is going to be limited by that graft remaining. So I like to use an alternate technique. Here one example is demonstrated by Flavio Rezende, my friend and colleague in Montreal. There's another publication forthcoming from Ray Iezzi at Mayo, from whom I learned the technique that I perform, whereby the graft is placed on the epiretinal space, relatively large graft to allow the hole to close underneath it. So here's a case I had preoperatively with elevated edges, post-operatively flat open failure, for which I performed amniotic membrane grafting. And you can see here's a relatively large graft overlying the hole under silicone oil to help keep the graft in place. You can see 1 month post-op. The graft is hyperreflective on the inner retinal surface, but deep to it, the macular hole is closed. So outcomes of some of these techniques, autologous ILM transplantation, good closure rates in appropriate cases, autologous retinal transplantation, even with the best studies, while we do have very, very good hole closure rates, the vision improvement can really be relatively modest. In nearly half of patients, vision remains stable. Anterior and posterior capsular plaques, some of these have pretty good closure rates, although we have very few studies to really evaluate that technique. Amniotic membrane grafts, we have several studies with very, very good closure rates, and some of them with fairly impressive improvements in vision, and more studies forthcoming. And I'd like to close by speaking a little bit about the CLOSE study group classification. So this is part of a systematic review of surgical techniques for full thickness macular hole in 1,135 eyes. One of the valuable elements of this paper is that they propose a new classification scheme trying to look at some of the prognostic factors influencing outcomes. So they stratify holes into large, XL, XX, and giant, and we'll go over those in a minute, as well as some of the relevant OCT biomarkers that portend success or failure. And some of the top-line conclusions include for minimum linear diameter holes of between 535 and almost 800 micrometers, likely ILM flap techniques should be sufficient to improve closure rates. But for holes with an MLD of 800 micrometers or greater, some of those more advanced techniques, including human amniotic membrane, BSS hydrodissection, or autologous retinal autografts, confer the best closure rates. So here's that stratification: small, medium, large. XL is 550 to 800. XX holes that were 800 to 1,000, and anything greater than a millimeter was giant. And again, some of the prognostic factors include whether there's a cuff of fluid or whether the hole is flat open, edema at the edges, whether there's VMT or epiretinal proliferation. All of these things likely influence hole closure. So in conclusion, the CLOSE Consortium offers us a novel classification scheme as well as a management guide for failed full-thickness macular holes. The variety of ILM flap techniques may increase closure risk in selected at-risk cases, but for the cases at highest risk of failure or reoperations, either autologous retinal autografts, capsular flap techniques, and amniotic membrane grafts are probably of the greatest benefit. And lastly, I'd like to thank my colleague, Dr. Nitin Chowdhury from Toronto, for sharing a number of these slides, particularly the excellent overview of the literature and some of the early techniques. Thank you again for allowing me to be part of the Retina Symposium. All right. Well, it's a wonderful opportunity to be able to give another talk, and I'd like to thank Dr. Jenny Lim again for allowing me to be part of the Retina Symposium this year. And shifting gears from pure clinical surgery, now we're going to be talking a little bit about some research efforts in our laboratory group, and I'm going to be discussing surgical guidance in ophthalmic microsurgery using deep learning and computer vision. These are my disclosures of note. You'll see some technology from Alcon, of which I'm a consultant, but they have not supported any of this research. But it is important to point out that I am a co-founder of Microsurgical Guidance Solutions, which holds intellectual property for some of the technologies that I'm going to be discussing with you today. So I'd like to begin with an examination of our microsurgical environment to try and frame opportunities for digital enhancement. In our current microsurgical paradigm, the surgeon controls the instrumentation and receives feedback in turn, and likewise interacts with a visualization system. A feedback loop allows for direct interaction of the surgical instrumentation with the visualization system. This approach is increasingly used in consumer automotive, rail, robotics, civil, and military aviation applications. And this kind of imaging instrumentation feedback loop confers the potential for greater precision and safety. Let's begin with a working definition of surgical guidance from a surgical data science perspective. The first component is integration of information from imaging modalities, our instrumentation, and the surgeon. In our case, data analysis is executed using a deep learning neural network and computer vision, followed by output to the surgeon and instrumentation that must be able to enhance instrument performance or support surgical decision-making in real time. This is an overview of our technology pipeline for vitreoretinal surgery. Imaging is acquired from a commercially available digital stereo microscope or optical microscope with HD camera, and a neural network performs image processing. Then a set of phase-specific digital surgical tools is activated accordingly. The network output is then injected back into the surgical frame or relayed to our instrumentation. So let's proceed with an overview of some of the tools we've developed for cataract surgery. This is an example of surgical guidance during capsulorhexis. Contrast equalization improves visualization of the surgical field and appreciation of the margin of the rhexis. The faint inner yellow circle provides a template to facilitate the execution of a circular, well-centered, and symmetric rhexis of the desired diameter, which can be customized by the surgeon before or during the procedure. During nuclear disassembly, surgical guidance tools detect and track lens particle motion as a surrogate of turbulent flow, project a tactical safe zone, and quantify the extent of exposure of the posterior lens capsule. We evaluated the performance of our algorithm by comparing the results of phase identification against the gold standard, phase identification via manual curation of surgical video by ophthalmic surgeons. The F1 score combines recall and precision to yield an important measure of network performance, which in this case was well above the 90th percentile for each of the defined surgical phases. So let's conclude with pars plana vitrectomy and surgery of the posterior segment. We'll discuss 4 categories of digital tools: AI-mediated enhanced visualization, surgical templates, tissue and tool tracking, and laser delivery. So here's an example of image enhancement. The video on the left demonstrates suboptimal visualization from media haze. AI-mediated image processing yields an improved view of the surgical anatomy. Note the retinal vessel immediately proximal to the forceps tip. It's difficult to discern in the unenhanced image, representing a potential source of complication during peeling. Similarly, during cataract surgery, image enhancement shown on the right improves visualization of the margin of the capsulorhexis in comparison with the unprocessed view on the left. This is a sample of tool and tissue tracking. The locations of the vitrectomy probe, optic disc, and macula are being tracked in real time. The labels are shown only for network performance demonstration purposes. Instrument and tissue tracking is performed in the background. In this surgery for complex diabetic retinal detachment, the macula, optic disc, laser probe, and a retinal break are simultaneously being identified and tracked by the network. Laser spots at the margin of the break are also identified as they are applied. This serves as the foundation for semi-automated delivery of retinal laser. Note that in spite of compromised visualization associated with surgery under air, the network maintains excellent performance. And here's an example of the capability of our network to allow for semi-automated delivery of PRP laser during vitrectomy. A customizable template for scatter laser is generated, sparing the disc, arcade vessels, macula, and other critical anatomy. As the surgeon sweeps the aiming beam across the target area, the network identifies when there is overlap between the aiming beam and template and then delivers a laser spot. The individual spots are registered as noted by the template points changing from black to green. This feedback loop to the laser delivery system allows for rapid, semi-automated PRP. The laser is only activated when there is overlap with the template, allowing the surgeon to continuously treat the intended target area and still achieve the desired pattern and spacing of spots. AI-based collision avoidance methodologies have outstanding potential in VR surgery. The VitProbe, detached retina, and area of scleral depression are tracked, and a proximity alert is provided when the cutter is at risk of interacting with mobile retina. While this may not be necessary feedback for the experienced surgeon, the real value in these data is the potential for feedback to the fluidics engine and automated modulation of fluidic parameters to improve performance and reduce the likelihood of iatrogenic retinal breaks. Some of our data was published in Ophthalmology Retina, entitled Feature Tracking and Segmentation in Real Time via Deep Learning in Vitreoretinal Surgery, a platform for AI-mediated surgical guidance. So you can see the area under the precision recall curves, both for detection and segmentation of relevant features, is quite strong for the vitrector tool tip, membrane forceps tip, the endolaser, optic disc, fovea, and retinal tear, as well as detachment. Detecting fibrovascular proliferation was slightly less robust, but still quite reliable, and the area of greatest challenge was monitoring the entire area where endolaser spots had been previously applied. So in summary, we've developed an automated pipeline that applies surgical guidance tools to facilitate intraocular surgery in real time using deep learning and computer vision. We've achieved effective instrument and tissue localization using region-based convolutional neural networks. We've been able to enhance visualization of the surgical field. We've implemented phase-specific surgical guidance tools, and we've achieved real-time performance. I'd like to acknowledge my partner and PhD student, Raj Nespolo, as well as the rest of our academic research group. Thank you again for your attention today.
Dr. Michael Heiferman is our newest faculty member at UIC, having completed his college medical school and residency at Northwestern, and then coming to join us as a retina fellow at UIC. He did such a great job, we decided to keep him as faculty, but first sent him off to work with Prithvi Murthanjaya at Stanford for an ocular oncology fellowship. He is now an assistant professor of ophthalmology here, and he is also one of our ocular oncologists. His research interests include studying ocular tumors to identify novel therapeutic targets and improve prognostication. He's also interested in using advanced imaging techniques and AI to study retinal diseases and ocular tumors. And Mike gave us that really nice presentation on ocular lymphoma.
Hi, everyone. I'll be talking about the diagnosis of intraocular lymphoma. I have nothing to disclose. I'll start with a case and then we'll go through the differential diagnosis. We'll discuss the classification of lymphoma in the eye, epidemiology, clinical features, and then we'll spend a lot of time on the evaluation. So we'll start with a case. It's a 65-year-old female who presents with blurry vision in her left eye. She has no medical or ocular history. On exam, her left eye vision is 20/70, and she has vitreous cell in both eyes. The differential diagnosis for vitreous cell is quite broad, and we'll use a detailed history and physical exam in order to focus that differential. So for our patient, this is a color photo montage of the left eye. The media is hazy. There are yellow, creamy lesions in the mid-periphery with overlying pigment changes here. The OCT horizontal raster through the fovea of the left eye, demonstrating vitreous cell as well as hyperreflective material in the subretinal space. Fluorescein angiography demonstrating early blockage and late staining of these yellow lesions with overlying blockage from the pigment changes. So this presentation is most consistent with the diagnosis of intraocular lymphoma, and it's important to be precise with our classification for this disease entity. Most often, intraocular lymphoma is related to metastasis from systemic lymphomas. However, when we're using the term intraocular lymphoma, we often mean primary CNS lymphoma ophthalmic variant, which was previously known as primary vitreoretinal lymphoma. This is most often a B-cell lymphoma, specifically a diffuse large B-cell lymphoma, and this is rarely a T-cell lymphoma. Another diagnosis is primary uveal lymphoma, which is much less common, and is classically a MALT lymphoma. Primary CNS lymphoma that presents in the eye has CNS involvement in about a quarter of cases, and so a careful review of system for neurologic findings is important in this disease entity. Almost all patients with CNS lymphoma in the eye go on to have CNS involvement by 3 years, which is the reason that the nomenclature was changed from primary vitreoretinal lymphoma to PCNSLO. Primary CNS lymphoma at presentation has ocular involvement about a fifth of the time, and overall both these entities have a very poor prognosis with a 5-year survival around 25%. Primary CNS lymphoma makes up about 1% to 2% of all extranodal lymphomas and 3% of all CNS tumors. The incidence has tripled over the past 20 years, and it's not well understood why this is the case. On average, they present at age 40 to 50 years old with a female predilection, most often bilateral presentation, and the time of diagnosis from the onset of symptoms is quite prolonged, which is why the workup and evaluation of this entity is so important, which is what we'll talk about for the rest of this discussion. Ocular screening is important in CNS lymphoma, with the patients being evaluated at the time of presentation with a fluorescein angiography, as well as eye exams every 6 months or when they develop ocular symptoms. A biopsy is not always necessary if they have known primary CNS lymphoma and classic eye findings of ocular involvement. However, it's important to maintain a high level of suspicion for viral retinitis, as these patients are very often immunosuppressed. The classic clinical features of primary CNS lymphoma in the eye include a mild AC reaction, classically homogeneous large vitreous cell that can be clumped or in sheets, as well as retinal vasculitis that's much more mild than other uveitis, and then the creamy or yellow infiltrates in the subretinal or sub-RPE space are very typical. The workup is very involved and is one of the main reasons for the delayed diagnosis of this disease entity. An MRI of the orbits and brain with and without contrast are used to evaluate for CNS involvement at the time of presentation. A PET CT of the whole body is used to evaluate for adenopathy, given the high rate of systemic lymphoma metastasizing to the eye. A testicular ultrasound and a high volume LP are also used to evaluate for lymphoma elsewhere prior to considering a vitreous biopsy. The next step in this evaluation is a vitreous biopsy, in which communication between the ophthalmologist, pathologist, and laboratory are very important. Coming up with a thorough plan, including how the tests will be ordered and how the specimens will be handled after the biopsy, is very important to do the day prior to the vitreous biopsy. In terms of technique, a syringe is attached to the vitrector and 1 to 2 cc's of undiluted vitreous specimen are obtained. The vitrector is removed from the eye and then fusion's unclamped. Additional diluted specimens are then taken, and in general, you want to target the areas of prominent vitreous cell while performing the biopsy. There are many tests available to evaluate a vitreous biopsy specimen to evaluate for intraocular lymphoma. Cytology remains the gold standard to actually look at the cells under a microscope. However, flow cytometry is a critical test as well. There are a number of molecular tests that are more recently used, and they come with various sensitivities and specificities depending on how they're used. MYD88 is an oncogene that drives many B-cell lymphomas. A specific missense mutation leads to the activation of NF-kappa B, and this is important in the pathogenesis of many diffuse large B-cell lymphomas, and particularly in primary CNS lymphoma. This test has a very high sensitivity and specificity, and it's the very high specificity that has led to increasing practice to take an aqueous humor specimen to test for MYD88 prior to the day of vitreous biopsy. IL-10, IL-6 ratio is used to differentiate from causes of inflammation. IL-10 is an immunosuppressive cytokine secreted by B cells, while IL-6 is a pro-inflammatory cytokine secreted by macrophages, and T cells. So a ratio of greater than 1 suggests intraocular lymphoma, while a ratio less than 1 suggests inflammation. A few days before the biopsy, I'll send a plan that looks something like this to the pathologist, laboratory, and operating room staff in order to clarify any of the details needed on how to handle each specimen. This was a representative plan for a patient with the highest suspicion for intraocular lymphoma. You see I'm prioritizing MIT88 in the undiluted specimen, and it's only in the second diluted specimen that will get cultures as well as viral PCRs. This will change depending on my level of suspicion in other cases. Unfortunately, the vitreous biopsy does not always come back positive, and in those cases, we have to consider our options, including taking a biopsy of vitreous from the other eye and used additional molecular testing in those cases. We also consider subretinal biopsy and aspirating the material from the subretinal space, and then also taking a complete chororetinal biopsy is another option. Once we have a diagnosis, the management can include intravitreal methotrexate and Rituxan, as well as external beam radiation and systemic chemotherapy. It's critical to closely monitor these patients because recurrence is the rule. Takeaway points from this talk: intraocular lymphoma is an important masquerade syndrome for uveitis and completely responsive to steroids. Patients with primary CNS lymphoma should be monitored closely for the development of ocular involvement, and communication with pathology and the laboratory is critical to optimize the chance of a diagnosis at the time of biopsy. Thank you.
Hi, I'm Bill Mueller, and I'll be speaking on the management of small melanomas and ocular nevi. Thanks to everyone for partaking in the Retina Symposium, and of course, this will be the concluding talk. I hope everyone's having a great day. I have no financial disclosures. When we talk about pigmented choroidal lesions, we recognize that roughly 2% of patients have a choroidal nevus, defined as an elevated area in the choroid, located pretty much anywhere throughout the choroid, varying degrees of pigmentation. More common in Caucasian patients, and of course, the degree of pigmentation is quite variable from patient to patient. A number of things looked at in terms of prognostic factors: the patient age, the size of the tumor, including both the basal dimensions, along with the overall thickness of the lesion. Most times we define a nevus as up to 2.5 millimeters, and beyond that, more concerning pertaining to a melanoma. We're concerned about the location of the tumor within the eye, as well as the anterior and posterior border of the tumor itself. Of course, for choroidal nevi, we don't really have histopathology, but yet histopathology plays a key role in terms of prognostic factors for life and perhaps spread of the tumor as well. When we talk about nevi, it's important to make the distinction between this entity and many other things in the differential diagnosis. The number one entity, of course, is that of a melanoma. We recognize that with echography and indirect ophthalmoscopy, we can be quite certain in most cases, but in that range of 2.5 millimeters or less in thickness, we tend to call these lesions nevi, and when it's greater than that, we get concerned more for a melanoma. Other things in differential diagnosis are listed on this slide, and most importantly, a melanocytoma of the optic nerve is quite difficult to define sometimes. These lesions tend to be more jet black in appearance and oftentimes occupy a portion of the optic nerve and rarely become thicker than 1 millimeter or 2 in overall thickness. Other entities in differential diagnosis include congenital hypertrophy throughout the pigment epithelium, or CHRPE. These lesions, of course, are flat, jet black in color, oftentimes have lacunae. In contrast to nevi, these have a very distinct border and don't blend into the surrounding choroid. Other entities include things, as you see here, such as the congenital simple hamartoma, a jet black lesion. Looks like it's jumping out of the pigment epithelium, oftentimes located in the juxtamacular region, and quite easy to distinguish from that of a nevus. But recognize you have to make the distinction between all these entities and that of a choroidal nevus. I mentioned earlier that the clinical examination combined with echography yields about a 99.7% diagnostic accuracy when we look at a nevus and or a melanoma, accepting the challenge between differentiating between a larger nevus and a small melanoma. We look for signs of tumor activity. If there's been documented growth of lesion, that makes us more suspicious of the lesion being a melanoma. Look at a breakthrough Bruch's membrane, presence of subretinal fluid, relatively sparse drusen, and ill-defined orange pigmentation. of course, being lipofuscin. If that's relatively absent, it tends to imply a more active tumor. Yeah, we really can't use these factors to correlate with overall prognosis, but it makes us suspicious that the lesion may be more of that compatible with a melanoma. There are a number of things we look for in terms of trying to predict tumor growth. Tumor thickness greater than 2 millimeters. If there's the presence of subretinal fluid. If there's symptoms of visual loss, vision of less than 20/50. The presence of orange pigmentation or the basically lack of orange pigmentation. If there's hollowness on ecography, any base dimension greater than 5 millimeters, all are things that we look for in terms of monitoring tumor growth. We also look at risk factors for transmission from even into melanoma, and in fact, I just mentioned in the previous slide, are present at 1 year, roughly 1.2% will transform, by 5 years, roughly 6%, by 10 years, roughly 14%. So as you can see, these lesions do have a tendency to be a precursor of a melanoma and can grow into a true melanoma. Highest risk factor, the highest hazard ratio. I just mentioned in the previous slide are present at 1 year, roughly 1.2% will transform, by 5 years, roughly 6%, by 10 years, roughly 14%. So as you can see, these lesions do have a tendency to be a precursor of a melanoma and can grow into a true melanoma. Highest risk factor, or the highest hazard ratio, is associated with tumor thickness of greater than 2.0 millimeters. And one more time, looking at risk factors for growth, the patient has 0 risk factors. At 5 years, the risk of transformation is roughly 1%, increasing to greater than 50% if the patient has 4 or more risk factors. So keep in mind that when you monitor these nevi, watch for these factors as listed here, because they certainly can help predict the chance that the lesion may grow into a true melanoma. So summarizing this issue, choroidal nevi are scored according to these 6 risk factors as listed. The tendency to treat earlier, especially since the rate of transformation of a nevus into melanoma is very substantial. And keep in mind, though, that treatment does not come risk-free, especially that of visual loss and or inducement of radiation-induced maculopathy or retinopathy. If we do treat a patient, what are the options? Well, we can certainly observe. We can use laser. We've done that in the past, though it tends to induce sometimes additional tumor growth. We don't do that too commonly. It comes down to using brachytherapy in the form of iodine-125 or rarely ruthenium-103 or that of charged particle radiation. Other treatment options, of course, are available for large lesions. These really are not employed for most nevi. And other therapeutic options are listed here once again, generally for large lesions. But I will point out the bottom issue, that of brachytherapy when employed for lesions in the macular region. Oftentimes we do employ bevacizumab concurrently in an attempt to diminish the risk of radiation-induced complications. I'll come back to this shortly. Brachytherapy, of course, is the mainstay of therapy. Iodized radiation in the form of a plaque delivered from a point source, most commonly that of iodine-125, occasionally ruthenium-106 in foreign countries. And then once again, these are the majorities utilize are with I-125, a low-energy gamma emitter. Advantages, of course, are with brachytherapy, the preservation of at least some degree of vision, hopefully diminishing the risk of transformation into a melanoma. And keep in mind that there's no apparent increase in mortality in retrospective CH-MAS controlled studies. Complications are quite numerous. There can be tumor unresponsiveness, there can be optic neuropathy, cataract, neovascular glaucoma, even scleral necrosis as pictured here. Most commonly, when we treat lesions that are small, we're concerned about radiation-induced optic neuropathy, retinopathy, and or maculopathy. We see here in this photograph an image of a tumor just off the edge of this photograph temporally. This patient developed radiation-induced ischemia and edema, did respond partially to brachytherapy with bevacizumab, but still had a diminished visual function due to the macular ischemia. A number of studies have been done combining brachytherapy with bevacizumab, published starting back in 2019 and even a bit earlier, looking at starting the anti-VEGF therapy at the time of the treatment itself, not treating every month, but every 3 or 4 months. Patients at the greatest risk for retinopathy were those patients who were younger, less than 50 years of age, had larger tumors and those with more posterior in location close to the optic nerve and or macular region. I should point out also that the Diabetic Retinopathy Clinical Research Network, Protocol AL, is looking at a study patients undergoing brachytherapy for a melanoma, randomized 1 to 3 arms out of sham, faricimab, of course, which is the Ang2 VEGF-A blocker, and every 2 months. age are larger tumors than those with more posterior in location closer to the optic nerve and or macular region. I should point out also that the Diabetic Retinopathy Clinical Research Network, Protocol AL, is looking at a study, patients undergoing brachytherapy for a melanoma, randomized 1 to 3 arms out of sham, faricimab, of course, which is the Ang-2 VEGF-A blocker, given every 2 months, Reluvian, a fluocinolone steroid implant given every 18 months, looking at visual outcomes at the end of a 3-year time frame. Of course, this is ongoing right now, and there's no data to report at the present time. So are there alternatives to observation versus brachytherapy? Well, the answer is yes. Aura Biosciences initiated a clinical trial back in 2017, utilizing a novel infrared dye conjugated virus-like nanoparticle injected in the vitreous cavity of eyes with small melanomas. This agent binds to the melanoma surface heparin sulfate, light activation with a photosensitive drug, and was employed in either a single fashion or repetitively. It has a dual mechanism of action, inducing tumor cell necrosis after light activation, and also works as an immune-mediated tumor cell killer. The initial trial, when this was injected intravitreally, showed promising results: visual preservation in roughly 92% of eyes, local tumor control 2/3 of eyes. Yet the problem was inducement of intraocular inflammation in roughly 85% of eyes. This led to a switch in the delivery system from intravitreally to a suprachoroidal fashion. The study was reorganized. The agent is listed here. Same agent, of course, delivered the drug suprachoroidally. This eliminated the vast majority of inflammatory responses. Phase 2 data was reported at the AAO in 2022 and the Mac Society in 2023, just last month. 9-month data showing tumor control with preservation of visual function after 3 cycles of either single or ascending doses of the agent. 20 patients were treated. It's a relatively small number. Follow-up only at 9 months. But keep in mind, these patients had shown previous evidence of tumor growth, even though these are all small tumors. There felt to be a risk of visual loss, so stabilization at the 9-month time frame was felt to be a very favorable outcome in this relatively small study, which of course is still ongoing as well. So in summary, it's important to be able to recognize the typical distinguishing features of small melanomas and be aware of risk factors for growth. I pointed out the 5 or 6 risk factors that we look for. While there's a tendency to treat smaller tumors more aggressively, keep in mind that complications of conventional brachytherapy treatment and or charged particle radiation include radiation maculopathy, retinopathy, and or visual loss. And keep in mind, of course, that these smaller tumors that are located close to the optic nerve and or macula have a greater risk of these complications. Early results of the data from the suprachoroidal delivery of the AU-011 clinical trial are encouraging when delivered suprachoroidally from a tumor control standpoint and visually as well. Recognize the numbers are relatively small, only 20 patients in the most recent trial, and the follow-up has only been 9 months, but the tumors appear to have been stabilized in terms of stopping further growth with preservation of visual function in 19 of the 20 patients who were in the relatively small study thus far. Of course, we need more time and longer follow-up to further solidify these results. So I want to thank Dr. Lim and colleagues for being able to partake in the Retina Symposium here in 2023, and this will conclude the conference once Dr. Lim wraps things up, but thank you very much.
We just heard from Dr. Bill Mueller, who is the Cless Family Professor of Ophthalmology and Vice Chairman for Faculty Affairs, Director of the Vitreoretinal Fellowship Training Program, one of the co-directors, and also Director of Ocular Oncology. He is a UIC Distinguished Professor and has received the Life Achievement Honor Award from the Academy, the Gas Medal, the ASRS Founders Award, and is a past FARVO as well as past Macular Society President. So we heard Bill give us that nice talk on small ocular tumors. And now, last but not least, we're going to hear again from Dr. Larry Ulanski. Larry had to leave to do a case because he's on call for the group. Thank you very much for doing that for us. And he will now be giving the last talk of the day, which is recognizing pentosan polysulfate and macular lesions. Larry?
Thank you, Jenny. I'm going to change the share and let's see if this works. Okay. Unfortunately, some of you guys missed the grand first talk. We had a nice little video chat with one of our fearless leaders of our country. It was kind of fun. That's the first time I've ever embedded a video in my presentation. I worked all night just to make that work. Recognizing pentosan polysulfate and other macular lesions. This is going to be a very short talk. I know it's the end of the day and everyone wants to eat. So my disclosures, again, I'm a federal employee. Have pity on me. I have no financial interest. I can't even take a pen. So pentose polysulfate. Pento, I'm thinking pentagon because I'm a federal employee, something, 5-sided. And polysulfate, well, it sounds like there's a lot of sulfate in that thing. Almacron is the name of the drug as far as trade name. And Zycosan is the horse dosing. And apparently this is very popular for racehorses for osteoarthritis, and they really dose them up with that. So interesting little tidbit. It's indicated for the relief of bladder pain and discomfort associated with interstitial cystitis. It creates a protective coating along the bladder wall to prevent damage, and it almost kind of like relines it, reducing fluid permeability. It's like a natural aminoglycan that lines your bladder wall. So it's a very interesting thought process. You can both take it orally, and you can also do it through a bladder infusion. So I don't know why anybody would want to not have it through the bladder infusion and take it orally, but we'll see why they might not want to take it orally. The other names for it are Hemoclar. I've heard that one before. Anthrocon, Fibrase, Fibrocid, Thrombosid, SP54. The veterinary names are in yellow below in case you have somebody that really wants to, hyperdose. Silvet, Pentosan, Zydax. It was approved in 1996, so it's been around for a while. And again, there is a lot of sulfur. I don't know why they say polysulfate because there's only 4. There's a whole bunch of other oxygens and hydrogens and carbons in it, but it's a large molecule. It has adverse effects, also include diarrhea, heartburn, stomach pain, hair loss, headache, rash, insomnia, bruising. And I thought this was interesting. It does affect clotting. And so you might want to consider stopping it if someone's going to have a large surgery. And they do recommend stopping it. Also, retinal toxicity. It mimics pigmentary pattern dystrophy. Approximately 1 million patients have interstitial cystitis in this country. This is US patients, 85% of whom are female. The symptoms include urinary frequency, pelvic pain, nocturia, causation. The causation for these symptoms are multifactorial, so it's not one particular pathway. It's associated also with fibromyalgia, IBS, and depression. So the paper that really broke the news on this was the paper by Pierce out of Emory. And they had 38 patients that they found on pentosan polysulfate in their service that presented to the ophthalmic service for genetic screening for genetic dystrophy. And 6 men were noted in that group between 2015 and 2017 with maculopathy, but they didn't follow the normal pattern. Their mean age was 60 years old. All of them had interstitial cystitis. And they looked at the mean cumulative exposure was 2,200 grams. Mean duration of exposure was 186 months. So this is a long-term use of this drug. And their mean vision was actually pretty good. It was 20/25. Most of the patients came in with complaints of having problems seeing near vision was their issues. Nyctalopia was their issues. Low contrast sensitivity was their issues. But they actually had reasonably good vision. I put the conversion scale from Logmar here to Snellen's. They're about 20/25. They had these subtle pericentral hyperpigmentation at the level of the RPE. They had kind of these surrounding vitelliform-like deposits in the RPE, and no eyes in this particular group had a CNVM. So this is what they kind of showed as far as their symptoms: metamorphopsia, paracentral scotoma, general dimming of the vision, near vision difficulties. And what I really want to show was, these patients had prolonged dark adaptation. Visual fields were pretty much not really very useful. They might have had some central scotomas, but some of them did not have paracentral scotomas, so you couldn't use the visual fields to really make the diagnosis. The full field versus multifocal ERGs, some had findings, some did not have findings. So there was no home run hit as far as making this diagnosis based upon diagnostic testing. And this is from the original paper, showing the range of disease of these patients. And these are color photographs, fundus autofluorescence, and near infrared. And you can see these kind of both white hyperpigmented and hypopigmented mixed lesions in these areas in this perimacular region. In this color photographs, you can see on the right-hand side here, this very large kind of like atrophic geographic atrophy type changes. And I think this is one of those things that, once you see this in your head, hopefully you don't forget it. It should be burned in. This is a very kind of classic type pattern. But I do believe that there's people that I do see come in that probably have this pattern that I don't think about interstitial cystitis because it's not the first thing on my list. It seems, quote, it seems unusual to speculate on a new drug toxicity 21 years after FDA approval, particularly given the dramatic and characteristic imaging findings of this maculopathy. And the one thing I thought was nice about this paper was the one thing that I ever read that wasn't described first by Lee Jampal. So Lee didn't get this one, but it does show that things are still out there for us to find and to discover, even at this point in our careers. So a little bit more examinations and studies were done. PANF examined 219 patients just with interstitial cystitis. They did a retrospective chart review, 2014 to 2018, and then they brought in the patients that they thought would be of interest. They had both with and without exposure to pentatosin polysulfate. 14 patients showed with classic findings, and of those 14 patients, when they did that analysis, the odds ratio of having maculopathy in this population was 11.25, which is huge. That's a huge odds ratio. In 2020, FDA updated its labeling to include language about drug-induced maculopathy. It's the government, give them credit, it only took them 2 years. I mean, that's pretty good. That's like, that's rocket speed for us. So some of the things, the color photographs can be very subtle. You might see some little perimacular pigmentary changes, but you can see this in the fundus autofluorescence, this hypo and autofluorescent peripapillary halo, which is very classic. In inherited maculopathies, that halo is spared. You don't see that haloing effect. The near infrared is these hyperpigment spots with prominent hyperreflectivity. And then the OCT, you can have perifoveal thickness, which correlates to duration of exposure. You can get retinal thinning, you get RPE atrophy. They're still kind of defining what are the characteristics on OCT, but these patterns that you see are very classic. And this is one that I really want to show was, you have to figure out what's the number one thing you want to look at. And the number one thing you want to look at is the near infrared. And I don't know how many people do near infrared on routinely. Obviously, Amani does, and Matt, you do. I don't really do it very often. I mean, I really have to kind of think about if I'm going to pull that trigger, but it might be a good thing to do because you can see in this patient here, the fundus looks normal, autofluorescence looks normal, and this near infrared, you can start seeing some stippling there in the center with that hyper reflectivity right there. OCT really did not have much changes in this patient. Again, showing that, you can't necessarily use one test. CNVMs may be present in these patients. They are responsive to anti-VEGF, so it does mean that you should still be looking for those things to develop. They can have these vitelliform deposits, and I really want to show this peripapillary hypofluorescent halo, which you can see kind of like around these nerves, and that is the distinguishing characteristic feature, is the peripapillary hypoautofluorescent halo. The disease, they're starting to look at some staging and how we can stage this. Mild disease is disease within the vascular arcades, which is just looking like atrophy. Moderate disease is extending not more than 2 disc diameters beyond the temporal arcade or non-central atrophy. And severe disease would extend greater than 2 disc diameters beyond the temporal arcade and have central RPE atrophy. That's at least one proposed grading system. Differential diagnosis, of course, you always have to think of AMD, pattern dystrophies, inherited dystrophies, maternally inherited diabetes and deafness syndrome, and pachychoroidal pigment epitheliopathy. The screening, they recommend color fundus photos just for baseline, fundus autofluorescence, OCT, and the near infrared imaging. The question is how often do you have to screen? One paper said screen when they start the drug and then maybe every 3 to 5 years afterwards. Another paper suggests do an annual screening just to get the patient into the habit, and so it's always part of the thing. I like the annual screening. I think that it makes us also more cognizant of what's going on with our patients and have more connections. So it was a very straightforward talk. Everyone wants to get going here, and I want to say thank you.
Thanks so much, Larry, for that great talk. Has anyone here seen a case of Pentacin? Yeah, so I think we all have, so it's great. I mean, the awareness is out there, and hopefully the patients will not suffer from this going forward. But the changes do continue, right, after you stop the drug, which is a problem. So without further ado, I'll close the meeting. I'd like to thank everybody today for coming. I had some slides here thanking everyone. Let's see if we can get that open here. And particularly like to thank my staff that helped me, Miss Jenny Baker, Lauren Kalinowski, Robin Tate Casper, and Patrice Wilbur for all of their help. Thank you so much. Couldn't have done it without you. Please note that there will be 15 days to request CME. Please complete the CME survey at the end of the meeting in order to receive the CME credit. I'd like also to thank all of our exhibitors without whom we could not have fed you today or flown in our outside speakers because we don't pay our inside speakers. These include Allergan, Apellis, Bausch & Lomb, Coherus, INUC, and OMIC. Thank you all. Thank you to all of my UIC colleagues and UIC affiliated speakers. And thank you all to my invited speakers, Anita Agarwal, Amani Fawzi, Deeba Husain, Matt McCumber, Dimitra Skandra, and of course, our Cless speaker, Phil Rosenfeld. You guys were great. All of you were great. Thank you so much. And hopefully we will see you next year on March 15, 2024. That is hopefully the date of the symposium. Thanks all and have a good night.